Good morning, everyone. Welcome back to KHUMANG 2020. Today is the day 3, and today is the last day. We have a very exciting day 1 and day 2. We have a different presentation from different parts of science and different part of, I should say, different research areas. Today we will have 2 technical sessions. Session number 5, which will be chaired by Dr. Puja Khare, and session number 6 will be chaired by Professor Rakesh. After that, we will have a valedictory function. In the valedictory function, we will be announcing the 5 winners of the Young Scientists Award. After that, we will have a vote of thanks, and we will officially conclude KHUMANG 2020. So let's move forward and let's start the technical session. Good morning, everyone. Am I audible to all?
Yes, yes.
Yeah. Good morning, everyone. Today is the day 3 of KHUMANG 2020. We have a successful day 1 and day 2. In terms of technicality, there were no issue from the speaker and from the oral presenter. And on day 3 and on day 2, at the end of oral presentation, there were some audio issues. So we will be sharing the oral presenter, which were not shown yesterday, today in between the 2 technical sessions. So after these few words, let's start the technical session number 5, which will be chaired by Dr. Puja Khare. Dr. Puja Khare is working as a scientist at Central Institute of Medicinal and Aromatic Plant, CIMAP. She has a work experience in the field of environment and energy, and her current research interest is working on the remediation of contaminated and infertile soil of aromatic crops. So with these few words, I would like to invite Dr. Puja Khare to kindly chair the session number 5.
Good morning to all. I'm audible?
Yes.
Hello. Yes.
Yes, you are
good
audible.
morning to all. Now we will start the session, today's session. So our first speaker are Dr. Barun Okram from GNF, California, and he is a medicinal plant chemist. So I would like to invite him. He will speak on modern drug discovery paradigm. Hello.
Dr. Thank
Barun.
you. Yeah, I can hear you. Can you hear me?
Yeah,
Yes.
yeah.
Thank you, Dr. Puja, for your kind introduction. Good morning to everyone. Here is Thursday evening, 9 o'clock. I hope everyone is doing well and this difficult time. Let me try to share my screen. Looks
Yes.
like there is some problem into the host disabled participant screen sharing something.
Okay. Can you try it now?
Oh yeah, now it is. Can you please see?
Yes,
the
it is coming now.
slides? And let me try to hook the. Laser pointer I want to use. Okay. All right, I will start from here. This is the picture from San Diego downtown, beautiful picture. But you know, even though it's very close to my place, I've not been there for the last 6 months due to this difficult time. I would also like to thank the organizing committee and then Dr. Ragumani from the BARC for reaching out to me, and I'm really delighted to participate on this conference and to share some of the science we do. Today, my talk is on modern drug discovery paradigm, discovery of Zacadia, a drug for the lung cancer. I was fortunate to be involved in the drug discovery program. Today, I will try to focus on with the consideration of the young students and also the young research scholars. Please bear with me if you are well familiar about the drug discovery and if you are in this field. As you know, we all have been touched by the magical or wonderful drug in our life, in the daily life, or whether in order to cure the disease, to improve the disease, or to improve our life. So the mission of the drug discovery is to meet the unmet medical needs and improve the quality of human life. I'll just try to start with some kind of historical background of the drug discovery. In the ancient time, we all know how rich were in the Vedic time in India. From there on, there are rich Ayurvedic, for example, for China, there is a rich Chinese medicines, and in other parts of the world as well. So at that time, these are all based on the plant and mostly herbal medicines. So with the civilization of human, along with the science hand in hand, we started developing or discovered many very important drugs in our life-saving drugs. For example, for pain relief, you have aspirin, morphine, and we all know the importance of the antibiotics. We all know how the penicillin was discovered, and then many vaccines to eradicate or to prevent many deadly diseases in the past. And actually, right now, we need one as well. So with that, I will try to highlight a few drugs, modern drugs that have been discovered by the modern technology, you can call it. First one is the Gleevec, which is again from the Novartis. It is a small molecule targeting a kinase protein called the BCR-ABL that is responsible for driving some type of the blood cancer, for example, AML and CML. So this is first of its kind as a targeted therapy, or you can call a precision therapy, because it tried to focus on only into the cancer cell without touching or minimal touching to the normal cell. So this drug itself is considered as one of the blockbuster drugs from our company. And then I'll move into the SID. We all know that this deadly disease have been impacted to the whole world for the last several decades or so, a real burden to the human society. But we have been discovering very good, effective drugs from year to year with improving properties or effectiveness. Now, I would say any HIV patient can live life as long as a normal life with good health, even though this drug cannot cure the disease, but this can really control the disease very effectively. But in the near future, there should be eradication to cure the HIV. So this is one of the landmark, I would think. And then in currently, the emerging approaches I wanted to highlight, very, very promising to tackle many difficult diseases like cancers. First is the ADC, the name suggests, which is antibody drug conjugate. So it says you have the one antibody, you have the drug, and then conjugate them. So in this case, what we do is that what the scientists do is that they select a particular antibody that identified or recognized the antigen of a particular cancer, and then hook it with drug. In this case, we select the most potent, you can call it the poison. But once it is conjugated to the antibody, the whole cargo is now safe, not toxic. So once it is released into the system, the antibody will we'll take the cargo into the antigen of the cancer cell, and then we'll internalize and clip and release the drug, and the drug will attack to the cancer. Very, very promising technology, but there is a still flow. So many companies are working to improve how to safely journey of the ADC to reach to the cancer cell. In between, if it clips, then it will be very, very severe side effects. The second one, all we know, immunotherapy, gene therapy is very, very promising and then very well-known research field, hot area in the scientific community, both in the academic and the industries. So I will just try to highlight here the Kymriah is the CAR T cell which was approved in the 2018 for a subset of the blood cancers. Very, very effective. Again, this is from the Novartis. I'm sure many people are aware how it works. So what happened to the cancer patient is that when the cancer cell grows, your immune system, the T cell, try to attack the cancer cell, but it could not recognize anymore because the cancer cell, in this case, is very smart. They try to disguise, and the T cell cannot, identify anymore. So what happened is that we take out the blood from the patient and then purify into the T cell, the white blood cell, and take it to the laboratory and then engineer it into the CAR T cell. Now bring back to the hospital, infuse it to the patient. Now the patient is living with the living drug so that this CAR T cell can keep attacking to the cancer cell and kill it. So it's just the beginning. What happened is that right now, it's more than 3,000 clinical trials, both from the immunotherapy and the gene therapy, including the combination therapy, are still going on for different types of cancers. The third one, protein degradation, this is another very emerging approach to tackle the cancer and other disease. For example, when I said about the Gleevec, what Gleevec does is that it inhibits or it binds a protein that is responsible for the disease. In this case, instead of inhibiting, simply taking the advantage of the proteasome called the subset of the protein, which can attract the other interests of your protein and simply degrade and then trash it. So these are very promising. And another one is the system therapeutics. As an example, SID, there are, instead of the one drug, you give the 3 drugs. That is very effective. So instead of targeting one target, you try to find out the networks and you shut down the networks, and that's how it works. So we have this emerging process, and I'm sure in the near future, it's very promising to tackle especially difficult cancer. But nevertheless, there are challenges predicted by many scientific community that the emergence of the resistance to the antibiotics, we call it the superbugs. Imagine if the antibiotics doesn't work to us, what we'll do from a small injury to a difficult disease. That will be really, really challenging attacks in the near future or the longer future. So the scientific community already started working on how to tackle that. And then we have cancer. From nowhere, we have a pandemic coming out, COVID-19. So we have to be prepared to fight these challenges in the future. Just try to give you the recap what I said. Before 1950, it was mostly herbal medicine, and then paradigm shifted. After 1950, it's most about the chemistry and biology. We have just discovered many important drugs. These drugs were not enough to tackle some of the difficult disease, and the paradigm again shifted from the 2010. Then we started working on looking into the pathology of the disease and the working on the pathology directed, for example, human genetics, molecular biology, systemic biology, etc. So these are the preparation from the scientific folks for the future challenges. Now I will switch gear into the process and highlight the difficulties of the drug discovery process. FDA, Food Drug Administration, which is the federal agency in United States. They are responsible for approval drugs to bring into the market. But in the last few decades, they have really tightened the regulations of the approval. The reason is simple. They really consider the safety of the patient for giving the drug. Once it was quoted by a very well-known toxicologist, Paracelsus, all drugs are poison, dose matters. This was many hundreds of years ago. So that is true. More than 500 approved drugs have been withdrawn due to the side effects so far. The side effects encompasses from the hepatic, hematological, gastrointestinal, dermatologic, carcinogenicity, respiratory, and so on. So this set of the huge landscape gives you an idea to us while you are developing a drug so that your candidate is free or minimal if, side effect from this information. I will not, I'm not trying to scare you. I'll just give you a few examples. A drug called the Vioxx, it's the inflammatory drug approved in the 1999, withdrawn 2004. The drug was available in the market for hardly five years, and then it found that it's linked to the 28,000 patient heart attacks in the United States. So the company voluntarily withdrawn at the time. And the second one is for the acne drug approved in 1982, withdrawn in 2009. It's pretty much more than 15 years were available in the market. And they found that it's linked to the birth defects, miscarriages. Now, the 7,000 patients filed a lawsuit. And then out of that, 3 patients, in favor of the three patients, and they get $10 million each. And then finally, the company was voluntarily withdrawn. And very recent drug for the weight loss called the Valbic, approved in the 2012 and withdrawn in the 2020. It's again linked to the cancer. The comparison here is 7.7 with the drug, 7.1 with the placebo. So only difference is the 0.6, but it is cancer. So again, the company Arena voluntarily withdrawn. I'm just trying to highlight how safety is important for the drug discovery, in addition to the effectiveness of what you are developing the drug for that particular disease, but equally you have to consider for the safety too. Here I'll try to bring one very common terminology during the early drug discovery program, also later on. So if you take the 2 drugs, you have to consider that 2 drugs, the effect would be different from you take the 1 drug. So the concept is that one. So here I'll give you the couple of examples. Grapefruit juice, for example, this is vitamin C, nutrients, it's good for health. But in certain cases, if you take certain drug like the cholesterol lowering drug, Lipitor or Jacozole, then what happens is that when you take the drug, the normal case, there are enzymes, particular enzyme in the intestine that breaks down the drug. And then according to that data, the dose was met. But in this case, if you take a lot of grapefruit juice with this particular drug, what happens is that the juice will block the enzyme, and then it fails to break down the drug, and then you will get a lot of drugs that will give you severe side effects. In the opposite case, the very well-known allergy drug, the Alligraf. So if you take the Alligraf with the grapefruit juice, what happens is that without the grapefruit juice, there are transporters in the body system who will transport the Alligraf in your body. But if you take with the grape juice will block the transporter, and then it fails to transport to the body, so you will get a very little amount of the drug in your body. So probably, even if you take the drug, it may not be effective. And the second, I think I just brought this one. It could be relevant to our society too, if you take with the herbal and the drug interaction. So herbal drug known as St. John's wort. If you take with the HIV drug or heart or birth control drug, it might give you, side effects. And then second one is pretty interesting to me. I just read it. And then aspirin is very common drug, right? With the ginkgo biloba, which is also known to us in Manipur as a langtray. So if you took together these two, probably you will have the liver injury. So the whole point is, if you take the herbal medicine and if you go to the doctor or your physician, it's very important to tell them that you are taking that herbal medicines. Accordingly, doctor will decide the prescription. So then I'll jump into directly the process of the drug discovery. It is risky, expensive, and time-consuming. Different stages start from the discovery and the preclinical, lots of candidates, and then you optimize. Once if you find a preclinical candidate, it goes to the clinic phase 1, 2, 3, and the whole data package you submit to the FDA, and FDA review it, and if it is approved, and then registration, and then to the high production becomes available in the market and ultimately landed to the patient. So it is estimated that for developing one drug from starting to the market, it's around close to $2 billion and very less success rate, 1%. 48 new drugs were approved in 2019 and in the US. You can imagine how much money to develop those more than $70 billion. Not only that, it can again take more than 10 to 14 years from starting to the market. Now, I will bring the concept relevant to the recent crisis. Why we hear about the hydroxychloroquine, remdesivir, favipiravir instead of developing new drugs. So we cannot start from the beginning to take the 10 years to treat for the COVID-19. We need the drug immediately. So the company or the academic or the scientific community, what they do is that they are looking into the existing drugs. If that works, then these drugs have already profiled phase 1, 2, 3, and then it can bring to the market very quickly. That is the whole idea behind that. This is the steps of the discovery and the preclinical. I will not go through all details, except I'll tell you that it consists of several people from the different expertise, biologists, chemists, etc., to work on a particular project. Very two important term in terms of the safety and effectiveness, pharmacokinetics and pharmacodynamics. What happened is that if you take the drug from the mouth or from the IV injection, it's important to consider how much absorption, how much distribution, how much metabolism, how much excretion, and then how much it will affect to the receptor site concentration, and finally clinical response therapeutic outcome. That is the pharmacodynamics. So this whole each term is considered when you do the the optimization of the drug, including the potency, PK properties, efficacy, and the PK/PD relationship. So now we have the preclinical candidate. Then it will go to the clinic, and then we have the 3 stages: phase 1, phase 2, or phase 3A, 3B is the final stage. So the phase 1, generally it goes to the 20 to 100 healthy volunteers. It also depends on the type of the disease. So here you are not giving the drug to the patient. Here you are giving to the healthy volunteer for the sole purpose is the safety and the doses. So here it takes almost 1 year, and then only, again, it's a high percentage, 70% success rate goes to the next phase. Once into the phase 2, it expanded to the 100 to 1,000 patients. Now it's the real patients, and then almost takes the 2 years. Only just 32% success rate moving to the next phase. Why? The key reason is that in the preclinical, whatever you study is the animal model. Now it's coming to the human patient. So there is always disconnection, and the scientists these days trying to narrow down this disconnection is another very key research going on. Then once you pass to the phase 2, phase 3, it's again expanded and the rigorous testing. it's again 1 to 4 year, only 25% success rate. And then the whole package is submit to the FDA, and then once they approve, then it will go to the market. So now I'll try to bring here another related topic. So how many vaccines candidate now goes to the phase 3? I think it's 6 to 9. So when we hear from the news or something like that, the vaccine candidate goes into the phase 3, we are really excited, but we really need it. So, but you also think into this data is 25%. So it is not that surprising that 2 days back, the Oxford AstraZeneca vaccine candidate just post is not terminated due to the fact that they find one adverse event to one particular patient they are working on there. So I just wanted to highlight the difficulties and then the route of the drug discovery here. So very high attrition rate. So what happens is that into the preclinical stage, we try to develop a very high quality preclinical candidate so that it reduces this attrition rate. I'll just check the time, 24. So from there, I'll just switching gear to different, coming back to the story of the Zycadia. So in overall, career in the drug discovery industry is very challenging, frustrating. At the same time, it's very exciting. So if you're interested in the biomedical science, desire to help human society, and a very important factor here is the team player. It's a little different from the academic. You have more freedom. Here, if you fail together, success together. Just wanted to advertise a little bit about the Novartis. The CEO is Vasan Narasimhan. Vas is an Indian origin, MD from the Harvard Medical School, master from in the public policy from John F. Kennedy School. He was appointed in the year of the 41 years old, one of the youngest CEO in the big pharma. And the NIBR is a part of the R&D part of the Novartis, Novartis Institute of the Biomedical Research. The president is the James J. Bratner. It's again the Harvard product. Former professor, and again, he was appointed in the year of when he was 42. So it's a very exciting time, very young, dynamic scientist in the lead to work at the Novartis at the moment. Where I work is a ZNF, is a part of the Novartis. It was founded in the year of the 1999 by Peter Schultz, very well-known scientist in the field of chemical biology. He was my former postdoctoral supervisor, and he left, and now currently Jerry Joyce is institute director. He's again the professor from the Scripps, well known in the field of RNA biology. We have around 550 employees. The aim is to provide first-in-class therapies and giving the new drug target and the new drug preclinical candidates to the Novartis pipeline. In a very unique area, the behind the scene, what you are seeing, the blue is the Pacific Ocean, and surrounded by many well-known industries. Institutes like the Salk Institute, where polio vaccine was invented, the Scripps Research Institute, Burnham Research Institute, very academic or very scientific kind of atmosphere around the company. It's very unique setup. Integrated expertise consists of chemistry, biology, and the support from the protein science, project management, engineering, etc. And it encompasses different diverse disease areas, starting from the metabolic disease, oncology, immune regulation, infectious disease, regenerative medicine, pretty much cover everything there. When Peter Schultz founded this company, he hired many engineers, and people were surprising. But now later on we realized that our engineering group had invented many robotic screening robotic system capable of screening 2.5 million compounds per week, and it's also capable of complex biology screening, for example, the three-dimensional assays, disease-related cellular assays derived from the blood, patient blood, live pathogen, parasite, fungi, bacteria, screening, etc. So we also sell this robotic system to the other big pharmas, GSK, Pfizer, Merck, etc. So now the story of discovery of I'll just give you as a case study. The drug is called the Zycadia for a subset type of the lung cancer, is a non-small cell lung cancer. It was approved in the 2014 April. I was fortunate to be part of this drug discovery. Just to give you a little bit of the non-small cell lung cancer is one of the top deadliest cancers in the world. So already 228,000 new cases, 135,000 deaths already in the 2020 in the US. Just give you the treatment options, either it would be surgery, radiation targeted therapy, immunotherapy based on the stages of the disease you have, whether it's stage 1, 2, 3A, B, and the final stage 4. So accordingly, doctor will decide the treatment according to the stages. So we are looking at the targeted therapy, the Zycadia, targeting a protein called the ALK. So the targeted therapy is that we all know how horrible the chemotherapy treatment. The reason is that in addition to targeting the cancer cell, it also kills the normal cell. That is obvious. So, but if the targeted therapy is, if you have the right choice, it could be very, very well tolerated than the chemotherapy. So this Zycadia is the targeted therapy for the ALK. I'll just give you the landscape here. It will be very, very interesting here. So 49% still we do not know, no mutation. 25% KRAS is a big chunk of the percentage. So many companies are working to target this KRAS, very difficult target. There are few drugs already available for the EGFR, 13%. Now the Zycadia is targeting only 5%. But if you look into the 135,000 that you know, the 5% is pretty significant. I will not go through all the biology here, but that's just to highlight that the ALK, the protein, is not expressed in the normal tissues, strong oncogene, rare and common disease, 5%, and it is the opportunity to expand into the other cancers. Strong medical need for cryogenically relapsed patients. So here I do not have time to explain everything. The reason is that there was already one drug for the ALK positive cancer called the cryogenically, but it doesn't work very well. It's relapsed to the patient very quickly. So they really need for the second line therapy. So the Zycadia is for the second line therapy. Here I'll give you very quickly how we tackle the two real problems during the development of this Zycadia. So one, before that I'll forget. So after working almost 3 years into from starting to the optimization stage, we thought that we reached two candidates. One is the TAE 684, one is the GNF 0912. And then we thought that we have a very good quality preclinical candidates, but later on after profiling, we realized that two problems. One is it forms the reactive adducts into the middle structure here. It's oxidation into the body. And then this is very reactive. So inside the body, the GSH protein can form the covalent adduct, and then now the drug is not working anymore. In addition, the second one is that it also gives on the glucose homeostasis. This is another side effect, we think. But at that time, we think that these candidates are also hitting insulin receptor that could be potentially driving this side effect. So basically, at that time, we were almost given up because this is a serious problem. We cannot take this one to the clinic. But science is all about the perseverance, hardworking, never give up, that kind of attitude. And then we try to look into the structure of this compound, TAE 684. The A is the structure protein ALK, where the TAE is binding into the binding pocket. I think my pointer is not working, unfortunately. So what happened is that the B is the same. B is the protein, is the insulin receptor binding by the TAE. So we were trying to mapping the differential residues between the two proteins. And we found that L258 is smaller than the F258 from the insulin receptor. So in this area, so the methyl group, methoxy, can fit into this both the protein. So that's why it's very active in the both receptors. So what we realized is that why not we increase the size? So instead of methyl, we change into the isopropyl. Now isopropyl still fits with the ALK, but it clashes with the insulin receptors. That's how we reduce the activity of the insulin receptor. So that's how we tackle the first problem, but we are still having the second problem, the adduct formation. So we went back into the whiteboard and the lots of brainstorming and then reaction mechanisms, etc., so on. So there's many strategies we have applied. So one of the strategy, very simple one, why not we remove one of the nitrogen by replacing by the carbon? So that is the success of the Zycadia. Simple modification to solve the problem. So I'll show you a couple of animal model here in the mice, two cancer model. So the black one is the vehicle where you can see the tumor is growing, tumor size along with the days of the treatment. So the blue-green is the doses are increasing. Please concentrate onto the green and the dark. I cannot even see the violet. So it's a 12 and 25 milligram per kg. So what happened is that the tumor is totally, completely 100% reduction. I mean, the regression there. And on the right-hand side, it's also for the lymphoma model, it's the same case. When it's treated with the 25 mg per kg with the Zycadia, it's almost 93% reduction. So it's very exciting data in the animal model. That's the reason we moved forward. And then again, the left-hand side is not in the mice, here now with the rat. And again, 25 mg per kg, the green, if you look at, is completely regression. This is very exciting. The data into the clinical trial. The left-hand side, whatever you are seeing is black one is the tumors, cancer cells. It's pretty much this patient is pretty much spreading by the cancer beyond the lung. They call it the metastatic. And then now after the treatment of the 3.5 week, I think it's the 150 milligram per kg, 150 milligram doses per day. After 3.5, you see a dramatic reduction of the black spots, the cancer cells. So that's the reason. That's the reason why the FDA gave breakthrough therapy, meaning they can accelerate the clinical trial in a very fast manner. That is also depending on the need of the patient, and you can imagine how many people, new cases in per year, how many people died. So a little bit, you know, the effective will be a great deal. So we started, IND filing was the 2010 and then approved into the 2014 as a second line therapy. I mentioned the first line therapy is the crizotinib. Crizotinib doesn't work very well. If it doesn't work, then we give the Zycadia as a second line therapy. So again, taking the 5 years to compare between the Zycadia and then and the crizotinib first line therapy. So recently in the 2019, now FDA approved again, reapproved again Zycadia as a first line therapy for non-small cell lung cancer. So this is the story of the discovery of Zycadia. It took a long time, a lot of effort, a lot of money, but at the end, it was a success story. So this is the acknowledgement. This is the whole team. It's the up to the preclinical stage. Beyond that, again, hundreds of people involved to discover this drug. Probably you can see me here somewhere. So with that, I thank everyone for your kind attention. And if you have questions, I can answer, or offline you can ask me or send me email or anything, you can get the email from the organizing committee as well. Thank you, everyone.
Now forum is open for question answer. Hello.
Yeah, you're audible. If anybody has any question, kindly raise. Yeah.
I have a question.
Please.
You have any idea about the doctors not concerned about the patients? When the patient complains that when he takes the medicine, he has more other problems.
Definitely. For example,
They don't like to concern. They are doing just what you tell only, or how they are doing that?
that is the sad part. The problem is, I mean, if you really abide the law, it is very dangerous. For example, I work for the Novartis. If patient come and to me and said that, hey, Barun, I take this drug made by your company, I have some headache here. So if I don't report to the agency within 24 hours or 48 hours, I can be in big trouble. So that kind of the strict law has to be implemented, but you know, it's sad that it doesn't work everywhere. That's the problem.
Yeah, this is what some of the doctors in general, okay?
Yes.
They want that particular part only should be solved, not the consequence of that, okay?
I totally agree.
When treated for chemotherapy, I think they're given extra dose, okay? They are the radiation therapy was so concerned, okay? But the effect be come to the patient, you know?
I totally agree.
Kind of thing, you have any idea about how this psychology goes on among doctors?
I think this has to be dealt by the lawmakers. I guess it's not by the scientists.
Yeah, okay, good.
But I understand that is the real problem. I mean, it happens to everyone.
Anyway, can I ask another question?
Yeah.
Yes, please.
How you decide that, how heck happens? What is the reason behind it?
Can you please
any
repeat again?
Of the artery or vein or whatsoever, because when you have something only there comes.
So you are talking about the if a patient
mechanism how this headache happens.
Oh, it can be many factors to get someone get headache, right? It can be, I mean, indirectly it can be coming from different disease, but it's of course it's your blood system or the blood circulation that impacted. But I'm not a doctor, but I'm not a person to understand that.
It's all right. It's all right.
Yeah.
I think it will take another full session to explain how actually a headache happens.
Yeah.
So, Dr. Puja.
Yeah. So thank you, Dr. Barun, for nice presentation and for giving us information regarding the drug development, and also the development of the area. Thank you, sir. So now
It's my pleasure. Thanks a lot.
Now I invite Dr. Akshay. He's from SCID, Florida. He's a specialist in infectious disease. He's MBBS from Manipur University, and he will speak on bacteria versus humans. Dr. Akshay, please. Dr. Akshay.
Yeah, he is online. He will be sharing.
Okay.
Can you hear me now?
Yeah, we can hear you.
Okay, thank you. Dr. Okram, thank you for that. That was a really fascinating presentation on how medications are made. It applies to my presentation a lot as well. So I'm going to share my screen.
Okay, you want me to share the screen from my side?
Oh, no, I'm doing it.
Okay, that will be nice.
Just let me know if it's...
Yeah, it's coming.
It's coming up, okay. Okay. Thank you. So thank you for the introduction. My name is Dr. Akshay Manohar. I'm an infectious disease physician. I was trained in both clinical medicine, and also I did training and research. And right now I'm more practicing in clinical medicine. So the presentation I wanted to talk about was bacteria versus humans and the struggles of our species that we've had to overcome theirs. So wanted to share certain interesting facts. So our bodies, you and me, we are made up of around 30 trillion human cells. Now, our bodies are made of 30 trillion human cells, but in each and every one of us, there are 38 trillion bacterial cells on average. So 8 trillion cells outnumber. But if that number sounds pretty high, there's an even higher number, and there's 380 trillion viruses exist in each and every one of us at any given time of our life. So the other question to ask, our bodies, who does that really belong to is one question. The other one is, who does our planet really belong to? And so the first single cell organisms came to be about 3.8 billion years ago. Viruses about 3 billion years ago, but it may be as old as life itself. Multicellular life for the first 2 billion years did not exist, and around 900 billion years ago is when multicellular life began. Fungi began 600 million years ago, and the first vertebrates, which we belong to, was 500 million years ago. Now we're going from millions to hundreds of thousands, and the first human was only 300,000 years ago. Penicillin, just so you know, the one that kills bacteria, that was only put into use in 1942, was discovered earlier, but put into use only 78 years ago. So this number is really fascinating because for most of us, you and me, we belong to these first two digits, maybe the third if we're very lucky. In these four numbers, all of human history, all of our poetry, our cultures, our religion, everything belongs over here. And beyond that is how long bacteria have existed and owned this planet before we came to be. So I wanted to show an example of what infections look like. So there's 3 infections here, and these 3 infections have one thing in common. They have a pus, okay? So this first one, if you look over here, this is what a normal bone looks like. And you see this area is an abscess. It's called a Brody's abscess. It's a bone infection. And if you look here, the same thing seen on this CT scan and the same thing seen on this bone over here is basically an abscess. Now, what's interesting about all this is this paper was published in 2019. This is an abscess in a 14-year-old girl. This is an abscess from 2,500 years ago. This is a CT scan of an ancient Egyptian mummy of a man who lived in 500 AD. And this is from 200 million years ago. This is a dinosaur with the same type of infection, the same type of abscess. This probably came from a bite, not the same way that this is spread. This may have probably been spread by blood. But if you look closely, you can see there's a white ring around this, the way the bone is remodeled. And the bone is remodeled here. And then if you look closely at this, and unfortunately this is not a zoomed image, but there is some remodeling that was seen. So this dinosaur actually lived for a little bit with this infection. So the infection burden around the world in 2001, infectious diseases caused about 14.7 million deaths on one source. And the global data for sepsis in 2017 showed that there are 48 million infections, or 48.9 million infections, 11 million sepsis-related deaths. So what is sepsis? Sepsis is a very severe bacterial infection that gets into the bloodstream and leads to drop in blood pressure, gives you the classic signs of infection like fever and so on. And what it does is that it actually, it's probably the worst infection, and a lot of the infections are caused by something called a gram-negative bacteria, and we'll talk about that in a minute. So this is the data from India in 2017. If you see here, the big boxes mean that they're the bigger causes of death. Lower respiratory infections, TB, and diarrhea are the biggest causes. And this is actually in 2017, the data for both sexes and all ages, the causes of deaths in Manipur. And it was lower respiratory tract infections, TB, diarrhea, and HIV. So infections are caused by different things. They're caused by viruses, like the coronavirus that we're dealing with now, and HIV, bacteria like streptococcus and staphylococcus tuberculosis, fungi like yeast and molds, parasites like malaria, insects. We usually call insect infections of our cells, we call them infestations. And there's also something called a prion, which is it's a molecule that causes an infection of the brain. It's not necessarily a living organism, but then again, what is life? One of the things that it does, like mad cow disease, is an example of that. So let's talk a little bit about antibiotic resistance. So bacterial resistance to antibiotics is, people sometimes talk about it as a problem of the future, and it's not a problem of the future, it's a problem of the present, and it's a problem of the past. It's been ongoing for so many years, and it's been getting worse. And what we need to understand is that this is not bacteria are not conspiring together to say, hey, the antibiotics are here, we're going to fight them. No, this is actually evolution in motion. And usually we used to have natural selection. Now we have artificial selection. So artificial selection by us, by creating antibiotics, because we're killing off there's many sensitive strains of bacteria, and the ones that are left to survive and reproduce, the ones that are not able to fight against them. So the causes of antibiotic resistance, this is from the WHO. This is actually a big issue for the World Health Organization as well. Antibiotic overdose is one of the biggest causes. It's either because doctors are overprescribing antibiotics for things that don't need infections, or the general population is self-treating because they have free access to antibiotics and without the need of a prescription. There's also patients who are not finishing their treatment. There's overuse in livestock and fish farms. A lot of livestock and fish farms there's use of antibiotics in those cases. And there's poor infection control in hospitals and clinics. And this it happens all the time. I mean, I saw it happening in my own hospital once, where one person got an infection with a very resistant bacteria, and then 2-3 other people also got it. And they all were in the same area, and it was because people were not washing their hands. And so lack of hygiene is another problem. The other problem, as Dr. Okram mentioned, was there's also a lack of many new antibiotics being developed. So where do antibiotics come from? So in the year 1928, this is a pretty famous story, penicillin was discovered. And I like to use the word discovered because that's what it was. It was not invented by us, it was discovered by us. And Alexander Fleming he was looking at Staphylococcus, and he had a petri dish very similar to this one. And it had Staphylococcus bacteria colonies spread all over it. But what happens is that, and when, if you've worked in a lab with microbiology lab, you often find these plates, because they're so rich of nutrition, they end up getting contaminated by molds from time to time. So a colony of penicillium mold, and penicillium is very common. If you have bread lying around for too long and you see mold on there, a lot of times that actually is a penicillium species. So it contaminated it. But what was interesting was that you see that the bacteria is growing very, very well, but around this penicillium mold is a halo. And this is an area where penicillium is injecting penicillin into the area. And that's why the bacteria was getting inhibited and it's not able to grow. So where do we get antibiotics and where do they come from? So penicillin comes from penicillium mold, cephalosporin from cephalosporin, carbapenems from streptomyces, fluoroquinolones is actually from, it was discovered on accident when they were looking up the cinchona tree and trying to get chloroquine from them. And vancomycin comes from what was used to be called Streptomyces orientalis, and clindamycin from Streptomyces lincolnensis. Tetracycline again from another Streptomyces species. So what we have in common here is that most of our major antibiotics are actually coming from from fungi, and they come from plants as well. So antibiotics were discovered less than 100 years ago. Penicillin 1928, fluoroquinolones 1960s. But antibiotic resistance is not new. Antibiotic resistance is old. So how old? So I said penicillin was discovered in 1928, but 1915, there was an isolate of Shigella that was revived in sequence in 2014, and it had resistance to penicillin and and erythromycin already. There was gut bacteria in a Peruvian mummy from around 1080, and that already had resistance to beta-lactams, to aminoglycosides, vancomycin, quinolones, and so many other drugs. And bacteria from a 30,000-year-old permafrost was found to be resistant to beta-lactams, tetracyclines, and vancomycin. And the mechanisms of resistance are very much the same and found in bacteria today as well. And to top that, there's bacteria from a cave in Mexico that had been isolated for 4 million years, and that was found to be resistant to 14 different commercially used antibiotics. So the questions that I'm trying to address here are: how do common antibiotics work? What are the mechanisms of resistance? What are the latest antibiotics, and what are the possible methods that we can use to overcome resistance and create new antibiotics? So let's start with how they work. So antibiotics work by many different ways. Some of them bind to ribosomal subunits, which are required for synthesis of various proteins. And this inhibits cell growth and sometimes even causes cell death. And so a classic one is something like azithromycin. They can block DNA replication and hamper the bacterial DNA synthesis, such as ciprofloxacin. And they can inhibit the biosynthesis of the bacterial cell wall. So penicillin, cephalexin, meropenem, vancomycin. By the way, these are the biggest drugs that I use in practice and many doctors use in practice. And this is probably the biggest way that we use to kill bacteria. So binding to the ribosomal subunits, we talked about that. So it basically blocks protein synthesis for the manufacturing of important proteins that are needed for bacterial survival. And so the bacteria cannot grow and sometimes they die. And when DNA replication is blocked, during cell division, DNA cannot make proper copies of itself. And so the bacteria cannot reproduce and they die out. And the inhibition of the production of the cell wall. So many bacteria contain a cell wall, not all bacteria, and that's why you need the other medicines. And cell wall synthesis is inhibited by drugs called the beta-lactam drugs, and I'll talk more about them. And those are penicillin, cephalexin, meropenem, aztreonam, for example. So without a protective cell wall, what happens is that the bacteria becomes subject to osmotic pressure. It doesn't always die, but many times it does, and that's so that's why the cell wall is so important, and inhibiting it is one way how we've been able to overcome them. So what are the mechanisms of resistance? And so I have these little cartoons that I draw for my medical students sometimes, and it sort of helps understand what's going on. So antibiotic entry is denied into the cell. So that happens a lot of times with drugs like penicillin, and they're not able to enter certain bacteria because the bacteria like gram-negatives have a lipopolysaccharide layer that protects the cell wall from within, protects the cell wall within. There can be a change in the structure of porin channels, and that prevents the entry of the antibiotic into the cell. So this is, for example, what a gram-negative looks like, and this is the lipopolysaccharide layer and some of these porin channels that prevent the entry of the antibiotic. There's another way, which is that you're able to push the antibiotic in, but then the bacteria says, thank you very much, and then it pushes it out again through a pump. And blocking the pump may sometimes help get rid of allow the antibiotic to then work on the bacteria. Oftentimes, and this is sort of the biggest way that bacteria have resisted antibiotics, is that we put the antibiotic, it gets into the cell, but then beta-lactamase is a protein molecule that's produced by the bacteria, and it destroys the antibiotic that's actually inside it. And so that's how it's able to survive. And another way is that the bacteria is able to get in, it doesn't get destroyed, and it goes into the target site, but then the target site has another affinity. And so the bacteria can no longer work on the target site, and then the bacteria survives. And the biggest classical example of that is a protein called PBP2A, and that's found in bugs like methicillin-resistant Staphylococcus aureus. So just a little small introduction about bacteria. So bacteria, it's hard to see them by themselves under a microscope. You have to stain them. And so there was a gentleman by the name of Hans Christian Gram, and he created a method of staining, and they called it the Gram stain. So he used crystal violet and safranin and found that bacteria stain either purple or they stain pink. So purple, we call it Gram positive. If it's pink, we call it Gram negative. And this is actually probably the most useful differentiation of bacteria, because this has differentiated them into ways that when I see a patient, I can predict sometimes by looking at the infection itself, whether it's a gram-positive infection or a gram-negative, just by the way the patient presents. and because the treatment can be different for both of them as well. So for example, this is the cell wall of the gram-positive, and because of the lipopolysaccharide layer, the cell wall doesn't get stained, and so the it looks pink. So the other way to differentiate bacteria is by the shape. So circular organisms like this are called cocci. If it's a long stick, we call it a rod, like a stick, or we call it bacilli. And examples like we talked about, gram-positive cocci, Streptococcus, Staphylococcus. Gram-positive rods are anthrax, tetanus. Gram-negative cocci, which I don't have on the slide, they look like this, but they're usually two together, meningococcus, which causes meningitis, and gonococcus, which causes gonorrhea, gram-negative cocci. And then there's gram-negative rods, which are bacteria like E. coli, Salmonella, which causes typhoid, Acinetobacter, and Klebsiella. So these organisms, the gram-negatives, are mostly found inside the gut. Gram-positive cocci are mostly found on the skin, in the oral flora, and in the mouth as well. So the cell wall of bacteria, this is both gram-positive and gram-negative bacteria. It's made of a peptidoglycan layer, which allows the bacteria to resist the high osmotic pressure. And these are made up of a disaccharide monomer of N-acetylglucosamine and N-acetylmuramic acid pentapeptide. So what happens is there's a cross-linking that occurs with these two chains, and that's what gives the stability and the structure to the cell wall. So there's pentapeptides. They're made of alternating L and D stereoisomers, and they terminate in something we call diala-dialase, dialanine. And this is a very important site, because this is where there's a certain, for these two to join, the diala to L-lysine, there is an important protein that's involved in that reaction to cause the bond, and that's called PBP. So what is PBP? It's penicillin binding protein. And why is it called that? It's because penicillin binds to that protein. So PBP, it's a stem peptide, and it attaches to the L-lysine component, which in turn binds to the diala by an amide bond, and that's catalyzed by this transpeptidase. So when this, how does penicillin work? So penicillin and similar drugs, they bind to the penicillin binding protein with the help of a ring in the molecule called the beta-lactam ring. And the beta-lactam ring is found in 4 antibiotic categories. It's found in penicillin, cephalosporins, carbapenems, and monobactams. This is what the beta-lactam ring looks like. It is this a nitrogen atom. It's attached to the beta carbon atom that's relative to the carbonum. So how do they work? So basically, we talked about what the penicillin binding protein does. So when the beta-lactam ring attaches to the penicillin binding protein, these two chains can no longer attach, and then the bacteria gets exposed to osmotic pressure. So we talked about one form of resistance was PBP2A earlier, and that's if the penicillin binding protein 2A is present instead of the other penicillin binding protein, then the beta-lactam ring can no longer attach to the target site, and then the bacteria retains the cell wall and can do a lot of harm. For the biggest example is MRSA, methicillin-resistant Staphylococcus aureus. So what's the difference between gram-positives and gram-negatives? So gram-positives are made up of cell walls that are about 50 to 100 molecules thick. Gram-negatives is about 1 to 2 molecules thick. So technically, penicillin should work better on a gram-negative, but why doesn't that happen? Because it has this lipopolysaccharide layer, which is kind of like a force field. It doesn't allow antibiotics or many things to enter. Again, that's a picture of what they look like. So it's a thick layer that covers the peptidoglycan layer. It's made of tightly bound hydrocarbon molecules that block the entry of hydrophobic antibiotics. And hydrophilic antibiotics enter through porin channels. So what is a porin channel? It's a beta barrel protein, but it's the way I like to think of it is like this. The gram-negative rod is like a fortress. It's impenetrable. Nothing can go in, nothing can come out. But the bacteria needs to survive. So what it has is secret tunnels and doors through which things can go in and out. And that's what a porin channel basically is. And there's other different channels where things are able to come in, like iron, and so on. So what did we do is we added an amino group to penicillin. When I say we, I mean humans, not me personally. And that allowed the penicillin to now enter into the cell and destroy the gram-negative. But then what happened was, as soon as this whole thing happened, we ran into a problem with beta-lactamases. So beta-lactamases are some things that destroy the beta-lactam ring found in all of these antibiotics. So even though they went and they got destroyed, and the antibiotics stopped working. So beta-lactamases are they're actually penicillin binding proteins. They covalently react to the beta-lactam ring on the antibiotic, and they quickly hydrolyze it. And it can either be encoded on the bacteria by chromosomal genes, or it can be transferred from other bacteria and plasmids, or by transposons, which are basically a cut paste or copy paste genes from one segment to another segment, and that helps produce the beta-lactamases. So there's different varieties of them. These are the biggest problems we face in clinical medicine. There's named after the drug that is a penicillinase. It destroys penicillin-based drugs, and carbapenem and cephalosporinases. And there's many different types. Carbapenems we consider as the last line of defense, and that's what we're having a lot of threats to. Recently, I mean, the past 10 years or so, there's something identified called the New Delhi metallo beta-lactamase. It's not specific to India, but it was just first isolated in an Indian strain, and that's why it's called New Delhi metalloprotease, which the Indian government doesn't like the way it does, because it makes it look like we have all the bad bacteria, the resistant bacteria in India, and that's not true, because NDM1 is found in multiple countries. Metallobeta-lactamase it's a type of beta-lactamase which NDM belongs to, and in this particular one, bacteria uses zinc to destroy antibiotics. So beta-lactamases, there's 4 different categories. We split them into A, B, C, and D. And A, C, and D is serine-based beta-lactamases. They use serine to hydrolyze the beta-lactam ring. And the metallobeta-lactamases are plasmid-coated, or they have a zinc cation that hydrolyzes the beta-lactam ring. So what is a plasmid? It's basically, before the internet was a big thing, if you had a movie that you wanted to watch and a friend wanted to watch it, but you didn't want to give them the CD, you meet with your friend, and then you make a copy, and then the friend keeps a copy and you keep a copy. And that's basically what a plasmid is. We used to call it cassettes to make it, but the new generation has no idea what a cassette is. So beta-lactam rings, it's just to show you what they are again on the slide. Just pay attention to this molecule and talk about NDM. So this is the beta-lactam ring. So this was a study that was done where they looked at a docking study after a 3D structure of an NDM1 as model, and they tested it against many antibiotics, and that were docked into the active site of NDM1. So the beta-lactam ring bond is polarized by binding to the substrate, and there's a few steps involved. So one is the carbonyl oxygen coordinates with zinc ion 1, and the beta-lactam nitrogen coordinates with zinc 2. And oxygen from water molecule actually leads to an oxyanion that's stabilized by zinc 1 over here. Then what happens next is that the CN bond is cleaved, and then the beta-lactam nitrogen is expelled as an anion. It's stabilized by zinc 2, as acting as a general acid. And then the final step is the protonation of nitrogen. So when they were doing the study, they noticed there's an old drug called colistin. The volume was too bulky, and so it didn't attach to the active site. So that's why colistin is actually one of the drugs we can use on NDM1. So it was first discovered in 2008, and in clinical medicine, we try different classes of antibiotics when we try to cure an infection that's very resistant. But the problem with drugs like colistin is that they're very toxic, they're less efficient, and but the good thing is that they're not used so much that there's less resistance that we see with them. But many bacteria can be pan-resistant, resistant to up and down the scale, and I've actually run into situations like this. And at that point, you just hope the immune system is going to kick in and do its job. And there may not be a treatment available. So NDM, so there was another study recently published in March of 2020, where Chinese licorice, there was an extract of a molecule called isolicoritin. It's actually used a lot in Chinese medicine. And by itself, it didn't have any effect on the bacteria, but in combination with a beta-lactam ring drug like meropenem, it actually prevented the hydrolysis of meropenem because it bound to the NDM active site. And it didn't, of course, it didn't work on the other strains of that had different molecules such as NDM5 or other beta-lactamases, but at least there was hope at that point. So overcoming the beta-lactamase. So beta-lactamases produce by bacteria to destroy the antibiotic. So what have we done is that throughout history, we've had beta-lactamase inhibitors. We just didn't have one specifically for NDM1 as of yet. So examples of beta-lactamases are clavulanic acid, which is seen in amoxicillin clavulinate. There's a drug called Augmentin. I'm sure many of you have probably seen it or taken it. This tazobactam in a drug called ceftolozane tazobactam, and also piperacillin tazobactam. So how do they work? Beta-lactamase inhibitors don't actually kill the bacteria on their own, but there's a few situations where they actually can. But what they do is they bind to and inactivate the beta-lactamase, and that allows the beta-lactam drug to then go inside and bind to penicillin binding protein, and that prevents cell wall biosynthesis. So how do we create new antibiotics? So one of the ways, there's different ideas of how to do it, but if you can overcome the mechanism of resistance of the current antibiotic, then you can probably create a new drug. So beta-lactamase inhibitors are often added to drugs we already have, and then they make the drugs more usable. Drug efflux, so if there's a problem with drug efflux, the drug goes into the cell and then the bacteria pushes it out. Maybe there's a way to block the pump. If the bacteria changes its target site, then maybe you need drugs that will attach to the new target sites or attach in a different site. So the other thing, of course, is to have a novel mechanism of action. Maybe you're finding a site that's important for bacterial survival and then destroy it. But ideally, sites should be specifically bound on bacteria and not on humans as well. So that might reduce the side effects. Nanobots maybe as a future technology, who knows? So the newest antibiotic that's out is cephalosporin. Cephalosporins, we talked about, is derived from a fungus. This is a molecular structure for cephalosporin, and by substituting components at position C1, C3, and C7, we've made different compounds. So R1 is C7 is actually if you change that particular structure, you can affect the microbial spectrum of activity. And if you change it in the other arm, you can actually allow the antibiotic to reach a different body site, such as cerebrospinal fluid, or you can prolong the elimination half-life. So the newest drug is cefiderocol. The side chains are similar to cefepime and ceftazidime, which are excellent drugs. They're probably one of the broadest spectrum drugs that we have. And it actually has enhanced stability against beta-lactamases, including NDM1, KPC, OXA, Vim, IMP, and etc. So far, it's only been approved by the FDA, Food and Drug Administration of the US, for urinary tract infections, but hopefully we'll be using it more. Well, hopefully we don't have to use it, but if we find a lot of resistance, hopefully we'll find new ways of using it. So it has a novel mechanism of action. So the way it works is that bacteria need iron for multiple processes, and in iron-poor environments, one mechanism they use is to produce and send a molecule called a siderophore. It goes outside of the cell, and the siderophores go and scavenge. They're like scavengers. They go scavenge for free ferric iron, and they form a siderophore and iron complex, and they're taken back into the cell by iron transporter cells. Cefiderocol uses the siderophore iron complex pathway, and that's how it's able to penetrate. It so uses the iron pathway because it has a siderophore in it, and we kind of, even the name of the drug, the pharmaceutical name is I believe it's called Fitroja. So it's like iron is Fe, Troja is from Trojan horse, and I is probably antibiotic. So the reason why they came up with that name, I think, is because it acts like a Trojan horse. So if you think of the old story about the Odyssey and the Iliad, Greek history, they created a Trojan horse, and then all the heroes got inside it, went inside Troy, and then they were able to get out and destroy it. And that's basically what this drug does. It's a Trojan horse. So the last thing I want to talk about is, this is actually a project that I worked on. It was finding a drug target for resistant Acinetobacter baumannii. And what we did was, first we selected a strain. So we picked a wild type strain, something that's found in, that was found in a patient. And we did a full genomic sequence of it. And then we randomly mutagenized the strain with a transposon. And off that, we did growth curves, and we found that there's a particular one that was killed the most. So the second step we did was, we found out where the mutation occurred, and it occurred in a gene called BFMR. So what we did was we reimplanted the mutated gene into a third strain in multiple areas, and then we did growth curves to see what would happen. So the growth curves were done in LB medium, human serum and ascites, and they were performed for over 24 hours. And some growth curves are performed in a complement inactivated serum and ascites as well. And I'll show you the graph for that. So what happened was we noticed that the mutated strain, which had a BFMR mutation, that actually didn't survive well in serum, but when we deactivated complements, it was able to survive better. The other thing we noticed was that when we reimplanted the BFMR molecule into the bacteria, it actually was able to grow and survive in serum as well. So what we did was the next step was to test it against different antibiotics. So BFMR we realized was increasing resistance to meropenem and colistin. So this is meropenem, and if you can see, this is the mutated strain. This is the wild type strain. And colistin, again, the MIC dropped for colistin. It had no effect on tigecycline, which is a different drug. So the next thing we did was we assessed druggability, and then the crystal structure of the receiver domain of BFMR was determined and was assessed for potential druggability by computational prediction and small body hotspots, followed by in silico ligand binding and analysis and blocking docking strategy. Blind docking strategy. So what we found was BFMR is a putative transcriptional regulator as part of a two-component signal transduction system that modulates gene expression in response to environmental signals. And it's considered a potential drug target based on in vivo mouse models. And it actually belongs to a protein family, and that particular protein family is absent from humans and other mammals, which is what makes it a good drug target. So BFMR, BFMS is part of a two-system process. And so with an environmental stimulus of the sensor kinase, the response regulator leads to transcription of DNA and RNA. And we notice that it's important for biofilm formation, resistance to antibiotics, resistance to host complement-mediated bactericidal activity, and scaffolding of the outer membrane protein A. And so we concluded that the response regulator BFMR is a potential drug target for Acinetobacter baumannii. And this was my team. This is Dr. Thomas Russo, who was, and Dr. Jasmine Ahuja, and they were our major people who helped. I mean, it's actually Dr. Russo who spearheaded the study. Both of us trained under him. And this is my office right now, where we work in the Southeastern Center for Infectious Disease in Tallahassee, Florida. And thank you. That's the end of my presentation.
Thank you, Dr. Akshay. Now, forum is open for question answer. Please.
I have one.
Yes, please.
About these antibiotic, this resistance.
Yes.
What is the reason? Is it related to the anesthetic purpose, or it is just killing the bacteria?
Are you asking why does antibiotic resistance happen?
Reason? What is the way?
So
the
I think the reason is
they are
right.
It is,
I think it's because of evolution. Right. I think it's because of evolution, because you know how I mentioned that 3 billion years ago bacteria were formed and then fungi were formed also. All of our antibiotics come from other organisms. They come from fungus. They come from mostly they come from a fungus. So bacteria and fungi have grown in the environment together, and that's probably why for almost 30,000 years, even 4 million years, as far as we know, there's been resistance because of evolution. Because the fungus will create a mechanism to destroy the bacteria, and then the bacteria are no longer able to grow, and then they overcome it by their own mutations and random mutations that form resistance and so on. So it's all evolution. And now what we're doing is
the bacteria or the microbe is not destroyed.
It's not always destroyed, no. Even when I have, so if I have a patient with a bacterial infection in the blood, for example, and I start an antibiotic, there's always something called a small colony variant, which are mutated strains that are inside, that are part of the infection. So I kill off the competition, and then sometimes the mutated strain can become resistant to the treatment.
This
and
drug
that's just
is slightly disturbing its existence only. Is it so?
It's existing. It's getting rid of the competition, basically. So if there's no competition, the bacteria that's left behind is able to multiply because there's no competition anymore.
Yeah, it is just inhibition only. not killing.
No, they do kill. They do kill. So destroying the cell wall, for example, will cause the bacteria is living. The environment that it lives in has a high osmotic pressure, so that will destroy the cell wall, and so the
Not
bacteria will
only
die by it.
osmotic pressure, is there some
There's many
correlation
things,
with the
yes.
charge of the drug?
Yes, there
Because
are certain charges as well that help. So drugs like cefepime have a positive charge that's able to get the drug into the cell faster, and it actually works better as well. So the charge also has an effect on the killing of the bacteria and attaching to the sites. You're right.
Thank you.
Sure.
Any
I believe
other?
there was somebody had asked, Dr. Samjit Sabam had asked, why do some infections recur even after taking prescribed antibiotics? It may be because the antibiotics may not be working, or because of these small colony variants that now they're able to overcome whatever's been going on and cause it. Or it could be a new infection sometimes. And I think Dr. Rajesh Kumar said, why do super infections occur after taking prolonged antibiotics? Super infections can happen because super infections just happen because they happen. So sometimes I have patients in the hospital who are having Foley catheters, or they have IV lines in their arm, and that can get infected on top. But the word super infection we actually use when there's a viral infection, and then a bacterial infection it sets into dead, damaged tissue. So bacteria can survive better where there's no blood supply. So when blood supply is compromised by things like atherosclerosis, which is blocking the vein from high cholesterol, or diabetes, or even a clot somewhere, what happens is that part dies off, and then bacteria can go inside. That's why dead tissue smells so badly, because bacteria have overcome and taken over it.
Hey, Dr. Akshay, I have a quick question. That was
Sure.
a very great talk. I thoroughly enjoyed. So do you find any, the different, for the existing antibiotics drugs, the resistance, among the demographic, for example, Asian, then American or Europe?
Okay. There's actually, I don't know about resistance, but there's actually one of the things I was working on was hypovirulent Klebsiella. And that was found on people who live in the Pacific Rim, for some reason were more susceptible to it. Why, we don't know yet. Are some people more likely to get it from others? It can happen to do also with diet and where we live and how we live. Because one of the biggest sources of bacteria in our body is the gut. Now, we cannot live without bacteria. We would actually die without them. They're so important for us to survive. And so different people have different bacteria, and there have been studies that looked at the microbiome of humans, and there's a difference in where we live. So in the West, because of our diets, people who come here, even from people from Japan, have different bacteria, whereas Japanese people have. That was one study which looked at the microbiomes for different people. The other thing was they looked at the microbiomes for people in the West, and they found that there was a much higher diversity in people who live in tribes in certain tribes in Africa. So there's many different reasons why we're not all the same. Our infections are not always the same, and our location also has an effect on it. A lot of drug resistance is actually even found in water. So bacteria that live in the water, when you drink the water, they can transfer, because they can transfer data to each other by plasmids. They're able to spread antibiotic resistance to each other because of that.
Makes sense. Thanks.
Any other?
I think there is no other questions.
No other questions. So thank you, Dr. Akshay, for a very excellent and pictorial presentation. We enjoyed. Now I invite Dr. K. Sarjit from NIO Goa. His research interest is in discovery of bioactive molecules from marine origin. He is also involved in the synthesis of bioactive heterocyclic compound. So he will speak about marine natural product in pharmaceuticals. Dr. Singh, please.
Am I audible?
Yeah, we can hear you. You can share your screen now.
Yeah, sure. Is it visible on my screen?
Yes.
Okay, thank you. Thank you, Dr. Puja Khare, for the kind introduction, and Dr. Niraj Luwang, Sapna, and Zaida for the invitation. Today, I am going to talk on the marine national product in pharmaceutical opportunities and challenges. Actually, the topic is so big and broad, and it is not possible to cover each and everything in the next 40 minutes. So what I am going to do is I will select some of the important things and what I thought it will be useful to the audience, and I will try to talk only those topics. So the content of my talk will be, first I will speak on the brief introduction of marine national product, linking chemical ecology with secondary metaplanet productions by the marine organism, and chemical defense of marine spawns, marine polysaccharide molecular modification cultivation, and challenges of the MNP in the drug discovery, and how to weigh out these challenges. And I will speak a little bit of marine natural product derivative, that is kojic acid, because we have done some of the analog, but out of that I will pick up only one. And finally, I end up with summary. So marine natural product actually is conceived in 1951 when Bergman and Finney isolated very unusual nucleoside, this asparagine and asparagine from the marine spawn. This spawn is nothing, it is one marine animal who they are sessile. Sessile means they don't move, they remain stick on a rock, and they are filter feeding animal. That's what they call spawns. They just like our spawn thing, they eat their food through filter feeding from the water. And this two nucleoside, after all, this become the very important lead structure of antiviral drugs. And in 1967, there was a very small symposium on the USA, in the USA. The title was Drug from the Sea. And from that started the interest on marine natural product. And during 1950 to IT, a very few marine natural product lead compound have been identified, but the progress was very, very slow. But after 1980, with the use of scuba diving, because those before 1980 people hardly use scuba diving, so the accessibility of the marine organism, it was a question. But after using the scuba diver, people can go inside the water and they can collect the marine organism. So the research was so progress after 1980. So from 1980 onwards, so many marine natural product has been discovered. So far, more than 15,000 secondary metabolites have been discovered from marine organisms, including spawns, soft coral, and marine microbe. So this is the two guys who lead the marine natural product research. The first one is the Faulkner from the Scripps Institute of Oceanography. Actually, he was organic chemist who involved in the synthesis. Later on, he shifted his research interest to the marine natural product, and at the end of his career, he looks on the chemical ecology, and the remarkable work of his contribution in the marine natural product is considered as the 17th consecutive review on natural product report, which is very useful to the people like me who are working on the marine natural product research. And from 2003 onwards, the job is taken by the Blunt from the New Zealand. What are the opportunity of marine natural product? Marine environment has tremendous potential that everybody knows, because it is the house of the several fascinating molecules. When we call about the lot of the fascinating molecules, because they have, not unlike terrestrial, they have always some stress conditions are there, because since they are in the water, there is constant change in pressure, salinity, and the competition for this space, and pH changes. Those lot of environmental stress are there. So the marine environment has a very vast, it covers 70% of the earth, and it represents the largest unexplored wealthy resource. Extreme environmental conditions, just now I spoke, stress conditions such as very high temperature, pressure facilitate the production of novel compound for adaptation with the surrounding. Deep sea organisms, including microbes, are known to produce novel bioactive compound. Limited study has been done so far and need to be explored more. So, in marine organism, there are input are like photosynthesis, photoprotection, heat and cold stress, attack, defense, and nutrient starvation. Those so many stress conditions are there. These are the input, and the output are their primary metabolite for this one, carbohydrate, protein, fat, nucleic acid, and secondary metabolite, terpene, alkaline, pyrrole, peptide, and so many secondary metabolite bioactive compound are there, and these are the output, because this is mainly they use for their survival chemical defense. And so marine bioprospecting is very important in the pharmaceutical, biofuel, cosmetic dye, and food as well. This is the marine national product actual contribution from different organisms. You can see microorganism contribute 38%, spawns 20%, and this is the pie chart. But if you look, suppose 20 years back, this contribution was quite different. Around 20 or 30 years back, marine invertebrate spawns, then soft coral contribute more than the microorganism. But this day, microorganism research is more and more increasing, because many novel compounds can be discovered from microorganism. And this is the same thing from the 5-year bar chart during 2012 to 2017, the number of bioactive compounds from marine organism. You can see the microbe is the highest contributor. So what are the bioactivity we usually encounter from the marine organism? See, anti-cancer, antiviral, anti-diabetic, so many bioactivity compound we can get from the marine national product. And as I mentioned, that 15,000 that secondary metabolite have been already discovered from the marine national product, and it is application in pharmaceutical, chemical, ecology, and high value ingredient in the fine chemistry, and food is already underway. And I just give the selected marine national product which are in market and under clinical trial. This is just a representative. And first one, this one, the antiviral drug, ERA A, ERA C. This is come from the spawns, and these are the suppose zincotides come from the conuts, and brostatins come from this species, and picozatin is antibody and from red algae. And these are some of the compound that the FDA approved marine drug. Bedravine, just now I spoke about the ERA A. This is the treatment is bedravine and cyprobratin and zincotide and so on. So I'll come to the chemical defense. You can see in the marine environment, especially in coral reef, there are a lot of organisms are there. There is constant competitions of the space, food, and they have to protect from the predator. So in order to do this, they produce chemical bioactive compound, and you can you can see that marine spawn is this one, which is very soft, and they eat their food through filter feeding only. And it is very interesting that it is a soft, this a soft body compound, organism which doesn't move, are the maximum producer of secondary metabolite, while this one organism with the armor, and those organisms which can move, they lack the secondary metabolite, or some case they produce very few secondary metabolite. So from this we can understand that this soft body organism, in order to defend from the predator, they produce the bioactive compound, because otherwise they don't have any armor and they cannot move here and there. So they remain fixed on a particular place, or say rocks or in the soil. And this is the picture of the spawns and pistularies, and this is brominated compound. this brominated compound also they use for chemical defense. And just now I spoke, see those armor and the organisms which can move, they produce very few secondary metabolite, or they usually lack secondary metabolite, and so body organism like a spawn, sea slug, tunicate, and mobile animal like a squid, they produce a lot of secondary metabolite. And there is this one organism spawns aegis species, and this aegis species, this is a very interesting marine spawn, and it's found in many places like Okinawa of Japan, Caribbean island, Mediterranean Sea, and South Pacific Ocean, and they produce bromopyrrole compound like bromotyrosine and this pyrrole imidazole alkaloid. This compound is characteristic of this organism, whether you isolate it, whether you collect it from Japan, Caribbean, Mediterranean, and South Pacific Ocean, you will find this bromopyrrole compound from this organism. So from this, what we understand is the chemical, the secondary metabolic productions by the marine organism depend on chemical ecology, as well as there are some genetic factor also contribution, because you can see this one whether they collected from the Okinawa, Caribbean, Mediterranean, or any other part of the world, you can see this brominated compound. So I will speak a little bit about a halogenetic compound from marine origin, because halogenetic compounds are mostly bioactive, and their bioactivity is mostly on chemical defense. So halogenetic compound we get from algae, bacteria, mollusks, and sponges. These are some of the representative of the halogenetic compound that we encountered in the marine ecosystem. And this is the, you can see bromodiphenyl ether. I just saw one example. There are a lot of bromopyrrole compound where this, instead of OH, carboxylate, ester, and bromine position changes. So there are maybe 40, 50 modern the bromopyrrole compound are there. So we are compiling a review on this bromopyrrole compound of marine origin. And this is our collection site where we collect the sample. See Rascot, this is Gujarat, Ukha, and near Mumbai Island. This is Ratnagiri, and our institute is here in Panaji, and Mangalore, and Mantapan, Tamil Nadu. This site this are the collection site. We usually collect by wading like this, and sometimes scuba and trolling. So how we process the organism: first sample collection, crude extract, bioactive fraction, fractionation, and pure bioactive compound using chromatographic technique, and structure elucidation, and activity validation. And we do anti-fouling, antimicrobial in NIU itself, and we have some collaborations for other activity screening. And these are some of the work we did in the NIO. And this is the terpene from the Cyanuria. And this is also another terpene from the other Cyanuria species. And we found some interesting things, that this the brominated compound, one collected from Ukha, Gujarat, and this one is collected from the Mandapam, Tamil Nadu, and you can see little different in the chemical structure. And this is the mass spectrum, only we characterize the compound. And this also one aegis species, bromopyrrole compound, and this compound hexospongin alkaloid, or we can say this is the quinidylidine alkaloid. So we can see, because you will see quinidylidine ring here, and this was isolated from marine spawn Oceanopia species, and we have characterized with the single crystal structure as well. And there are a lot of compounds are there in this organism. So we isolated almost 15 compounds. Out of them, few only I have shown here. And characteristic of this compound in NMR is this one, this there's a carbon 13C of this CS carbon. You can see around from 89 to 95 chemical shift, and this is, if you see that this one signal, then you can conclude that this is the quinidine rings are there, and most of the compound, only 1 or 2 we can isolate, remaining we characterize through mass spectrometry by MS study. See, this is the mass 479. For example, I take this example, 479, here the mass spectrum. Actually, mass is 478. This is the M plus is the protonated one here. And here also you can see some peak here. So this clearly shows that this compound is dimeric in nature, because you see M by 2 plus is 240 is corresponding to the this molecular mass 478. This actual mass is 478. How about the 478? 239 plus is 240. So correspondingly, you can see for all these. So these are the dimeric in nature. They show a lot of bioactivity, and from the IIT Hyderabad, they identify some similar kind of compound, and it shows anti-SID properties. And this is just a sterol isolated from the arsenite species, and these are the bromodiphenyl ether. Just now I spoke about this. This we isolated from Dicydia species. The interesting of this compound is it shows both gram-positive and gram-negative bacteria activity, and only we are trying to replace this bromine through synthesis with the fluorine, because bromine is toxic. So even though it's so bioactivity, it has no value, because so we are trying to replace it with fluorine. So now I'll speak about a little bit of the marine algae polysaccharide. So this is the marine microalgae seaweed. Also we know as a seaweed, and there are two are there, microalgae and macroalgae. Seaweed is the macroalgae, and they are distributed almost everywhere in the coastal region, particularly in the tropic, subtropic. Even you can find these species in the temperate zone. They have a lot of application in the South Asian country like Thailand, Indonesia, even in Japan. Some of them are used as a foodstuff. And this marine algae is the foodstuff because you can get a lot of material from here, because they can grow very fast. So there are a lot of biomass. From that biomass you can get the chemical. But the only problem here is the solubility. So many people are looking research for the chemical modifications of the polysaccharide, that I will speak a little bit about that later on. And this is the one red algae. Then this Reliance Life Science is actively involved in doing research, because they are found is active in the coronavirus COVID-19, and this activity is due to this one sulfated polysaccharide. Sulfated polysaccharide are usually very good for antiviral activity. It is well known, established, and they have tested for the COVID-19 as well. And this Marinomed Biotech, one company, has filed a patent about the activity of this red algae on coronavirus, beta coronavirus. And this is one of the PhD students did. That is a small work on the marine pigment. Most of the marine pigment are already known, because you will find hardly any new pigment. Most of the pigment are well established. But only interesting thing is that in that seaweed we found very good concentration of the two pigment, pyrophytin A and cucurbitacin. The interest in the marine algae is sometimes you get very good concentration of cucurbitacin and sometimes beta carotene. So the people are looking to commercialize those algae, and to do that, you have to be identified in which algae this important carotenoid, pyrophytin, fucoxanthin, or beta-carotene content maximum, and you have to see the season, in which seasons they contain maximum to you using HPLC, so that you can collect in particular season and you can process to collect those valuable compound. And this compound, we have tested some anti-feeding activity against the shrimp. So this is the control and this is the experimental. You can see in the experimental where we mix the fish food with this compound, you can see a lot of the fish food remains left after the experiment. This is we did in aquarium experiment. And another polysaccharide from marine is chitin. Chitin is the most abundant polysaccharide next to the cellulose, and it is mostly extracted from the marine source like crabs, shrimps, and clam, like that. 70% of the chitins are come from the marine source. They show antibacterial activity, and they are biodegradable, non-toxic, low toxicity, and high abundance and low cost. And there are two reactive sites. About that, I will talk later on. The two sites are there, this one and this one. This is the active site, reactive site of the chitosan. So to this active site, you can modify, you can put a lot of functional groups so that the chitosan become soluble to the common organic solvent as well as water. And usually when you extract some chitin, you will have mixture of this. The acylated one is chitin and deacylated one is chitosan. If you get, after the process, if you get say 80% of deacylated, that you can say that chitosan is a very good quality. Sometimes you get 90%, sometimes you get 85%, that is good. If the deacylation degree of deacylation is less than 60 and say 50%, then solubility will be extremely difficult, and you cannot use for other reaction. And if you want to make some product from there, then the deacylation degree of deacylation should be above 80%. So this is, I am just showing the real chitin, and this is the ideal. In the ideal, completely acylated, and in the case of chitosan, it is completely deacylated. But when you extract from the nature, you will not get 100%. Always, this is the mixture of the things, chitin and chitosan, acylated and deacylated mixture you will get. This is one of the dissertations then she did very recently, and we extract the chitin from the prawn. That prawn, what we directly purchase from the market, and after remove this flesh, and in the everything is removed, then only cover. The cover we dry it and we do the demineralization, deproteination, and you get the chitin. And after deacylation, you will get the chitosan. But for this chitosan, you have to see the degree of deacylation. So whether your chitosan is good or not, you can check by using 2% acetic acid. If your chitosan is soluble in 2% acetic acid, then you can consider that it is above 80%. Otherwise, it is less than 80%. So you have to repeat the experiment, deacylation process again and again, so that the degree of deacylation will increase. So higher the degree of deacylation, higher is the activity. And some research paper are there. So like 96 or 97% deacylation, antimicrobial activity was extremely good. Deacylation is usually depend on the source and process, condition, time, and how you do the deacylation. These are some of the applications of chitosan, which is well established. The heterogeneous catalysis, you can see a very good review on this in the coordinates chemical chemistry review, and they have been used in the tissue engineering as antimicrobial agent, wound healing, dressing, burning, and treatment of bone damage, than artificial skin antiviral. We recently put a proposal under Indo-Portugal. This is funded by the DST. That is, the project is under review. And in that project, we look on the skin, on the artificial skin. And I will talk about this also. This recently, you can find this one, molecular connection, this one paper, where they found that this chitosan, what is the chitosan? They don't mention. The one chitosan derivative is two chitosan derivative are found active in the this beta coronavirus. That is the COVID-19. There are 15 natural products based compound which are active against COVID-19. Out of that, 2 are derived from chitosan, and these are the patented, already patented. And chitin is also the source of a lot of chemical. You can see, you can get a lot of chemical. So the not only chitin that we plan to do something on the seaweed also to get the chemical. It may be known that it doesn't matter, but for the self reliable or for the self-sustainable this one way, we can get a lot of chemical from chitin as well as from the biomass of the seaweed, which is abundantly found across the coastal region. So polysaccharide, whether it is chitin or any other cellulose or any other polysaccharide, the main difficulty of the polysaccharide to use in the other application field is their solubility. So how to make soluble? So there are molecular modification of chitosan, and it can be used in the even in the polysaccharide as well. So first is the chemical modification, physical modification, biological modification. Biological modification, you can use microbe and other things for enzymatic degradation of the polysaccharide. And physical modifications are there, and chemical modification, you can put the functional group like selenation, sulfation, carboxylation, phosphorylation, and so many things you can do it. So after doing these things, and you may get some polysaccharide derivative which may soluble in water or some organic solvent. And this is the sum of the work for the chitosan we have done, because as I mentioned that this is NH2 is there. They saw the characteristic reaction of amine, and this OH also you can do functionalization. And here you can put this with the 2-chloroethylamine. You can put this thing. These are all become water soluble. And the most interesting thing is chitosan is soluble in 2% acetic acid, not water soluble. After modification, you don't require even acetic acid. You can directly put water, so it becomes water soluble compound. So this is the work we propose. Some of the work we have done, and some are under progress. So I will not speak much about this, and this is already done. This is a highly water soluble compound. And this is the these are the students. That one students came and see it during the lockdown only, and this is the mixing the chitosan and cellulose, and we get this kind of things. And we have not characterized what is this. We have to do more. This is just during the lockdown, see it, and work is not complete yet. We have to do more on this. Here, this is the chitosan. You can link the chitosan, cross-link can be done. Chitosan cross-link, chitosan cellulose, even chitosan and collagen. Collagen also you get from the jellyfish, from the marine source. So you can do some cross-linking. Cross-linking, the stability and their mechanical strength is increased, and this can be used in other material. And you know that chitosan is cationic in nature. If you put in water or little acetic acid, that NH2 will be protonated, so it becomes water soluble. So in that you can put a lot of anion. So through that anion they can be linked. So you will get the gel type of things, and you can put the middle and otherwise you can do through covalent cross-linkage. This is the covalent cross-link, and this is the linkage linking to the metal ion, and this is using the anion. And this cytosine, these rings are the sugar moiety, and this is the OH and NH2 here, OH and NH2. And this is the active site of the cytosine. You can put any metal and they will form like this. It may not be directly bonding, but they will form aggregated material. And this is the methyl loaded cytosine, one can say like that. Or other, and in that way, other organic compound also you can load it in the cytosine. So there is paper here, so that they loaded niclosamide, which is a bioactive compound, and they use the twin IT as surfactant, and they got like this. And this is the niclosamide cytosine nanoparticle. They found it is a good, this is biodegradable, and they are used in the cancer therapy. So that all about the what is opportunity, what are things going on at present, and this is I just brief a little bit, and now I will speak on what challenges faced in the marine natural product chemistry in the drug discovery as far as drug discovery research is concerned. So first of all, the main challenge is, I will say, the biodiversity. Because in the marine natural product research, to get secondary metabolite are usually contained in a very small amount. So to harvest 1 gram of secondary metabolite, you have to remove a lot of the organism. So it affects the ecosystem. So biodiversity will be vastly disrupted if you rely on the marine organism only. So this is one of the disadvantages of this marine natural product. This is not only marine natural product, on terrestrial natural product also it will be true, because when you extract that compound from any organism, since the secondary metabolite contain very small amount, you have to harvest a lot of the organism. So this is the ecologically, it is not a feasible one. And second one is the supply issue. The supply issue means you can get a secondary bioactive secondary metabolite in a very small amount. So there is the supply chain will be in strength, and other one is the reproducibility. When I talk about the reproducibility, means when you get some compound, next time you collect the same organism from the same place, you may or may not get the same compound. Especially when you deal with the marine spawns. Marine spawns is 60% of the marine spawn is constituted with the microbes. So 60% of the marine spawn is contribute with the microbe. So microbial community changes with the change in the salinity or pressure or water, the surrounding water environment. So when you extract the compound and the microbial community structure is changed, then you will not get the same compound, even though you collect the same organism from the same place. So there is some reproducibility issue is there, and of course the sustainability. And third one is the technical issue. So there is challenges in the technical, some technical issues is there. First is the difficulty in the isolation. Isolation of natural product from the organism is extremely difficult task, and not only this one, isolation, characterization also is very, very difficult. So there is technical difficulty in the isolation as well as characterization. So what is the way out? I add little bit on the way out. So one is the marine biotechnology, where you involve aquaculture, mariculture, and fermenter. So with that, you can do some sustainable way of production of marine natural product. Another one is chemical synthesis. There is also genetic engineering also there that I skip. I will not speak about that. So I will concentrate on the chemical synthesis. In the chemical synthesis also, you can do two ways. One is organic synthesis and another one is the through catalysis. So the catalysis advantage is the steps are usually very few and good in atom economy, and efficiency is moderate to high, and there is chemoselective and ratio specific, and the most of the reactions protocol are environmental vegan because environmentally very friendly, this catalytic way of doing synthesis. So already many people has done the natural product synthesis through catalysis. So you can see the review article and Advantek and Nature Chemistry. Here they use sheath bond activation for the synthesis of natural product compound. I saw very few, one or two. There are a lot of application of sheath bond activation in the natural product synthesis is already established. So as I mentioned just now, the alternative is the aquaculture as well synthesis, and I concentrate on synthesis. We are trying to develop synthesis of this compound. Why we are trying means we have isolated this compound from our natural product work, and they found to be very promising. This also kojic acid. This is very small molecule, but it has a lot of activity, anti-tyrosinase, antioxidant. Even the complex of this compound, so anti-diabetic, anti-cancer, and there are a lot of scopes is there with this small compound, this kojic acid. This people has used for skin whitening as well. So we are trying to do some synthesis of this 4 analog. So I will stick only on this kojic acid derivative. So kojic acid, you can see it has a lot of applications there in cosmetic, chemical industry, pharmaceutical, agriculture, and in the chemical industry, you can get a reagent, you can use as a ligand, and you can use as a raw material for the ligand design. Yes, this is image is a little poor, sorry about that. So how this act as a anti-tyrosinase. So they inhibit tyrosinase, their anti-tyrosinase activity. How they do is, I'll just explain a little bit, because tyrosine contains copper ion. Because of the copper control the activity. So once you put the kojic acid is a very good binder to the lot of transmetals. So it bind the copper here. So copper will not be available. Once the copper is bind here, the activity will be inhibited.
Thank you, Dr. C.
Now, I will finish up. It is some connectivity problem is there.
Okay. So you want to continue, no? Okay.
You can go to day 3.
No, just a minute, Niraj.
Okay.
Because some connectivity issues there. So,
yeah, instead of joining like this, you can just click the day 3 from the email. It will be faster.
I'm doing the same thing.
I have shared the ID and password in the speaker WhatsApp group. If you can just put in there. Yes, you are there. I think Dr. Sarjit has gone offline again. I think most of the presentation he has already shared. I would like to request Dr. Puja to kindly conclude. Yeah, he is back online.
I'm sharing now. Okay.
Yeah, it is not coming.
Yeah. Just now I'm doing.
So we will move forward. Hello.
Dr. Sarjit, how many slides were left?
Okay. It will come now because this one, no? Because this one, no, the PowerPoint was closed.
Okay.
Dr. Sarjit, how many slides are left?
Some remaining.
Okay, then I will share it from here.
No. It's visible now.
Yes, it is visible.
Okay.
Yeah.
Yes.
Because I think connectivity is lost suddenly. Sorry for that. I think I was speaking somewhere here, right?
Yeah.
So, sorry about the disturb. So we synthesize some of the kojic acid derivative.
You can go full screen.
Okay. So why we are doing this kojic acid research is it has a lot of Then this kojic acid you can make the their derivative here by putting the amine. You can get a lot of the derivative of kojic acid, which show antiviral and antimicrobial activity. Recently, they published the paper, and this is the kojic acid complex, which show anti-cancer, radio protective as well as anti-diabetic property. And this is we did the work here in the NIO, and we prepared a complex, and this is very good for catalysis, homogeneous catalysis, and we use this one. Shear annihilation in water, and these are some of the derivative what we prepare in the lab. And we prepare this derivative through the shear bond activation. What we do is halogenation. Here the bromine comes here, because I will show how the bromine is this. Here is the lithium carbon bond carbon bond functionalization will take place. This is annihilation, alkylation, and alkylation. Kojic acid, what is our strategy? You cannot directly do the functionalization. You have to make first methyl complex. So Kojic acid, you do this pyridine, you can introduce here, or otherwise some ketone or something. Then after that, you will do Shays activation. Once you do the Shays activation, you get the compound like this when you use ruthenium. So just the previous slide, I was talking about the bromination here, because most of the bromination in the case of ruthenium, it takes place opposite to the ruthenium carbon bond. That means para position to the ruthenium carbon bond. That's what the bromine, we expect that bromine should come in this position. So once it is this complex, this is the intermediate species, then you can do the arylation. So from this Kojic acid, you can prepare their analog. This is we call the marine natural product inspired synthesis. From that, you can do a lot of things, activity as well as for the ligand, and you can do a lot of complex preparation you can do, because these are the good binding sites here. And we have done some few work, catalysis work to prepare this Kojic acid analogs. We use ruthenium, then we with alkene and alkylated product, we got alkylated product. When we use palladium, it is the alkylic product we got, because hydrogenation also takes place. After that, you have to do deacylation, this one, then you get the derivative of the Kojic acid. I think that is the last. This is a summary. So ocean not only support and provide livelihood to millions of the people, but it also houses the several marine natural products of biological significance. There is very strong linkage between the secondary metabolite production by marine organisms and ecology. Most of the biotic compound produced by marine organisms is for their survival and adaptation. And you can see brominated compounds are abundant in marine organisms, even though bromine concentration is much lesser than chlorine. Sessile organism, that is those organisms which cannot move, which remain fixed, which soft body are usually rich in biotic compound, while blood and armor organisms are either leg or pure secondary metabolite they produce. Then marine polysaccharide like carrageenan and chitosan have kept a lot of interest in recent years. This is due to their biocompatibility, and their application in the biomedical research. We did some work on the modification of chitosan. The main limitation of MNP is supply issue, chemical difficulty in reproducibility. Just now I spoke about that. And to address this, you have to do biotechnology, that is marine biotechnology, aquaculture, mediculture, fermentation technology, because this is mostly used for those people who use for the microbe. You can get the microbe even from the deep sea. And of course, the chemical synthesis, that is what we are doing. This is we call marine natural product inspired synthesis. So that natural product synthesis is not easy, because this is very complex procedure. So we usually take marine natural product at their scaffold or a small part of the marine natural product, and we build. So this is much easy, and sometimes their activity is proved to be better than their parent natural product. So natural product research lie at the heart of the drug discovery program, even though there are some limitations there, but they lie at the heart of the drug discovery program, because you can see quinine, the anti-malarial compound, now hydroxychloroquine is there. This is inspired from the natural product quinine, and it is extremely important to identify the lead molecule so that it can be taken up for large-scale production through biotechnology or chemical synthesis. So collaborative effort among the natural product chemists, microbiology, biologists, pharmacology chemists can boost the advancement of pharmaceutical research and help whatever I mentioned just now. Thank you. This is our group and with the visitor. Thank you very much for your kind attention. Sorry for some disturbance during the presentation.
Thank you, Dr. Singh. Hello.
Yeah.
Thank you, Dr. Singh.
Yes, you are audible.
Please ask question very quickly, because we are already beyond the time.
My connectivity is little poor, that's all.
Okay. Anybody? Any question?
Yeah, I have.
Please.
About the seafood, we don't have access to the seafood in India. Is there something there at Goa? One. And what is the contribution of the sea product pharmaceuticals, means percentage in terms of the whole pharmaceutical thing? Okay. And the third thing is that, like in Japan or many other Southeast Asian countries and many other developed countries, they eat the seafood, and as a consequence of whatsoever, their life expectancy is much more, the highest in the world.
Yes.
So in India, why it is not so importantly taken? You have any idea on that?
Yes. Seafood contribution. Seafood contribution in the medical field is well known. For example, some algae are there. That is, they have some nutrients. Just during the talks, I mentioned like beta carotene and vitamin E. Some of the algae are rich in vitamin E. So some people use for especially diabetic people use some of the algae. As far as this one fish concerned, that seahorse, one animal is there in the sea. Seahorse is a lot of demand. They have a lot of medicinal value. Only problem is the mass production we have to do through aquaculture, and consumption of the seaweed is concerned. We need to do the environment should be clean, because seaweed sometimes they absorb the methane. If the methane pollution is there, they will absorb the methane like cadmium. So if you take those kind of seaweed, it will be harmful to our body. So to take the seaweed, we have to make sure that the surrounding waters should not be contaminated and should not be polluted. So regular monitoring of the pollution is required to consume those, if you wish to consume the seaweed.
Okay, our sea waters are generally polluted, highly polluted. Is it so?
Yeah, it is it depends on the this is actually how you control the pollution. In the NIU also we have taken the environmental impact assessment, where the government team are there. These are constantly monitoring the this one pollution. But of course, in the big city like Mumbai and all, the pollution level will be high. So we have to get the place where there is less pollution or negligible pollution. That has to be done through the cooperative manner, because you cannot just go and get it the things because if the water was polluted, then it will be affected. So pollution control is required.
Okay, fine. Thank you. Thank you for the nice presentation. But at the same time, what we generally do is that we eat such polluted things. That's why we have to finally eat tablets only or medicine only. Instead of eating that food as medicine, we are doing the reverse of that. Okay, this is how our life expectancy here is extremely low among the.
Yes, correct. Rather than medicine, it is better to take this one as a food. That is a much better option.
Yeah. Okay. Thank you.
Any other?
There is one query at the chat. Dr. Sarjit, can you kindly entertain the query?
Yeah, I cannot see any this one video.
Okay, this was raised by Nomita Sandram. What she was asking is that what is the main difference between marine and natural product found in our native place, mainly about the medicinal values, and which one is better for researching?
For natural product, okay.
Natural product as compared to the marine algae and kind of things.
Yeah, natural product means two things are there. In the natural product, one is the marine natural product, which deal with the marine environment. Another one is terrestrial natural product, which deal with the terrestrial plant or animal. So up to the organism level, it is different. Once you get the organism, the chemistry part is same, whether you are deal with the terrestrial natural product or marine natural product. So I think that is the question. What is the difference between the terrestrial natural product and marine natural product.
You want to say that it is almost a similar thing.
Yeah, but for
I
sheep
think
product, it may have the contaminants. That may be another factor.
No,
natural product.
actually, as far as contamination is concerned, it is always not contaminated because the sea coastal area is so vast. You can get the seafood far from the coastal area. So mostly pollution comes near the industrial estate, near big city. But if you go a little far, pollution level may reduce. So there is also constant monitoring out there for the heavy metal accumulation in the shells. There is a clam, oyster are there. Sometimes you get heavy metal in the oyster, and there in Goa also some particular areas there are lot of kidney failure are there. Kidney failure is mostly contributed from the consumption of the seafood. Seafood means mostly this one of clam and the oyster, because they like this seafood very much. But those seafood, if you collect from the polluted area, then of course, in fact, this one, the danger is there. Otherwise, there is no issue, I will say.
Okay. Thank you, Dr. Singh, for nice presentation. So maybe we move forward. Next speaker is Professor Shanta from ISI Delhi, and his topic is very interesting, prime numbers and its many faces. He's expert in number theory, so I invite him for his presentation.
Thank you very much. Can you hear me?
Yes, you are audible.
Yes.
Yeah. Okay, so let me first share it and then I'll start. Okay, so can you see the full screen?
It is actually not full screen. We can see your dialog box.
Okay, just
But still it can work.
Yeah, you can
because
yeah,
I use this dimmer, so that's why I think last time we.
Is it PDF?
Yeah, it's a PDF dimmer.
Yeah, okay.
So now, okay, I can. Yeah, okay. So first of all, I'd like to thank the organizers, particularly Niraj H and Swapna and other KHUMANG team, for this invitation and for arranging this nice web conference. So today I'm going to present on this topic of prime number in its many facets. So this is, of course, compared to other talks, this is quite mathematical, but I've tried to make it much more simpler so that a general audience can understand. So let me begin the talk. Okay, so first let me ask this question. So which one of the following numbers is a prime, right? If I ask you, if I give you this number or this number, then if I ask you, okay, which one of the following is the first number a prime or the second number a prime, then it's going to be quite difficult. I mean, if you cannot just right away show that which one is prime or not, but by looking at the way it's presented, in the first number, everything is even number, as you can see, 236 extra. So without doing anything straight away, you can say that the first number is not a prime. But in the second number, you have all the even numbers except one number, odd number, odd man, whatever. So number 3, so which is odd. Now you can't say whether it's a prime or not. The life is not that easy anymore. So this is it's a very simplicity about prime numbers, which all of you know is the basic building block of number. It's quite mysterious in nature. So I'm going to talk to you about some interesting properties and problems related to prime numbers. So what's a prime number? A positive integer which is bigger than 1 and which has only its positive divisors 1 in itself. These are prime numbers. So as you can see, number 2, 3, 5, 7, 13, extra, these are primes. And prime numbers are building blocks of numbers. In fact, every number can be written as product of primes in a unique way. So these are very important. And the set of primes is infinite. And a number which is bigger than 1 and which is not a prime, this is called a composite. And 4, 6, 8, 9, 10, these are composite numbers. So now if I ask, okay, is 1 is a prime or not? I mean, so we don't take 1 to be neither prime or composite. Why is it so? Let me just tell you. So this is the fundamental theorem of arithmetic, which tells you that every positive integer can be written as product of primes and is unique up to order of primes. For example, like 100, you can write it as 2 square into 5 square. And a given number n, you can write it as P1 power A1, P2 power A2, and so on. And we don't take 1 to be a prime, because if 1 is a prime, then you can write, say, for example, number 10. You can write as 2 into 5, 1 into 2 into 5, 1 square into 2 into 5, and so on. You can write 1 any number of times and still, you can write any power of 1 times 2 into 5. So then the unique factorization will be violated. So that's why we don't consider 1 to be neither prime or composite. And one takeaway from this fact is that every number bigger than 1 has a prime divisor. So this is a thing which all of you have learned in the high school and before 10. Now I will use it and then show you that the set of primes is infinite. Okay, so here, so this is the only proof I'm giving. I'm not going to, in mathematics doesn't work without proofs. And this is the proof, only proof, which is very simple. And this is one of the most beautiful and most easiest proofs in mathematics. And it's one of the first proofs which I'm presenting here. And this is the only proof I'm going to give in this talk. So let's show that the set of primes is infinite. So the method of proof works by showing that something like proof by contradiction. So what we do is that we assume that there are only finitely many primes set P1, P2, PR, and we consider number n, which is you take the product P1, P2, PR plus 1. And since number n is some prime numbers, product of prime numbers plus 1 is bigger than 1, so it has a prime divisor. Now every number n has a prime divisor, and one can see easily that the P1 doesn't divide n, P2 doesn't divide n, P3 doesn't divide n, PR doesn't divide n. So none of the primes P1 to PR divides n. So that means n should have a prime divisor which is outside P1, P2, PR. And also n is not equal to any one of these PI. So which contrasts the fact that P1, P2, PR are all the primes. So it should be so we made an assumption that there are only primes P1, P2, PR, and we get a contradiction. That's why the set of primes is infinite. So in nature, as you can see, the set of primes is quite, it's infinite. You can have as big as any prime you want, but unfortunately, even with the latest technology, we don't know big primes. So this is the currently top 10 largest non-prime. So that means right now the largest prime we know is of 24 million digits. We don't know of any prime bigger than 24.86 million digits. So I've written the primes, the number of digits, and the year in which they are invented. So discovered. So in 2018, they saw that this number, which is a prime of the form 2 power p minus 1. So this number 825,89933 is a prime. So these are called Mersenne primes. So this is the top 10 largest. So this is the largest prime as of now. In fact, in the first slide I showed you, I gave you this number 2 cube plus 3 cube plus 30 cube extra this number. If you try to show that this is a prime, if you start now, you can't even finish in next 1 or 2 years, because unless you really work with modern technology, with more mathematical advanced ideas. That's because this is one of the this number, which is 20,562 decimal digits, was proposed as a challenge for something called primality proving by using first elliptic curve primality proving method. So let me give you a glimpse of how much time it took to show that this is a prime. So the work to show that this number is a prime started in around September, October 2005 on 32-bit machines, and it finished in February, in between February to June in 964-bit biprocessor. So this ECCP requires a lot of steps: Fornace step, square root math step, PRP test step, etc. And the first phase took 1900 days. This is not the actual days, but these are the computer, timings with respect to AMD Opteron computer time. And second phase took 319 days. And so, like, if you don't use this, or the first elliptic curve primality proving method, if you start on your computer now and just try proving that this is a prime, it's going to take at least this many days, 1,900 plus 319, at least this many days, if not more. So even though we know that there are infinitely primes in nature, but showing that something is a prime or finding large prime is not not an easy task. Then the question is that why do we study primes? So mathematicians have tried in vain to discover some. So you know that primes are infinite. So but then can you give a sequence? Can you give a pattern in the sequence of primes? But then the problem with primes is that every time you try to understand something, and it becomes more or more mysterious. So that's why it's kind of intriguing, and that's how lots of mathematicians are interested to study primes. Now, I would like to take up this case of infinitude of prime. We know that the primes are infinite, so there are infinitely many primes in nature. But the question is that if you give a fixed number, say up to 10, up to 100, up to 1 million, up to 1 Google, or given any number x, can you say how many primes are there? So this is given by something called prime number theorem. So pi x function counts the number of primes up to x. So for example, up to 100, there are 25 primes. So the prime number theorem tells you that up to x, there are around x by log x primes. So here log x is log to the base natural logarithm, log to the base e. So in other words, prime number theorem can be written as the limit pi x by log x. When x goes to infinity, the limit becomes 1. So this is the well-known prime number theorem, and it was proved around 1890. So in the late 1800. Now the thing is that, but even then we still have long way to understand. If I take ex to be pi x minus x by log x, what we know is that when x is sufficiently large, pi x is very close to x by log x. So now let's look at the error term, which is the difference of pi x minus x by log x. This is called the error term, and it's trying to understand the magnitude of this error term is one of the important problems in number theory, and which has very wide ramifications. So I'll tell a bit more about it. Now let me define this so-called Ban-Mongol function, which is defined to be the this for when n is a prime power, p power k, you define it to be log of p. And when n is not a prime power, you define it to be 0. And say-by-say function is sum of the log p, where you take numbers which are powers of primes less than x, and for every power of prime you put log p. So then say-by-say function is this sum of log p where p power m is less than or equal to x. Then the prime number theorem tells you that this limit of this say-by-say function by x is 1. And why I'm bringing here, you'll see in the next slide. Actually, this is connected to well-known Riemann-Zeta function, which is quite another interesting function. And in fact, there are $1 million writing on it. So I'll tell you more about it. So the Riemann-Zeta function is defined as follows. You take a complex number S, which is sigma plus IT, where sigma is bigger than 1. Then you define zeta of S to be summation 1 of infinite series 1 over n power S, where n is from 1 to infinity. So 1 plus 1 over 2 power S, 1 plus 3 power S, and so on. It has so-called Euler product formula, which is product of 1 over 1 minus p power S. I mean, so I don't, if you're not that clear about it, just don't bother. But basically what it says is that because of this phenomenon of arithmetic is connected, though this is infinite series, doesn't have any primes, but it's actually connected to prime in a different way. And in mathematics, there's something called analytic continuation. So one can analytically continue this function to the whole of complex plane, and resulting function is called Riemann zeta function. Still, we denote by zeta of S. So this is the Riemann zeta function. So first, I will tell you about something. So Riemann zeta function, it satisfies a functional equation involving gamma of S. Don't bother if, I mean, this is just, I mean, you can just think of it as a formula, but it's not that difficult to show this. But what it tells you is that zeta function is related to zeta of 1 minus S by bringing in times some function involving gamma function. And from this functional equation, one can easily show that Riemann zeta function has zeros when S is say -2, -4, -6, extra n negative even numbers, zeta of S is 0. Therefore, it follows from this so-called gamma function, the properties of gamma function, and such numbers are called trivial zeros. So negative even numbers, -2, -4, -6, -8, etc. These are called trivial zeros. And any other zero other than this are called non-trivial zeros. And one can see that, one can show that actually the trivial zeros, non-trivial zeros are those lie between, so the real part of non-trivial zeros lie between 0 and 1. This is the Riemann zeta function. Now the $1 million problem is, I guess many of you might have heard of so-called $1 million problems in mathematics. So there are 7 $1 million problems, of which one of them has been already solved, something called Poincare conjecture. But the 6 others are still open. So among them, this one of the $1 million problem is the Riemann hypothesis, which talks about the zeros of this Riemann zeta function. So the $1 million problem is the following. If okay, let's take S equal to sigma plus IT to be non-trivial zero of this Riemann zeta function, then sigma, this number sigma has to be half. So you cannot have a non-trivial zero other than with real part other than half. So this is the $1 million Riemann hypothesis problem. In fact, it's called one of the holy grails of mathematics. If not for the whole, I mean, for number theory, if not for the whole mathematics, and it is very powerful conjecture, and it has lots of implications in number theory, other areas of mathematics, even in physics. In fact, showing that zeta, so 1 plus IT can never be a zero of Riemann zeta function. This implies the prime number theorem, which is a great theorem on its own. So I just want to tell you how a little bit of connection of Riemann zeta function and primes. So you can differentiate Riemann zeta function with respect to S, and it satisfies this equation n from 1 to infinity lambda n power minus S, where lambda n is, I mean, is equal to log p if n is a power of p and 0 otherwise. And using this, one can derive that the Chebyshev function psi x is x minus x power rho by rho minus log 2 pi, where rho runs over all the zeros of the zeta function, Riemann zeta function. Now, prime number theorem tells you that when rho is not equal to 0, then it gives prime number theorem. So that's not easy. Yeah, so that is easy. So when zeta of 1 is not equal to 0, and we know that zeta of 0 is not equal to 0, so that gives you prime number theorem. but our Riemann hypothesis to show that the rho is of the form 1 plus IT. And once Riemann hypothesis is true, then you see rho is composed of half plus IT, so that gives you psi of x is x plus some function which is bounded in terms of x for half. So O function is what is usually used in mathematics. This means that it's a function which is less than some constant times x for half. So once Riemann hypothesis is true, it gives a good understanding about distribution of prime numbers. So from here, you may not see this, but let me make it a bit more precise. So let me take the sequence of prime numbers to be P1, P2, P3, and P5, etc. So P1 is 2, P2 is 3, P3 is 5, and so on. So this is a sequence of primes. So Pn is the nth prime. Now the question is, we know that there are infinitely many primes, and we're interested in, okay, so what can be the largest, so can you give a bound on the largest gap between consecutive primes? So let's start with the lower bound. So between two consecutive primes, between Pn and Pn plus 1, what's the difference? How big it is, how small it is? One can easily see that there are a couple of primes such that like 3, 5, 5, 7, 11, 13, 17, 19, etc. But difference is 2. So such primes are called twin primes. So let me be more precise. A prime is called a twin prime if P and P plus 2 are both primes. And we call P and P plus 2 as twin prime pair. So as I mentioned, 3, 5, 5, 7, 11, 13, 17, 19, etc. These are some of the twin prime pairs. As of today, the largest non-twin prime pairs is given by P is this 375, this number into 2 plus 6, 6, 6, 9, minus 1. So this number P and plus 1, this minus 1 plus 1. So both are primes, and this is the largest non-twin prime of today. However, one of the important open problems in mathematics is so-called twin prime conjecture. There are infinitely many primes P such that P plus 2 is prime. So this is called the twin prime conjecture, and still we don't know how to prove it. You see, the largest known is just around 20 million digits. No, not 20 actually. 20, around 20 lakh digits. So, but we don't know any other twin prime larger than this. Or we're not technologically that, or mathematically technologically not that advanced to find large twin primes. But conjecturally, we expect that there are infinitely many twin primes. In fact, it's also another open problem in mathematics or in number theory, so that there are infinitely many primes given any positive number L, even number L. So given a positive number L, there are infinitely many prime Pn and Pn plus 1 such that their difference is 2L. These are open problems. So now I want to talk to you about the recent breakthrough of work of Jitan Zhang. So few years back, there's a huge interest after this work of Jitan Zhang from US, who saw that there's a fixed number B such that infinitely many primes, there are infinitely many n such that difference between consecutive primes. Yes, is there any question? Yeah, it's less than or equal to B. In fact, what Jitan Zhang showed was that there are infinitely many primes Pn such that Pn plus 1 minus Pn is less than or equal to 7 T into 10 power 6. And there's a notion of something called polymath project. In mathematics, nowadays, like sometimes people working on a single person alone sometimes is not effective. So some groups of people come together at different locations, then they share and work together, something called polymath project. And there's a work of Maynard. So they together show that there are infinitely many primes Pn such that Pn plus 1 minus Pn is less than 246. So basically, yeah, but we're still very far away from twin prime conjecture. For twin prime conjecture, I have to show that there are infinitely many primes n such that Pn such that Pn plus 1 minus Pn is less than 4, and we have to go from 246 to 4, which is not an easy task. We still have a long way to go. So this is another open problem on the gaps between primes. Now let's talk a bit more on the upper bound of this upper bound on the gaps between primes. Okay, so let me be more. Let me tell here. You see, in other talks, as you have seen many pictures. Unfortunately, in my talk being mathematics, I don't have any more pictures to show. But at the end, I'll show you some nice numbers, interesting big numbers. So that's all I'm going to do there. So let's try to look at the upper bound for the gaps between consecutive prime Pn plus 1 minus Pn. So many of you might be familiar with so-called Bertrand's postulate, which tells you that one of the interesting properties of primes is that given n bigger than 1, there's always a prime between n and 2n. So between 100 and 200, there's a prime. Between 5 and 10, there's a prime, and so on. So, and this is not an open problem. So because of this, Pn plus 1 is less than 2Pn, and hence Pn plus 1 minus Pn is less than Pn for all n bigger than 1. So we have a good, we have a trivial upper bound. So gaps between consecutive primes is less than that prime. Now the Riemann hypothesis, which I mentioned before, will tell you that the difference between consecutive primes is actually bounded by p power half plus epsilon for n sufficiently large. So given epsilon, there's an n which is quite big, such that Pn plus 1 minus Pn is less than p power half plus epsilon. And right now the base, without any condition, the base bound is due to Becker, Hermann, and Prince, which show that there are infinitely many. So for n sufficiently large, p power dn, which is p power n minus 1 minus Pn is less than Pn power 0.525. So actually it's another big problem in mathematics, particularly in number theory, to show that can you reduce this bound 0.525 to something very close to 0.5? If not 0.5. So that's it has wide ramifications, at least in mathematics, and and also in understanding of primes. So now I'll talk. So these are some of the results. Now I want to talk to you more bit about my result, which is around 5-6 years old. In a joint work with Ram Murthy, so we actually saw that. I'm going to tell you what is Green conjecture. So Green conjecture tell you that for n sufficiently large, the Pn plus 1 minus Pn is less than p power 0.46. So basically, we could get a result better than Riemann hypothesis if you assume Green's conjecture. And there's another famous conjecture in mathematics, which is also interesting conjecture, which tells you that, okay, so between 2 square and 3 square, there's a prime. Between 3 square and 4 square, there's a prime, and so on. So you can keep on continuing. And what we expect is that given any L, there's always a prime between L square and L plus 1 whole square. And by the way, this is we don't know how to prove it for all L. But if our Green conjecture, if you assume Green conjecture, and from our result, we can show that this legendary conjecture is true. In fact, the Green conjecture implies legendary conjecture. So this is quite easy to see. So suppose this, there's some L such that there's no prime between L square and L plus 1 square. Then you have n for which Pn is less than L square is less than L plus 1 square is less than Pn. Then from this upper bound here, I've put p power 0.46 is bigger than Pn plus 1 minus Pn. And you see Pn is bigger than L plus 1 square, and Pn plus 1 is bigger than L plus 1 square, and Pn is less than L plus 1 L square. So if you take the difference, then it's bigger than 2L, and L is bigger than Pn power half, so it's bigger than 2 times Pn power half. And here you have a power 0.46, which is less than half, and here you have half, so it's a contradiction. So basically, what we saw was that this under Grimm's conjecture, they infinitely there are, I mean, for between any two consecutive squares, there's always a prime. Now let me tell you the Grimm's conjecture. This is a conjecture even a high school student will be able to understand. So this is a very easy looking conjecture, but atrociously and innocently difficult problem. So this was formulated in year 1969. It tells you the following. You take a sequence of primes n plus 1, n plus 2, n plus k, which are a sequence of numbers. So, and suppose these are all composite numbers, then you can find distinct primes p1, p2, pk, such that p1 divides n plus 1, p2 divides n plus 2, p3 divides n plus 3, and so on. So this is just this conjecture. I mean, this statement. So any student, even high school class 5, 6 student can understand this, but unfortunately, we don't know how to prove it. So let's look at some observations. So you know that 24, 25, 26, 27, 28 are primes. No, these are composites. So now I can find distinct primes. So 7 which divides 28, 3 which divides 27, 13 which divides 26. So I can find, so for every number I can associate a prime, a distinct prime, such that which divides them. So this is how it works. Let's look at another example. So 90, 91, 92, 93, 94, 95, 96, these are composite numbers. You can see easily. And okay, so I can choose distinct primes. So for 95, I can choose 19, for 94, 47, 93, 31, 92, 23, 95. And 91, I can choose one, either 7 or 13. 96, I can choose 2 or 3. So not only one way, there can be more ways of choosing this primes, such that P1 divides n plus 1, P2 divides n plus 2, and so on. So this is the Grimm's conjecture. So the conjecture tells you that this is true for all n, such that n plus 1, n plus 2, n plus k are composite numbers. So though we were not proved it, but in a joint work during my PhD, we verified this conjecture for all n bigger than 10 power 10 for and for all k. So basically, if n is less than 10 power 10, the Grimm's conjecture is true. So we use some combinatorial arguments and some computation called some combinatorial argument called Marais theorem, which is a very interesting theorem in graph theory. So I won't go into the proof. This is how we do it. Now I want to tell you a bit about this Grimm function. So we say that a pair of positive instants nk has a prime representation if you can find distinct primes. P1 divides n plus 1, P2 divides n plus 2, P3 divides n plus 3, and Pk divides n plus k. So here I'm not assuming that n plus 1, n plus 2, n plus 3, these are composite numbers. Now I define so given n, I define the number Gn to be maximum of k such that n, the number pair nk has a prime representation. Now it's just it's not that difficult. It's quite easy to see that if we take n is p power r minus 1, where p is a prime, then n plus 1 is p power r, n plus 2 is 2 times pr. So and between PR and 2 times PR, there's a power of 2 in between that one can see. So numbers n plus 1, n plus 2, n plus 2 cannot have a prime distinct prime representation, because for n plus 1 I can only choose prime p, for n plus I can choose prime 2, and I don't have any prime to choose for n plus 2. So this tells you that Gn, which is less than n, Gn is less than n plus 1 for infinitely many n. So this gives you a trivial upper bound, which is true for infinitely many n. Now one of the important problems is to show, can you find the best possible upper and lower bounds for G of n? So in this regard, there are some results. So on the lower bound part, so there is the best known result due to Ramachandra Soren, Soren is my PhD guide. So they saw that there's some consensus that when for n bigger than or equal to 3, gn is less than c times log n by log n cube. So don't bother much about it. So basically, we don't know how to get. I mean, right now this is the. I mean, gn is at bigger than or equal to around log n cube. We don't know if we can replace log n cube by some big power of log n. On the upper bound, so what we actually saw was that gn is less than n for 0.46 for sufficiently large n. So this was joint work with Ramurthy, and what we are able to show was that the function Grimm function gn, though the upper bound was n, but we could show that is less than n for 0.46, and not only that, I mean, there are only few numbers up to, I mean, given large number x, there are only few numbers n for which this is bigger than n for epsilon sufficiently large. So you can ignore this part, but what I want to tell you is that the gn is less than 0.46. So this was a key work which I did with Ramurthy, and in fact, yeah. So there's a notion of so to prove this, we use something called smooth numbers. Yeah. So so this is a little bit more technical, but I'll try to give a glimpse, and then I won't go much into detail. So we say that a positive integer n is said to be y smooth if the largest prime factor of n is less than y for y. So that means every prime factor of if it doesn't exceed y, then n is said to be y smooth. For example, 100 is 5 smooth because every prime factor of 100, that is 2 and 5 are less than or equal to 5. So we call these numbers as smooth numbers. Interestingly, let me share with you that in number theory, we borrow a lot of terminology from this or no. Sometimes people point different like spoon number or strong numbers and different. So depending on their interests, depending on their properties, and depending on their.
Shanta, your voice is very low.
Yeah, is it better?
Yeah, it's a bit audible.
Okay.
Can you say something?
Yeah, is it better now?
Yeah, it's coming a bit better.
Okay, so maybe I'm going to hold like because I'm using my mobile for talking because.
Yeah, it was going good, but suddenly it just go down.
Okay, I hope the battery is not down. Just let me check it. Oh, okay, but battery is still there. Okay, so. Oh, okay. Yeah. So let me try to keep it close to my mouth so that it's fine. Yeah.
Yeah, it's better. It's better.
Okay. So the smooth numbers. This is what it says. So this is the yeah. So numbers which are composed of fixed primes, primes less than some fixed number, and if psi of x alpha counts the number of primes, the number of psi of x xy counts the number of integers less than x, which is y smooth, then it's like some function in terms of 1 over alpha. So I won't go into it. This is a little bit mathematical, but basically up to x, they're given alpha up to x, they're around rho power 1 over alpha y smooth x power alpha smooth numbers. So there's a interesting and more one of difficult problems in this about smooth numbers, which tells you that given epsilon positive for sufficiently large x, so a fraction of numbers between x and x power x plus x power epsilon are x power epsilon is smooth. So basically, if you take x plus 1, x plus 2 up to x plus x power epsilon, then there are a fraction of numbers, fraction of x power epsilon numbers which are x power epsilon smooth. So this is open, and if you assume this Grimm conjecture and smooth numbers, if you assume smooth conjecture, then I can show that Gn is less than n power epsilon for sufficiently large n, and that gives you a good upper bound for the gaps between consecutive primes. So basically, given epsilon positive, you can show that given epsilon positive, there's n sufficiently large such that Pn plus 1 minus Pn is less than Pn power epsilon, where constant times Pn power epsilon. So this is getting a bit more technical. I mean this, but what I want to tell you is that even understanding about smooth numbers, it can give a wide ramification for this distribution of prime numbers. So similar to this primes, we can talk about some interesting property like primes in arithmetic progression. So take a arithmetic progression, say 1, 1 plus 3, 1 plus 2 into 3, and so on. So like 1, 4, 7, 10, etc. So in this sequence, are there infinitely many primes? So this is called Dirichlet's theorem, which tells you that every arithmetic progression a plus q, a plus 2q up to so this sequence will with coprime a and q contain infinitely many primes. So this is the well-known Dirichlet's theorem, and if pi of xqa, if this is the number of primes in this sequence with term bigger than or equal to x, then number of primes up to x is like x by pi q log x, like the prime number theorem. So this is the Dirichlet's theorem, but still theoretically it's nice, but finding primes in a long arithmetic progression is a big challenge. In fact, it's an interesting question to ask. Okay, so if primes in a certain residue class, for example, if you take residue class 5, so can there are infinitely more primes in 3 mod 5 than 2 mod 5 or 1 mod 5 then? So these are kind of questions. Again, this is purely of theoretical interest, but then it is also interesting other consequences in other areas. So this is one aspect where it's quite mysterious. We don't know how to actually answer this. Another problem is, another theorem is this Green-Tau theorem. It was proved around 2010. It was one of the quite, at the time it became quite famous. Basically, it tells you that in the sequence, so what Dirichlet's theorem tells you is that if you take an arithmetic progression, then there are infinitely many primes. Now we ask another question. So take a sequence of prime, look at the sequence of prime, then given k, can you find an arithmetic progression of blank k? That's the converse thing. So I'll repeat it once again. Look at the sequence of primes. Now, can you show that? Can you say that there are given k? So, for example, are there 100 primes which are in some arithmetic progression, 200 primes in some arithmetic progression? The answer is yes. This is due to Green and Tau, and it was quite famous. But however, numerically, we don't know an arithmetic progression of primes of length bigger than 26. Right now, the largest known is this number of length 26, which is given by number written here. This is a big number. So 4314274. This is the prime, and common difference is this number. And this was discovered in 2010. After that, we don't know if, we don't know of any example, though we know that it exists, but it's a challenge, even computationally, to find large examples. This is on this sequence of arithmetic progression in primes. And so now I can talk to you about, okay, so there are many other questions which are easy looking, but we don't know how to prove it. Like if you look at this sequence of primes, so sequence of number 2 power n plus 1. For any n, you look at fn to be 2 power n plus 1. Then these are called Fermat numbers, and we know that f1 is 5, f2 is 17, f3 is 257, and f4 is 65537. These are known. These are primes, and these are known as Fermat primes. The question is that are there infinitely many Fermat primes? Or what we expect is that for n bigger than 4, it's not a prime, but we don't know how to prove it. This is known as Fermat prime conjecture. And another problem is are there infinitely many primes of form 2 power n minus 1? So this the primes of this form are called 2 power n minus 1. These are called Mersenne primes. So are there infinitely many primes of this form? This is, we don't know it. And also like a square plus 1. So n square plus 1, like 2 square plus 1 is a prime. Then 4 square plus 1 is a prime. 6 square plus 1 is a prime. So now are there infinitely many primes such that n square, which are of the form n square plus 1? This is a problem which we don't know how to prove it. In fact, for any number r bigger than or equal to 3, between n power r and n plus 1 power r, are there sufficiently large, so are there infinitely many, are there primes? We don't know how to prove it. So these are some of the properties which we expect is true, but we don't know how to prove it. These are quite difficult. We don't know how, we have no idea how to go about it. And another thing is, like even in, all of you know about this Fibonacci sequence. So these are so-called Lucas sequence. So basically start with a co-prime integer r and s such that r square plus 4s is 0. Put u0 is 1, u1 is 1, u0 is 0. This should be 0 typo. u1 is 1. And un plus 2 is r times un plus 1 plus s times un for all n bigger than or equal to 0. So these are called Lucas sequence of first kind. So these are binary recurrence sequences. When r and s are both 1, so that means you look at sequence 0, 1, 1 plus 1 is 2, 2 plus, yeah, so the next term is sum of previous two terms. So this is a sequence of Fibonacci numbers. So 0, 1, 1, 2, 3, 5, 8, and so on. So these are called, this is Fibonacci sequence. Even in Fibonacci sequence, if I know that there's certain prime 5, 13, 18, 9, and so on. Okay, are there infinitely many primes in Fibonacci sequence? We have no idea how to go about it, how to prove it. It's an open problem. In fact, the problem about Mersenne primes, which I mentioned, 2 power n minus 1 is a prime for infinitely many n. That's a special case of this problem. So 2 power n minus 1 is a Lucas sequence with r equal to 3, s equal to minus 2. So this is one interesting aspect. So basically we, so problems are limitless or mine is limitless, but still we have long way to understand about primes in the sequences and so on. Now, so I'm coming towards the end of the talk. So I want to talk to you about this, a simple number as factoring. So if I, always we know factoring. So if I give you 6 and I ask you factor, you know, okay, this is 2 into 3, very simple, you will say. And so if I give you the number 77, then you can see easily that this is 7 into 11. This is quite simple. Yeah, so you can easily factor it as 7 into 11. Now, if I give you this number 11639, so and I ask you to factor, it's not simple, but it's not difficult either. You'll take a little bit more time and you'll factor it as 103 into 113. Life seems, so a problem like given any number, can you factor it? This is a simple looking problem, but it can be quite difficult. Now I give you this number and I ask you to factor. So can you factor it? Then life is no more simple anymore. Then it's quite difficult. Like if I give you this number and I ask you to factor, I don't think, like unless you use the latest technology, latest methods, how these numbers are factored. If you try to do it by the normal way to factor this number, I don't think your lifespan will be sufficient to factor this. But then this is so-called a RSA 240 number. So I'll tell you about RSA 240. So there's this RSA factoring challenge. You can so go to Google and see. So RSA laboratories, so they design protocols for RSA crypto system, which are well-known crypto system to make our internet communications or communications secure. In fact, RSA is used in our this ATM cards, debit cards, and credit cards, and many other places. It was one of the first public key crypto systems. I won't go into it, but so basically it works with the principle that factoring large numbers is difficult. So you need large primes and a product and a number which is a product of two large primes. So and it's only USP or and we think that is secure only because you cannot factor large primes. So RSA numbers are numbers which are product of two primes, and right now RSA Laboratories is taken over by AMC Corporation. So AMC Corporation, this RSA factoring challenge is that they give you a list of numbers and ask you to factor it. And sorry. And of course, they announce prices ranging from $1,000 to $10,000 to factor those numbers. They give prices. So in December 2019, a research team led by Emmanuel Thom at INRIA in France, and with some group in some other group in France, they together factored that RSA 240 number. So these other team members included Fabrice Baudou, Patrick Gaudry, Oro Gulevic, Nadia Henninga, and Paul Zimmerman. So they factored this RSA 240 number. So why this is RSA 240? Because it's a 240 digit number, which is the RSA number. And number of bits is 795. And I'll tell you the factors. These are the factors. See, as you can see, this is a number which is product of two primes. So 240 digit number which is product of almost same size primes. And it looks very nice when you see the factors here, but let me show you how much time they took it. So to factor this number, they spend around 900 core years of CPU time. So basically 900 core years was on a 2.1 gigahertz Intel Xeon Gold 613 CPU. So if you work with this, then if you know about computing, you'll know that it took 900 core years. So this is a step called sieving step. So it took 800 physical core years. And RSA 24 matrix step, it took 100 physical core years. Actually, they did this factoring along with another discrete logarithm problem, where together it took around 4,000 core years. So basically, factoring took 900 core years and other took 4,000 core years. And they use something called Kedo NFS, which is open source software based on this number, which is to implement number field shift. So I cannot explain it here because it requires more advanced mathematics. And Kedo NFS comprises 3,000 lines of code written in C and C++. So so basically a problem which is a very easy to state and easy looking problem, like can you factor large number, it's it can be quite difficult. Actually, it's quite difficult, and that's this makes our communication secure. And so I just want to tell you that primes play an important role in making our communication secure. And the widely used crypto system, RSA crypto system, so it's based on primes. So to make the system secure, again, we need big primes, because you see, with technologically more advanced and more computationally more advanced, if I use a small prime, one can factor, so it's not good. So we need again to find large primes. So we need a new method to factor large primes, or a new method to find large primes, and also new method to factor large numbers. But we still have a long way to go in finding faster algorithm. So let me stop here. Thank you very much. So any questions, please feel free to ask me.
Question session? No question. Thank you, Dr. Shanta, for a nice presentation. We knew about the number theory. Thank you.
Yeah.
So now,
maybe I'll take a break. So I want to tell you a sort of joke. I mean, not the joke, but it's kind of So in a lecture, so if everyone, if there are no questions, that's either it means that either you have understood completely or you didn't understand anything. I hope.
No. That is not a thing.
This is just
but
a job. What I just want to tell you. I'm just
but
sharing. But
I think we are not in that area, so
yeah.
that's why we have
I
not
can
any
understand.
query.
But anyway, I just want to share something. So that's
yeah.
Okay. Thank you.
Thank you. Now I invite Professor Anurag. He is working as an associated professor in Geetgovind College of Engineering and Technology. He is PhD from IIT and is working in the synthetic nanomaterial. So he will deliver his talk on nanoparticle for high field magnetic resonance imaging application. Dr. Anurag, please.
Yes, I'm here. I'm trying to, thank you. I'm trying to share my documents. So one moment, please. Document.
Hello, Dr. Anurag.
Yes, I'm here. I'm trying to share
okay.
my document. One moment.
Okay.
So you can see. Okay. So let me.
If there is some connectivity issue, then if you can off the video, then somehow it also works like that.
I can't see. Actually, it's a connection, but I can see my document. That's a device storage and then downloads. But one moment. Yes, I got it.
Yeah.
Can you see the screen?
Yes. You can proceed now.
Yes. So my talk is, I'm working at Geetgovind Engineering College of Technology, Hyderabad, and my talk is on nanoparticle for high field magnetic resonance imaging and applications. So what I am doing here, I'm unable to continue my research here because it's mostly the private college and lots of teaching, but I will go through some work I have done in the University of Victoria on MRI and applications. So I will go on that and explain how I did there. And currently I am working on the gold nanoparticle for wound healing and some applications. So I will cover that too. So what is actually the MRI? As you all familiar, this is for imaging tool that stands for magnetic resonance imaging. It is a process where the contrast agent is used to improve the visibility of internal body structure or cell also. It can improve to the cell too. So basically this is categorized into T1 and T2 process, where T1 is the longitudinal relaxation of water proton and T2 is the transverse relaxation of water proton. So we have the body and lots of water molecules, fat molecules, and all around. So under MRI, we can differentiate each and every one from which part it is, what is, you can do that. Because the water proton relaxivity, the T1 is 3800 millisecond. Now, the T2 for the water proton is 3600 millisecond. So we have to brought back this to the really low, where they relax and energy is recorded. So we try under MRI, we develop the contrast agent that bring this water molecules relaxation from 3800 millisecond, that is T1, to very short, that could be 6 millisecond or 0.2 millisecond. Short is the better, because that will lead to better R2 relaxivity, because we deals in terms of how much the relaxivity it is, not the relaxation time. So we said, let's the relaxivity of particular R1 is how much. So R1 we calculate with respect to the relaxation time divided by normalizing with the concentration. So we can say R1 equal to, R1 is proportional to 1 by T1 divided by concentration of the nanoparticle. And similarly the R2. So, and the ratio is also improve the contrast. And any material you inject in body or throw in water that have both R1 and R2, it will be somehow like one is higher than other, and that's how we say it is R2 process or the R1 process. So suppose R2 by R1 is higher or dominant, then it is a T2 contrast agent, and if R1 by R2 is higher, then it's a T1 contrast agent. So that's how the relaxivity works. So we basically we use lots of the paramagnetic compound for this, and like such as gadolinium, Mn2+, iron 3+. These are potent desirable T1 contrast agent. Until now in literature, whereas dysprosium and holmium has high magnetic moment, that is 10 Bohr magneton, they are T2 contrast agent. And even the iron 2 is a potent or super magnetic iron oxide is also T2 contrast agent, because that can generate higher magnetic field during the operation. So the gadolinium-based complexes has been widely used. Till now we are using the complex that is for MRI application. It could be gadolinium DOTA or some other gadolinium-based complexes. So that gives both T1 and T2 contrast, because there are two coordination cells. I will explain later. If the water molecule directly binded to the through the chelate gadolinium ion, then it's going to lead the T1 contrast, whereas overall effect and the second coordination of the water proton, that is giving the T2 contrast. So that's a resident time. How short time to connect with the gadolinium ion and getting away. If the faster the relaxation rate, more is the contrast. So can gadolinium ion work as the gadolinium was already? Can nanoparticle also work? Lots of work has been done in the past. So I will quickly come on the what are the technique we use for synthesis of any nanoparticle generally. Then we'll go to, again back to. So currently, this is for mostly for the beginners who are new in the field of the synthesis of nanoparticle. So two way we do top-down approach or the bottom-up approach. So top-down means the bigger particle you are taking, disintegrating to the lower size, micron size or nano size. That one could be the ball milling. How we generate the ball milling? Ball milling, we generate lots of the rotating ball with certain RPM. They generate lots of kinetic energy, and those kinetic energy transfers to the particle in that, and they disintegrate to micron size and nano size. Sometimes it's hard to achieve the nano size with the ball milling processes. Then come the lithography, where you use the substrate to cast any shape, or the you apply the light source to cast any shape on the substrate. So this is a substrate based technique mostly for the electrical or electronic application. Then comes the pyrolysis. This is again where the nanoparticle of bigger size goes under high pressure and temperature. They disintegrate to the smaller size to nano level also, but this may not be advantageous because high pressure you need, and still it is an industrial process, but high pressure you have to manage, and temperature too. Then could be thermolysis, again disintegration process. So then comes the next is the bottom up, where you start from the bottom, like from the ions to atom to cluster to nanoparticle. That is easily doable in the laboratory scale, and even the industrial scale too. So in this process, the one can be the chemical reduction process that is mostly known. And so you can use the mild reductant or the harsh reductant depending on the process. So in this category, the second could be, let's say harsh reduction could be sodium bromohydride, or sodium boron hydride, or lithium aluminum hydride. So these are harsh reductant, but we can use some mild. So they may not be as applicable to the green synthesis. That's why nowadays most of the nanoparticle are coming by green synthesis, where the harmful effect of the synthesis for bio application can be minimized. So green synthesis is the other process where we use the synthesis reductant from the plant, leaf or root. Then comes the polyol process. This methods where you use the polymer as a encapsulator and reducing agent as well, or you can use external reducing agent too. Then comes the microemulsion given by Schulman and Way in, I think, 1961. And this is where you three-phase component system you use. One is polar, non-polar, and emulsifier with a surfactant that bring from the one phase to. So surfactant is at the interface of the polar and non-polar phase and forms lots of microemulsion of different size. Size is hard to control in this process because microemulsion can have different size on the interface. Then the next can be the laser ablation technique, where we use the Ply ladder to reduce the ions to atom to the cluster, and then it goes to the nanoparticle. And the similar technique is the micro radiation tool. So then becomes this work I didn't pass for the application. So we can say that if you use the synthesis for this is polyol method for synthesis of the silver nanoparticle and the gold nanoparticle, but still these can be helpful. These can be helpful because these nanoparticle has higher absorption cross section, because I'm going to talk on mostly on the lanthanide. Those have narrow absorption cross section. So if we coat those nanoparticle or connect those nanoparticle with this gold or silver nanoparticle, so we can increase the absorption cross section of those nanoparticles. That is a big hurdle for dual modality. Actually, dual modality means on the single step, if you want to perform MRI and optical imaging, we want to see what is the dimension of tumor, where is tumor. Though MRI will do, but you can see optical visibility is second pair. So if those absorption can increase by attaching of the gold nanoparticle and silver nanoparticle, so this also have been used in past to in the synthesis coating as a coating to improve the absorption cross section of the lanthanized nanoparticle. So then they have some work in the past have been done, like you can even for the sensor they have been widely gold nanoparticle for sensor. So I'm skipping this one. They have done, and then why we are using, because the lanthanide based nanoparticle, that is sodium dissocium fluoride or sodium gadolinium fluoride, that is getting used in the MRI application. They have two windows, in first NIR window and second NIR window, and the tunability of this gold nanoparticle with the respect to the their length and width is long, still first window to nearly to second window. So we still, by applying this gold coating, we still can increase the absorption cross section of the lanthanide based nanoparticle, because they have really narrow absorption cross section due to 4FF transition between them. Then we come how we are doing the synthesis of this nanoparticle, and this is the synthesis of sodium dissocium fluoride nanoparticle. So basically in this synthesis we use Senclane and use 1 millimole of dysprosium chloride plus oleic acid plus octadecene, and the mixture was heated to 130 degree under vacuum, and that will give the dysprosium oleate. And then you have to cool down the mixture to the room temperature, let it cool to 25 degree, then add 2.5 millimoles of sodium hydroxide and 4 millimoles of ammonium fluoride, and mix the mixture at room temperature for 1 hour nearly, and then heat the mixture again to 65 degree to remove the methanol, because we use methanol to mix all together. Methanol based mixture of 2.5 millimole all. So we have to evaporate methanol because that will be harmful in the controlling of the nanoparticle size. And then we heat the particle from heat the flask under argon flow to 300 degrees centigrade for 1 hour 20 minutes. But this reaction, all lanthanide based reaction, particularly sodium yttrium fluoride, sodium dysprosium fluoride, or sodium gadolinium fluoride, they are tricky. They are very similar property and in the same group, but their synthesis is not easy, because for the synthesis of sodium dysprosium fluoride, it's 1 hour 20 minutes, and then also you have to control lots of parameters to get the uniform sizes. I will show later how I control. But the yttrium, I had that's a very easy synthesis, but you have to be very careful while doing synthesis with sodium yttrium fluoride. It's 1 hour. If you're crossing the 1 hour more, just 1 hour, suppose reaction time passed by 1 hour 5 minutes, all the nanoparticle will collapse. You will find non-uniform particle due to the Oswald process. This Oswald process, I will come later, that's sustained till 1 hour, and after that they will become really non-uniform, all the particles. Then the mixture was, in this case, we wash the mixture and 3 times with ethanol, and it is dispersible. So as I said, the oleic is there, so that will go on the surface. That will give oleate stabilized sodium dispersion fluoride nanoparticle that is dispersible in hexane, chloroform, or some other organic material. They are dispersible because oleate is on the surface. So the growth process, basically for the, as we said, I think most of you are familiar with this one, hopefully. So how they grow. So there is a 2 contribution on this one, surface and volume contribution for the growth of nanoparticle. And so surface contribute for spherical particle of radius r, and surface energy per unit area is sigma, and the free energy of the bulk particle per unit area is delta GV. So the overall energy will be sum of, overall gives energy will be sum of the surface contribution plus volume contribution. And if we disintegrate this equation with respect to the radius of the nanoparticle, we will get the critical radius. This is 2 sigma by delta GV. So this, if the nanoparticle is below this RC, they will dissolve and they will grow on the surface. That is also a process of Oswald ripening. And if the energy, critical energy at that point is 16 pi r cube by 3 delta GV square. So this is the critical energy at that point. That is minimum energy needed to nanoparticle to grow or crystal to grow. Below that, they will redissolve. So then that's also a process of Oswald ripening, where the small particle grow on the bigger particle surface. So, and the MRI rely on the principle of NMR. So NMR was introduced in the clinical practice first in 1981. So initially the committee decided to put the name of NMRI, but they later dropped the name M. The reason was the N is related to, nuclear is related to radioactivity that could be harmful, but this is not harmful. So later is dropped to MRI. They dropped the N from the NMRI. So this is basically on the principle of MRI and the process. So this is a scanner on the next slide, how it works. So whole body can go in the MRI machine, and any part of the body can be imaging through that process. Then what are the advantage of MRI? It could be, advantage could be, this is a non-invasive technique. So we don't put anything like inject in body or something. Unless we are applying the contrast agent, then we have to, that's also oral, take the oral. So we are not digging the body or injecting something with the sharp thing. So this is non-invasive technique. And the spatial resolution of MRI is 10 times greater than the PET, that is positron emission tomography. So MRI provides better soft tissue contrast than the CT, that is computed tomography, and can differentiate better between fat, water molecule, and soft tissue than the CT. So MR imaging is not specific to the particular molecule or cell, but it can imaging any part of the body or even the cell. So MR imaging needs to increase its specificity or sensitivity by means of applying a contrast agent or applying a high magnetic field. Because the higher the magnetic field, larger the splitting. So when we place the sample in the magnetic field, when the magnetic field, though when the field is not there, they are randomly aligned. But when you place in the magnetic field, they will align with respect to magnetic field that is along G-axis. So all the magnetization is along G-axis. So we have to apply the radio pulse. On that point, the radio pulse is applied, and the G-axis magnetization is brought back to XY plane. So all the magnetization is coming to XY plane under the radio pulse frequency. When you switch off the radio pulse, so this loss in magnetization, so bringing from the G-axis to XY plane, this was the loss in magnetization, is actually the T2 contrast. That is transverse relaxation of that water molecule. And when you switch off the pulse, they try to go back from XY plane to G-axis. So gain in magnetization is T1 contrast. So these signals are recorded in the MR imaging machine with the computer. So general magnetic moment for the lanthanide ions predicted is given here. So if you see the dysprosium and helium is high, it is already said 10.2 for magneton. It's really high. So they are usually the T2 contrast agent, and gadolinium and other are a T1 contrast agent. So though there are both T1, T2 contrast agent, I already said they have, though they're very less T1, but still there. So because both processes are there. So how that is DOTA, gadolinium DOTA that is used at the clinical level works. So the water proton reaches to get the T1 contrast, the water molecule, after administration of the drug, the water molecule should go in the proximity of the gadolinium ion. If they are getting attached and running away immediately from that, relaxing faster, that will lead better T1 contrast. And whereas the overall migration of the nanoparticle, no matter is coming from, you can see this is second sphere is the red marks that is outside that second sphere. The first sphere that is the direct in contact with the gadolinium ion. So the overall relaxation, there is relaxation from both coordination sphere, that is from first, second, both. But for the T1, it is mostly coming for the first coordination sphere, where the water is directly contact with the gadolinium ion. So whereas the overall migration is giving the T2 contrast. So two design can be used. One could be the keeping T1, because T1 contact is important in T1 relaxation rate. So one can keep the outer sphere in the case of nanoparticle gadolinium, so that gadolinium should be in direct contact with water. If we keep dysprosium outside, that is T2 contrast, then water may not reach able to the surface of the gadolinium. So that will inhibit the T1 contrast. But on the other hand, if we make the porous structure where gadolinium is inside that porous structure, then still the water can reach to that gadolinium ion. So that will give the T1 contrast still. So two design is possible for this. One could be one can keep the gadolinium ion outside, or any T1 contrast generating material outside, and inner core could be T2 contrast agent. Or on the other way, you can make a porous such as silica structure and dope with the inner core with the gadolinium ion. So that will still lead to the T2 contrast. That will still lead to the T1 contrast. And so multimodal imaging, I already said how we use the gold nanoparticle can be used to increase the absorption cross section of the these narrow lanthanide ion, which have the narrow absorption cross section. So this is a scheme for multimodal imaging, where in case of sodium, if you are making outer core of sodium gadolinium fluoride, it is in the sense that still can generate the T1 contrast, and the inner core is sodium dysprosium fluoride. So if we want to do the imaging T1, T2 both at like image T1 MRI contrast, that is the brightening contrast, and the optical imaging also, optical picture also, then one has to use the dopant in that thulium or erbium, and you have to give light through the body, 980 nanometer NIR radiation that will excite erbium, and the energy transfer to the thulium that will give red. If we go further this with the gold, this narrow absorption cross section of thulium YB can increase. So that will give the better contrast in the signal. So that's how the gold can be used to enhance the further absorption of these material. And these are energy absorption cross section and energy level of the lanthanide ion. I'm skipping this one. And then results, we did synthesis of different size of nanoparticle. The first is 0.5 nanometer, second is 10 nanometer, third is approximately also 10 nanometer on high magnification, and the D is the 20 nanometer particle of sodium dysprosium fluoride for the T1, T2 contrast, because these are T2 contrast agent. And then we also did the core cell structure. For TM image, you can see this is just sodium dysprosium fluoride 20 nanometer, where the B is the sodium dysprosium fluoride coated with sodium gadolinium fluoride to generate both T1 and T2 contrast during MR imaging. And so this one, yeah. So lanthanum fluoride. And then we did also check the relaxivity with respect to, like, because as I said, the relaxivity depends on the parametric nature of the lanthanide or magnetization of iron oxide. So yttrium do not relax. So we try to coat yttrium and dysprosium over yttrium. So what try to seal the magnetic moment of dysprosium by coating with yttrium. And this paper is in JPCC, where we saw that when you coat with yttrium on the surface, dysprosium in the coat, the relaxivity dropped drastically. So because the magnetization parametric nature of the dysprosium is not reaching, or the water proton is not reaching to the surface of the dysprosium ion. So we did all different type of core cell structure earlier. And these are the X-ray diffraction. Mostly they are in the cubic when they are below. These nanoparticles are mostly in cubic shape when their size is below 5 nanometer, no matter what process is used. If size is 0.56, approximately 5 to 6 nanometer, they are in cubic shape. But as size grows more than 10 to 12 nanometer, they become hexagonal. That means they re-dissolve at the stage, the crystal structure re-dissolve and they grow to the hexagonal crystal structure. So then comes the SCID measurement data, where different size of the nanoparticle, 5, 10 and 20 nanometer, this sodium dysphosphoride fluoride nanoparticles, SCID has been measured. So as you see that more the number of dysprosium ion in the nanoparticle, greater is the magnetization. So for the 20 nanometer particle, there will be the highest magnetization, that is approximately close to 10 EMU per gram, whereas for the 10 is slightly lower, and for 5 nanoparticles is still lower. So for the T2 contrast, as I said, overall magnetic moment is very, very important. So bigger the particle, larger is the magnetic moment, and better is the T2 contrast during the MR imaging. So, but the hurdle is with these nanoparticle is because as the surface is covered with the oleate, so taking them in water is the big hurdle. I think that's the biggest hurdle, in the sense that whatever particle we are making in the hydrophobic medium, taking to hydrophilic is the big hurdle, because then sometimes your ligands goes away and the clusters they agglomerate and the size change. So these we struggle a lot with that. So further we modified this this structure of oleate stabilized sodium lanthanum fluoride nanoparticle with PMO-PG polymer to make them water dispersible. Though in the literature PG only used, PG also have been used, and PMO is re-modified and used. So that modifying with the surface modification is mostly to make them water dispersible or make the hydro. So in our case, we make them water dispersible by the surface modifying. So they become water dispersible, and you can see that after water dispersible, we did the TEM, and TEM looks uniform with the particle distribution around 4 to 15 nanometer. For this sample in particular, they are not 20. So this is still between 12 to 15 nanometer. And then again, we measure the relaxivity for all the particles. So measure relaxivity, when we measure the relaxivity T1, that we measure relaxation time, but from the relaxation time, you have to normalize to relaxivity by R1 proportional to 1 by T1 divided by the concentration. Then you get the relaxivity. So the relaxivity we got for the dysprosium fluoride also, there is T1, but R1 is very low. The R1, as this is the only T2 contrast, so this is R1 or is very, very low, that is 0.2 to 0.4. But the T2 contrast is R2 relaxivity is higher. That is because when you increase the magnetic moment, the relaxivity is very, very high. So that is R2 relaxivity is approximately 120 per millimolar per second. So, and you see at the lower magnetic field, whereas the field is low, the relaxivity is less. It's 20 per millimolar per second. But as the field increases, the relaxivity increases to 120 per millimolar per second for same size of particle. That's why there is needs of high field magnetic resonance imaging application of the nanoparticle. And why we can't use the gadolinium-based chillets? Because they tumble fast at that field. Tumble means they start rotating around the axis, and they do not generate the contrast. So that's one reason why the nanoparticle need, and why we need to, they need, because we can tune the tumbling time with further their functionalization, controlling the shape. So these are lots of any desired size we can tune. And for the gadolinium ion, and with the functionalization size will not vary much as compared to the nanoparticle. And additionally, the nanoparticle will generate per unit, per millimole contrast very high, because there is thousands of ion in one nanoparticle compared to the one gadolinium ion in the chillet. So these are the results that's comparing the how the relaxivity measurements compare with the all-label literature or the all-label contrast agent that is used at the clinical level. So for the 21 nanometer particle or 20 nanometer particle, the R2 relaxivity was 101 per millimolar per second, or that was very high with respect to the per nanoparticle. That was 6,479. And the ratio was R2 and R1 was also high, that is 306. That is drop for 10 nanometer particle was, the R2 was 350 per millimolar per second. That again, the R2 by R1 is still high, that is 230. And the 5 nanometer particle is still lower. So if we compare this with the commercially available T2 contrast agent, they are way high. Like the Feridex, that is iron-based compound, that is only R2 by R1 is just 22.6. And Rezovist, again, I think it's a gadolinium compound, that is 31. So with respect to this compound, our design showing will have the higher R2 by R1. That will be further modification and further improvement could be better contrast agent will be in the future for the lanthanide-based nanoparticle or even the iron-based nanoparticle. So we further study the toxicity with this nanoparticle, how they are toxic. So we injected these nanoparticle in the mouse body. This work I already studied. I studied in the University of Victoria, Canada. So that's from there. And how much we injected nanoparticle per kg of mouse body weight is given. So our observation was more like surface one, like they were happy and playing, making nest, fit into the blood sample analysis where they are gone or how they are dead. But we study basically their behavior study, and they look fine at the end of the 7 days. They were nearly fine as same before injection. So then we did the biodistribution study, how the biodistribution occurs. So after injection of the nanoparticle, the injected in this one was 6.6052 mg per kg of mouse was body weight. And our study suggests that the liver was the most dominant organ which took the nanoparticle, that took nearly 0.4 mg of that. Then the spleen was 0.5 mg, and whereas the brain was least, and kidney, pancreas little higher than the brain. Brain was least due to the blood-brain barrier, because nanoparticle can cross the blood-brain barrier. So only the virus can cross, I believe. So this is the MR imaging. Next is how we did the MR imaging with this one. This work was done at the Alberta University, I think, clinical facility. So after injection, or before the injection, the nanoparticle, they were whitish contrast of the tumor. In this area, figure A, one can see that the tumor was generated by hammering or something, and whitish contrast. So in the second, after injection of the nanoparticle, after 10 minutes, that become dark. That's nanoparticle was effective, and that remains for nearly 2 hours, maintaining the same contrast, darkening contrast. That's the tumor area. And after 24 hours, believe that at that point, what we had believed that the nanoparticle moved out of the tumor site, not from the body, but that moved up the tumor site. So the contrast, darkening contrast was gone. So So in conclusion, different size of nanoparticle of dysprosium and dysprosium yttrium fluoride core cell with the shell with the gadolinium have synthesized, and their size were varying from 5, 10, 20 nanometer. Then magnetization were measured for all the nanoparticle that was highest for 20 nanometer particle. Then the surface of nanoparticle was modified with PMO and PEG, and to make them water dispersible. And the relaxivity measurement data of sodium dysprosium fluoride nanoparticles particles at 3 Tesla for that of resophist at 10-fold of at 9.4 Tesla, which suggests that it is a better commercially available contrast agent compared to the better than the commercially available contrast agent. And the preliminary image of the nanoparticle showed some contrast in the tumor after 2 hours of the injection. So this could be the future works, like future work can be for core cell, or even it could be sodium dysprosium fluoride with gadolinium core cell, or we can use the sodium dysprosium, only we can use dysprosium fluoride shell of gadolinium. Reason is that when you add sodium in this matrix, the one dilute the magnetic moment of the overall nanoparticle. So removing from sodium from this matrix will increase the magnetic moment, and that will give better contrast. But since sodium is a polarizable, that also help in growing of the particle. So as my observation said, that dysprosium fluoride is hard to grow more than 5-6 nanometer. If some method can generate better particle close to 20 nanometer, that will lead certainly very good contrast. So maybe the new design needed to synthesize the dysprosium fluoride nanoparticle. At the moment in literature, only 5 nanometer particle have been grown, and if somebody is going by annealing or something, that may not be able to functionalize that. So and facilitate the target specific deliver nanoparticle at the tumor site improvement by surface coating. So the next point was to facilitate the targeted specific delivery by antibodies to making them more tumor site after surface modification. So, and my sincere gratitude to all co-worker where I have worked. Thanks. Thank you for nice hearing. Yeah, thanks for the time giving for presentation.
Hello. Thank
Yes.
you, Dr. Anurag. Very nice presentation. I have one query. Have you tested any toxicity of these nanoparticles?
Yes, ma'am. Behavior
because
toxicity.
They are very small.
Yes, we have tested the behavior toxicity, and that was if you can see this was the data for that, and mostly 27 slide I removed that. So this was but just the behavior test, like they were happy in making the nest, playing around. This was 7-day monitoring. That's we never saw the blood composition, how that varies, how it's toxic. No facility was there.
Okay. Because they are so small in size, no? Then they are inhalable.
Yes.
Professor Gautam?
Question?
Yeah, I have a question, ma'am.
Okay.
Professor Gautam, it's a good talk. Hello?
Please proceed.
Yes.
Yeah, I have just one query, because you are talking about the diffusion of your nanoparticle over the blood-brain barrier. Am I right?
Yes.
Yeah.
Yes, they will not. Nanoparticle cannot diffuse the BBB.
Yeah,
no nanoparticle can diffuse. Yes.
Blood-brain barrier.
Go ahead.
Yes. Yeah. No nanoparticles can diffuse blood-brain barrier.
Yes.
Okay, then I thought that your particle is getting accumulated over the brain by diffusing the blood-brain barrier. If it is not so, then
no.
Thank you. Thanks.
I want to know, in that sample preparation, how did you maintain the vacuum in 3 neck bottom in slide 7?
Slide 7. Okay.
Yes. In the first step, you maintain vacuum, no? How and why?
One moment.
Please go to slide 7.
Just one moment.
Slide
yes,
7. So,
Yes.
okay.
Yes, sir. Yes, sir.
Forget
Yes.
about that. And then what is
Yes.
what about the core cell? How do you know that it is core cell?
We did surface analysis also with the synchrotron facility, and the contrast you can see the core cell, because this is the method that developed in our lab. Was first you have to grow the dysprosium side. You are not this was the injected technique. So first you make the hexagonal shape of the nanoparticle in the system. When they will be approximately 15 nanometer, then we inject gadolinium. Initially, gadolinium will not be in the mixture. This is Jack's paper, I think came from our laboratory. That is for the sacrificial nanoparticle to generate the core cell structure. So once you inject the smaller nanoparticle, that is 5 nanometer over the 18 nanometer particle, mostly they go on as a cell. So once we design the dysprosium nanoparticle of approximately to 18 nanometer, we injected 5 nanometer gadolinium in that flask by syringe method, hot injection, and that dissolved on the surface and by Oswald ripening. So that's the cell structure was generated.
No, just presumption will not do. No, you have to do something like IITM or something because like that. No?
I think that yes, TEM contrast you can see. You can see the TEM contrast, but I think that paper but also tells the synchrotron facility. Because that was generated, sir. Actually, the process was established in our lab.
Injection is different. That is your assumption. But what is that actually you have to characterize?
Yes, that result I have not shown you. Yes.
Another thing is that how can you know that water dispersibility?
Water dispersibility through the zeta potential, which we measure the size of nanoparticles. So we make them water dispersible transfer. Oh, I have not showed here, so not remember exactly.
Hey, how much is that number? Without that, how can you say that?
You can check the JPCC paper. I have paper. I have given.
No, if you don't know that number, why did you present your data?
I think we can discuss it offline. As the data is already published. So.
Okay, fine, no problem.
Dr. Puja, you can kindly proceed.
Thank you, Dr. Anurag, for a nice presentation. Now I invite Dr. Narendra from IITM Pune, and he will deliver talk on climate change research for society, science and society. Dr. Narendra, please.
Thank you, Indra. Am I audible?
Yes.
Slide is seen.
Yes.
Good afternoon, everyone. First of all, I would like to thank the organizers for giving me this opportunity. And next is, for the last 3 days, very high-end research papers have been presenting here. So I am sure, and I wish the KKT be a center for excellence in the days to come. So today, I'm going to present a paper, Why is Asia-India monsoon weaker on the face of global warming and climate change research for science and society. So before coming to the answer to this question, first let me clear about this, that we have to know the recent tendency of the rainfall variability over Indian context, which is the source of water availability to the society. And next, we have to come to the why is the Indian summer monsoon rainfall weaker on the face of global warming. So my talk will be divided in 2 parts. First part will be GIS-based Indian rainfall variability, which is the source of water availability to the society. And in this first part, I will show a very simple observed fact, just like a monitoring of the recent happenings that I will show here. And I am sure all the participants, we are having the presentation of very high-end research of various disciplines, because this e-conference is a multidiscipline, very high-end research presentations are going on. So now coming to the first part of this presentation, just I'm showing here 200 years of instrumental All India monsoon rainfall, and this data is from 1813 to 2012. In fact, this data has been updated till 2019, but this 200 years instrumental All India monsoon rainfall is very famous. So I'm showing here, and this data set we have collected from the manually I myself, IMD. And here we can see very clearly that the mean annual rainfall, summer monsoon rainfall is almost 900 millimeters for every year. This is the mean for 100 years from 1901 to 2000. But let us see again year-wise rainfall from 18 to 2012. See, every year it is assured that almost 800 to 850 millimeters rainfall is assured for every year. However, in India, for every year, many suicides by the farmers are going on. So my question is whether this All India summer monsoon rainfall or All India annual rainfall series is very much useful to our farmers, or is it very much useful to the society? So answer is no. Why? Because see, rainfall is a very highly variable parameters. So even in a city or in a very, say, campus of a university or research institute, half of the campus may get rain and other half may not get rain. So rainfall is itself a very highly variable atmospheric parameter. So that's why this All India summer monsoon rainfall series or All India annual rainfall series a limited value to the society. So for that, understanding the variability, distribution, and the fluctuation characteristics of spatio-temporal variation of moisture regions, dry wet zones on annual, seasonal, and monthly scale across India, as well as its impact on societal issues are explored. Now, say when this annual or monsoon rainfall series for whole India, it is having a limited, value to the society. So that's why we have created, say, climatic moisture regions. 6 climatic moisture regions have been classified based on the rainfall. So in fact, these climatic moisture regions have been classified based on temperature and moisture availability by front weight. But here I'm preparing as I'm classifying it based on rainfall values. And this has been, say, internationally, well known, and we have published this paper way back in 2002, 2003, we have published in international journal. So this criteria based on rainfall is also acceptable by the researchers of the atmospheric science. Now, based on these threshold values, say, first is we have classified into 6 zones, climatic zones, that means climatic moisture regions. So area, this is for Indian context. So the area where the rainfall is less than 560 millimeter on annual scale, and those areas are treated as a arid zone. If the rainfall is in between 560 to 1040 millimeter, then semi-arid, and if the rainfall is 2,450 millimeters, then it is known as a per unit. Based on this threshold value, the thematic maps under different annual moisture conditions as well as the seasonal and monthly dry wet conditions have been prepared using GIS software. And now let us see. This is the moisture region based on this criteria, these 6 criteria. Based on these 6 criteria, this is a moisture region of India. Say arid region, this is for 100 years mean, 1901 to 2000. Arid zone, this is Rajasthan and some parts of Gujarat, 11.75% of the geographical area. I'm talking about the geographical area of the country. This is under arid zone, semi-arid zone, dry, humid and all. Now, this is the normal picture for 100 years, but during second 5-year financial plan of our country during the starting of 1960, by somewhere 1960 or 61, Professor Mahalonovis, he was giving a presentation and he was telling that by say 1990 to 2000, whole India will become a desert. It was his presentation. Why? Because that time he was taking the rainfall data, he was taking the temperature data, and it was showing that the arid regions have been expanding. It is spreading. But in fact, whether is it true or no, let us see. I'm going to show a very small PowerPoint animation. Now, first I will tell you, please concentrate on a particular say color. So suppose arid zone, this color, orange color. This how it varies every year. So I'm showing some animation now. We can concentrate here. See, sometimes this arid zone expands and contracts. So it means that the hypothesis what we are having is, in Indian context, based on our rainfall, the desertification or desert area, it is not expanding. It is contraction and expansion is going on. So for this now, for each and every year, this area under different themes, for every year it has been calculated. And based on that, every year, so 1871 onwards, I am plotting here, till 2006 I am plotting here. And here all the zones except this arid zone, unit and per unit, it is declining. Recent, I'm talking about the recent trends. And other semi-arid, dry semi-arid and moist subunits are increasing. Now, let us see the land use, land cover change over Rajasthan. This is the data is available only from 1860 to 1980, because this was actual survey. This data is based on actual survey, and the survey, the project was taken up by Mrs. Edwin Hines. She is an American, and she took up a project in India, and this land use, land cover for whole India. She was conducting the project there because she was not there all over the project time. It was started somewhere 1850s, and data is available from 1860, and the project was continuing till 1980. And based on that data, I'm showing the say for Rajasthan in particular. So we are having all the data set for all the states, and here also we can see the desert. So mainly our desert is in the Rajasthan area. So I'm showing the data for Rajasthan, actual data. This is actual survey data, and desert is also decreasing. It's not increasing. And this built up area and arable area is also increasing. This is for comparison point of view with our understanding through rainfall. And these are again the threshold values for all the seasons and months. I will not go into this because I wish to give only some glimpse. This is a winter season. This is again mean picture for 100 years, and dry. This is very dry. This is dry, wet, and this very wet. They are based on this color. And this is for summer. This is for summer monsoon. Again, summer monsoon, the same way. This color is also, this color is, I don't know which color is showing in the PPT, but here it is orange color is very dry, yellow is dry, and green is wet, and blue is the very wet. Now let us see again, I will show you an animation, how it varies, how it changes year to year variability. So in some part, this is actual data. I'm not doing anything. It is actual data available with us. So it means that the contraction and expansion of our rainfall is happening for the last so many years, so many decades. And now Why do I see this? This is then based on this area and the different categories. I have myself developed the index called the Rainfall Spatial Distribution Index, and higher the value of this index, it indicates that enhanced rainfall over the large area, because this index value is limitless, and this is the index, and if the index value is higher, then the larger area of the country gets good rain, means wetness, and if the index value is small, then it indicates that the larger area of the country gets drier. So this analysis of this is only for our understanding on the recent trends of rainfall variability. Now, this extensive examination of the spatio-temporal rainfall variability over India modulates for a detailed analysis of the smaller scale rainfall variability. Why? Because if we take a whole India, say mean rainfall for all India summer monsoon Hoya annual, then it has a limited values. That's why we need to study for a smaller scale rainfall variability. Now, so then I started to study for these physiographic divisions. Why do we need to study these physiographic divisions? Because wetlands are the first target of human interference and also the most threatened of all the natural resources, though it supports directly or indirectly millions of people through various angles. Around 50% of the India's wetland is estimated to have disappeared worldwide over the last 100 years through conversion to industrial, agriculture, and residential developments. Keeping in view societal importance and recent emphasis on the development and management of wetlands in the country, the studies of physiographic division, say rainfall fluctuations are asked for. Now, this is the physiographic division of India, and we have 15 major physiographic divisions and 49 minor subdivisions. This has been classified based on the formation of the land, climatic conditions, drainage patterns, soil type, flora, fauna, and so on. And this map have been taken from the NATMO, National Atlas and Thematic Mapping Organization, Kolkata. And that map, we have taken the map, and this map has been digitized by me. And I have digitized it. And after digitize this map, now I have calculated all the physical features of all the divisions and subdivisions, like geographical area, mean annual rainfall, potential evapotranspiration, and all, to see the recent tendencies, recent trends of the rainfall features. Now, coming to the recent tendency in rainfall fluctuation over the country, I'm very simply I want to show it in terms of geographical area. How much area is the decreasing or not? But I don't want to focus in this part. But this is to say when we want to see why this Indian summer monsoon rainfall is weaker during the global warming period, then for that we need to understand the recent tendency over India for this rainfall. So that's why I show very simple pictures I will show here based on this earlier analysis. This analysis I have done it, and based on that analysis I am going to show the results only. This is the recent tendency in rainfall fluctuations over the country on annual scale. On annual scale, the red color means decreasing trend on rainfall. Blue is showing the increasing trend in the rainfall. Green shows no trend. Here, please keep in mind that I am not showing the rainfall data. Here I am showing only the geographical area of the country. Area under this red are having the decreasing tendency of rainfall, not the rainfall amount. And the decreasing, how much decrease is, almost minus 4.11% is decreased if we consider it from 1960, 64 to 1960s are the, say, rainfall shift is there. During 1960s, our Indian rainfall is shifting. Before 1960s, rainfall patterns are different, and from 1960s onwards or early 70s, rainfall pattern is different. So if I take the 1964 as a year of bifurcation, then the rainfall is decreasing by minus 4.11. And if we consider for all the seasons, we can see that except this summer, and post-monsoon. Everywhere the red areas are more. Here in winter it is 47% decreasing, 37% increasing, and here when it comes to the summer monsoon, it is 75% is decreasing. 75% of our geographical area of our country gets decreasing rainfall, where 78% of summer monsoon rainfall is contributing to the annual rainfall. Means highest rainfall is contribution is by summer monsoon. It's almost 78%, and in that area, in that season, the geographical area of 75% of our country gets decreasing rainfall. And if you put months as well, we can see except June, all are decreasing tendency. So now I am showing the very simple pie chart. For annual case, we can see very clearly here. I hope this annual scale, 68% is decreasing. In terms of geographical area, again, I'm clarifying the 68% of the geographical area of the country gets decreasing rainfall, whereas 22% are increasing. On annual scale, winter 47% decreasing, monsoon 75% decreasing, post-monsoon 38% decreasing, where summer is 73% increasing. Here we can remember that summer monsoon contributes only around 10% to the annual rainfall, total annual rainfall of India. Only around 10%, 9 point something. Only 10% of this summer monsoon contributes to the annual rainfall. So though it is 73% of the geographical area is increasing, it is this contribution in terms of the rainfall amount. Yes. Now this it is discussed here itself. I don't need to go here. Now coming to the second part, which is a main topic of today's discussion. Why is the Indian summer monsoon rainfall weaker on the face of global warming? A climate change research for science and society. Here, I want to focus in different 2-3 things, 2-3 definitions. Here I'm writing here Indian summer monsoon rainfall. However, monsoon and rainfall are two different entities. They are different. Monsoon is a system, rainfall is a byproduct of it. Monsoon is a system and rainfall is a byproduct of it. So, but we used to say that Indian summer monsoon rainfall, and we generally, many people, particularly our students, they think that monsoon is a rainfall, but exactly it is not the same. It is different. And again, global warming. Yes, warming is going on, no doubt, but this is a phase of our changes because it has been taking place from time to immemorial. And now the question is, why is the Indian summer monsoon rainfall weaker on the face of global warming? Yes, in Indian context, our monsoon rainfall is declining. But this is again contradicts to our physical understanding, because if it is warming, then we can expect more rains, because once it is warming, more evaporation, more precipitable water in the atmosphere, so it is expected to get good rains. But on contrary, we are getting the lesser rainfall in Indian context. Then why? This is a question we are asking. Now let us try to answer this question as a climate change research for science and society. Now, the climate change may be global or cooling. Yes, this is the reason why we need to study is humans and human society have evolved in the backdrop of climate change. Then I want to come to the climate change, what a climate change is. Because the climate change, the concept, what we see, and the concept in actually what a climate change is little different. It is we have to be very cautious to give any comment on climate change. Nowadays, every person talks about the climate change, every person. So in fact, the climate change is very simple. It is a significant gradual change in climatic conditions during a specified period compared to a preceding period capable of causing considerable impact on the settled ecosystem. It means that, see, it should be a gradual change. Abrupt change should not be there. It should be gradual change. And in the long-term period, in a period of around 15 to 20 years, if the settled ecosystem is changed, then we can say that the area is facing the climate change. Otherwise, it is not a climate change. If it rains very heavily this morning, then next morning we can see on the newspaper that due to climate change, it's happening. So it should not be like that. The duration of the period varies drastically from one scientific discipline to another. First of all, it should be able to change a considerable ecosystem, settled ecosystem, in a specified period of minimum 15 to 20 years, as far as the say hydrometeorological conditions are concerned for our society. So the types of climatic change are, say, as I said, different scientific disciplines have different definitions of the climate change. So geological time scale, the climate change is 10 to the power 6, and the spatial extent is global. Anthropological climate change, geophysical climate change, and now we are talking about the historical climate change of around 10 years, so 100 years, and climatological is about 30 years, and hydrometeorological climate change is about 10 years. We are mainly sorry, concentrating on these three climate change. Why we are concentrating only in this? Today I'm going to talk about the climate change research for science and society. That's why it's a direct impact to our society, because research should be a problem-solving research. So I'm going to focus on this historical, climatological, and hydrometeorological climate change. Now, the complexity of climate change, no doubt, it has a multidisciplinary, and it has a very complex research on this. So, for example, we can see only 1 year. Even in a year, some seasons are warmer and some seasons are cooler. So summer season, winter season, summer season, say autumn and all. And even in a decade also, few years may be warmer and few years are cooler. So if we consider a century, few decades will be warmer and cooler. So understanding the temporary resolution of climate variability is a complex issue. And the study on the climate change, and the climate change research is a multidisciplinary, and it has a vast subject to be discussed for, say extraterrestrial factors like solar radiation, arcs and geometry, interstellar dust, volcanic eruptions, atmospheric chemistry. In short, ocean, atmosphere, and land factors play a major role in this climate change research, climate change studies and research. As in short, it's a very multidisciplinary and complex subject. So now coming to the possible cause of weakening of the Asia, India summer monsoon circulation and all India rainfall on the face of global warming, mainly we have to address these four issues. Four issues are to be addressed. One is monsoon definition, a new perspective, and delineation of monsoon regime, and monitoring the real-time global weather. And number three is a 3D structure of the atmosphere. Now, the monsoon is, in fact, everybody knows monsoon is a system that carries the rainfall over our Indian landmass or any other part of the globe. So every part of the globe, they have a different monsoon system. Monsoon system, when the monsoon system is there, even though there may or may not, rain is a byproduct of it. Monsoon system brings with warm and moist air in the atmosphere. It means that precipitable water is available in the atmosphere, but for the rainfall, it needs some local conditions are required. So monsoon system and the monsoon are different. So earlier monsoon definition is seasonal reversion of the wind. So from the southern hemisphere, it crosses northern hemisphere, and direction is 180 degree, it is reversal. And when it comes from the southern hemisphere to the northern hemisphere, means oceanic region to the landmass, it comes with warm, moist air. So the monsoon definition was earlier seasonal reversion of the winds, along with say some warm and moist air in the atmosphere to give the good rain. Now, this concept, during 2006, I've say analyzed it, all the meteorological parameters, vertical, so surface to around 12 kilometers, and I have given a new perspective of the monsoon. This was given by me. It was during 2006. So monsoon is a seasonal spreading of equatorial type of hydrometeorological conditions on either sides of the equator, following north-south annual oscillation of the sun. So instead of giving the seasonal reversion of the wind, so I'm going to give the definition, new perspective of the monsoon. Definition is, monsoon is seasonal spreading of the equatorial type of hydrometeorological conditions. So the conditions over the equator, maybe the high tropospheric temperature, this is tropospheric temperature, this is temperature from surface to around 250 HPA, means up to 12.5 kilometers, low mean sea level pressure, lower tropospheric convergence, upper tropospheric divergence, warm and moist winds, cloud and rains. Whatever the meteorological conditions available in the over the equator, if it is spreading towards maybe in the northern hemisphere or maybe in the southern hemisphere, that the area where the equatorial type of hydrometeorological conditions are there, and that area is known as the monsoon region. This was given by me. Why do we need these meteorological conditions over the equator? So equator is the region where the variation is least for all the parameters. So suppose the temperature for this tropospheric temperature, I'm taking the column average, means surface to around 12.5 kilometers, the average temperature of this vertical average, and this temperature is, say, I'm showing the monthly, January, February to December 1951, 52 January to December, like that. I'm showing all the months. However, the variation, let us see minimum minus 8.2, maximum 5.2. Minus means only 3 degrees centigrade. Where if we see in any part of the country, say for example Imphal, minimum temperature we sometimes recorded as 0.5 or even 0 degree in the hilly district, and in the summer season it goes up to 36, 37. Means the variation is more than 30 degrees. But in equator, throughout the year, variability is only 3 degrees centigrade. So that's why we have taken this equatorial value is a reference value. So based on that reference value, say I'm trying to delineate the monsoon region. See, if any area, it means that tropospheric temperature is greater than the tropospheric temperature over that means plus 5 degrees. And the mean sea level pressure is less than the mean sea level pressure over equatorial belt. And precipitable water is greater than precipitable water available over the equatorial belt. ERP means equatorial value I'm putting here. And the more conditions are needed for rainfall. For rainfall, we need the mesoscale conditions. So it means that monsoon regime means monsoon system. When the monsoon system comes, then monsoon regime will be there. So my hypothesis, my argument is like this: any day, any time, wherever, anywhere in the globe, we can see the real time. Nowadays, we can see the real time data through satellites. That what we are doing now. So if the temperature, tropospheric temperature is higher than the equatorial belt, mean sea level pressure is less than equatorial mean sea level pressure, precipitable water is higher than that of the equatorial belt, then that area is a monsoon regime anywhere in the world. This is my argument. This is my hypothesis. Initially, when I proposed this hypothesis, many objections were there. Even when I present this in front of say, even our director and senior officials from scientists from the ministry and ISROs and all, they all were objecting. But afterwards, when I explained it, all are agreed that we can see if real time, if you see through website, if you can see the real time data, if the tropospheric temperature better than the equatorial value and less than the mean sea level pressure and higher of this precipitable water, then that area is monsoon regime. Because this monsoon regime, we need to understand, we need to know, unless this monsoon regime is there, there will not be no rain. Because rainfall is a byproduct of the monsoon system. So that's why, if we want to discuss about the rainfall, we need to know where exactly the monsoon regime is. That's why I have hypothesized this monsoon regime, and now it has been accepted by all internationally. And this is a monsoon delineation. So anytime, anywhere in the globe, if these conditions are satisfied, that area is monsoon regime. But for rainfall, we are not sure. It requires mesoscale conditions. Now, some monitoring part we will see some sea surface temperature, tropospheric temperature, geopotential height, mean sea level pressure, winds starting from surface to up to 12.5 kilometers. So a real-time data set getting from the satellites. I analyzed it, and here again I want to show, I want to give some glimpse of the tendencies happening nowadays. So once we know that, we come to know why this Indian summer monsoon is decreasing during the global warming, which is contradictory to our physical understanding. Now let us see. This is the climatology for 51 years. Tropospheric temperature, again 100 HPA to 250 HPA means surface to around 250 kilometers and 12.5 kilometers. Now, here this is the climatology, the global. So India is here. So climatologically, if we take the average of year column up to 12.5 kilometers, this area Tibet, Sena means Tibet, Himalaya, Karakoram, Hindu Kush and all, extended up to Iran, Iraq. This area is warmer than that of the other part of the globe. It is warming. This is a climatology for 51 years. Please remember this. This part is warming. Over Tibet, thicky hills, we can say the thicky hills. Tibet, Himalaya, Hindu Kush, Karakoram, and all. Now this is now I am taking the second half minus first half. This is the gridded data. Each and every grid I am taking the difference from the second half to the second half minus first half. Here I divided the data into two parts up to 1978 and 79 onwards, because 1979 is a climate shift, globally accepted climate shift. Global warming starts by the end of 1979. So 1978, so 1979 is the climate shift. So when I take the difference between the first half and the second half, so we can see here, India is here. Now this area location little bit shifts, but this area becomes negative. Negative means first half is higher. It means the temperature is decreasing. Tropospheric temperature is decreasing here. And you please remember, south of this minus around 35 to 40 degree is increasing. Okay, and we see the good monsoon year, composite of the good monsoon years, intense monsoon years. So I've taken the 10 good monsoon years, I club together, and I see the departure. Departure means this area is again positive. It it is increasing. Temperature is increasing here, and this part is decreasing. And you see for the big monsoon years, for 10 years again this is decreasing, which is similar to the present day situation. Okay, and next is again if we see for the surface temperature, again this is decreasing. Again this is a lower tropospheric temperature, decreasing. Upper tropospheric temperature is decreasing, and here again mean sea level pressure, again this part is increasing. Pressure is increasing means our convergence is not investable. And it should be low pressure. For the intense rainfall, good rainfall, it is negative, means pressure is low, pressure is forming here. Weak monsoon year, higher pressure is is seen over here, and this is somewhat equivalent to our present day scenario. Then similarly for sea surface temperature also, this north Pacific is here, and this area in say equatorial Pacific Ocean here. This equatorial Pacific Ocean towards the American coast is increasing here. The situation is increasing. Increasing means it's a El Nino year. And when there is monsoon year, it should be negative here. But nowadays it is increasing. So weak monsoon year, again the same thing, it is increasing. So it means that the atmosphere itself is conditions are happening. So based on all the meteorological parameters on isobaric level, such to 12 kilometers, I analyze all the parameters using the satellite data. Then I created this very complex diagram. I prepared it based on the analysis what I have done it. There is how much time can I take? Another 5 minutes. So now this is the diagram. Okay, the center of action involved during the active Asia Pacific monsoon. Now this is the 3D structure of the atmosphere, and this 3D structure of the atmosphere have been say prepared using the data set in the sense layer wise data set, starting from the surface kilometers. All the satellite data set and in situ means observed data sets. I used it, and I see each and every level, temperature, pressure, winds, precipitable water, and all everything, and geopotential height, temperature, but in particular temperature, I have analyzed it very extensively. I analyzed it because our monsoon system is say thermally driven phenomena, means it is mainly due to the thermal contrast between land and ocean, thermal contrast between the southern hemisphere to northern hemisphere. Based on this thermal contrast, our atmosphere is awesome. That's why I extensively studied the temperatures in these layers, and after that I created these centers of action involved during the active Asia Pacific monsoon. Now this is an area that is where I mainly focus on the diagrams based on the satellite data, where this is the Tibet, Himalaya, Hindus, and Karakoram belt. This is known as in our research field. We talk about the Tibetan height. This is around 4.5 kilometers higher than the mean sea level, and here this part, including our extending up to our Gujarat, Rajasthan, when this area the temperature is higher. When the temperature is higher, what will happen? The pressure is low. So then when the temperature is higher, the pressure is low, then it will start rising towards vertically. Vertically it rises. And again, the monsoon system or wind system in our atmosphere, globally, it is a fluid system. it will never stop as just like as a positive boundary. It is not like that. It continues. So once it rises vertically, it has to sink somewhere. Where to sink? That sinking locations, I term it as the center of action. The sinking locations, because it is the higher elevation of 4.5 kilometers above the mean sea level, and this is the area where the warming takes place. So in this warming area, surrounding winds will converge here and it will rise up, and after shooting up vertically, it has to sink somewhere and it has to come back again in the lower level. Just like a conveyor belt in our airport. Just like a conveyor belt in our airport. Our luggages will go here and there and come back again. Same thing. So from here, the intensity of the warming, intensity of the rising, it changes from time to time. So this time it will sink at 8 different locations. 2 in the poles and 3 in the southern means pole, northern pole, North Pole, South Pole, Azores High, Pacific High, and this is South Indian Ocean, Australian High, South Pacific High, and South Atlantic High. Like that, once it starts rising, then it has to sink, and it depends where does it sink. It depends our intensity of monsoon to our Indian context. So now our question is, if we throw a ball very high intensity, it will go in a long distance. If we throw it very slowly, it will fall down very close by. That is, same thing is happening in our context, or in this atmospheric science also. It is a fluid system. So now this intensity of the sinking locations and the changes of the sinking locations plays a major role in declining the Indian monsoon rainfall. In fact, to be very frank, all India case, on all India scale, Indian summer monsoon rainfall or annual rainfall is decreasing, of course, but it is not significant. Why? Because this rainfall regime, our rainfall regime is shifting north-westward. Earlier, it was having the rainfall over the Indo-Gangetic belt, where it is very fertile, and from where our maximum food grains were produced. But this belt has been shifted towards Rajasthan, Gujarat. So we have seen that every alternate 4 or 5 years, heavy floods, extreme weather conditions are happening in the Gujarat-Rajasthan belt. This shows that our rainfall belt is shifting towards northwestward. This is due to the position of the sinking of this sinking positions, changes in the sinking position and the intensity of the sinking position. Now, coming to the end of this talk, because I will not take too much time. So everywhere the temperature is warming. No doubt, temperature is warming for all the climatic zones, and we see if we subtract from this equatorial belt, it is decreasing. So now the increase, how much increase is there? So if we see for the global, this is the findings based on our analysis. Global tropospheric temperature is increased by 0.59 degrees centigrade. If we consider only for the southern hemisphere, for the tropospheric temperature I'm talking about, it is 0.87. Northern hemisphere is 0.32. Gradient is minus 0.55, means northern hemisphere minus southern hemisphere. Extratropics, 0.95 degrees. Arctic to Antarctic, minus 1.11. As I said earlier, the monsoon system is a thermally driven system, mainly due to the contrast between the temperature contrast between the land and ocean, temperature contrast between the northern hemisphere and southern hemisphere, temperature contrast between the Arctic and Antarctic, because mainly our southern hemisphere is oceanic region and northern hemisphere is landmass. So monsoon system, when the wind blows from the southern hemisphere to the northern hemisphere, it has to come with warm and the moist air, right? So in that way, this thermal contrast, wind blows based on the thermal contrast only. So this thermal contrast plays a role, and this thermal contrast, when we talk about the thermal contrast, the temperature, it is no doubt it is warming throughout the globe, but the warming over the southern hemisphere is at a faster rate than that of the northern hemisphere. That is one of the most important thing we have to consider. And another part is following north-south seasonal migration of the sun, the large area of the globe where the tropospheric temperature and circulation exceeds than that of the equator are defined as a monsoon region. What I explained, this was, I have hypothesized this. There is a decrease in the tropospheric temperature from South Asia's Cordillera to the 8 deep highs. 8 deep highs means when it is rises, it dips over 8 deep highs. This is center of action, what I discussed. The 8 locations where the sinking motion takes place. These are the 8 deep highs, which cause the decline in the intensity of the Asia-India summer monsoon circulation. Here also I talk about only the Asia-India summer monsoon circulation, not the rainfall. And the weaker monsoon is responsible for the decrease in the surface layer, means 1,000 to 700 HPA zonal wind studies, and the increase in the sea surface temperature over the Nino region. This is mainly these are the factors where we are getting the declining in the intensity of the Asia monsoon, summer monsoon circulations. And once the circulation is weak, it is expected to get less rainfall. And for better understanding of the rainfall, we need the mesoscale conditions. This is the main findings, and this is my concluding remark for the society. The survival of the eco, no doubt, we know that the Indian context, the rainfall is decreasing, but the survival for the ecosystem under changing atmospheric as well as the global climatic scenario, changing global climatic scenario, reasonably well suggest better management practices of the water bodies adopted by the people to combat or resolve the twin problems of flood and droughts. Because we have to adapt. We have to adapt. We cannot run away from our universe, from our mother earth. We cannot run away. That global climatic changes and ecosystem is changing, earth is warming also. So we cannot run away from our society, from our mother earth. We have to adapt. Reasonably well suggest better management practices of the water bodies. Thank you. This is my, this will conclude my talk. Thank you very much.
Thank you, Dr. Naren. Now, forum is open for question answer. Any question? So I have one question. Do you have any prediction that prediction about the reduction in rainfall and the agriculture system? Any correlation you have developed? Huh? So
Yeah,
we can so that.
But I'm mainly dealing with the atmosphere, atmospheric 3D structure. So we have another group. They are doing the agriculture meteorology. But yes, we do have. Since predictions means for prediction, what we know is the past conditions and present conditions and what will be the so that we have. We do.
Okay. Thank you. Thank you. Any other question?
Yes, ma'am. I do have one question. Thank you.
Please.
Yeah, thank you, Tamanaren, for the very useful information shared. I have one question. In your last slide, in the last but one, you have shared about the increase. I mean, the gradient of the increase in temperature or the decrease. It was like starting from around 0.5 to almost 1. What is it? What is the gradient, or what is the increase, decrease over a particular time, or what exactly is that? Could you please explain on that?
That is the area what I mentioned. Your 1979 is the climate shift our global warming starts by the end of 1978. So the whole range, entire period have been divided into two parts up to 1978 and 1979 onwards.
So
that is
yeah, does it mean that like the increase of around 0.5 or 0.8 degrees Celsius is from that 1970s until now, or is it per annum? That's my question.
No, not per annum, not per annum. It is the only period wise. These two periods, not per annum. Well, see, I've taken the average up to 1949 to 78 average, and 79 to 2017 average, and we take we take the difference.
Oh.
Because we have the means now. So mean for the first half, mean of the second half, and that is change in the mean.
So when we,
of course, we do have year-wise calculations are there, but for this simple presentation, so whether it is increasing or decreasing, to see that in the just a simple example. So I have shown the difference between two means, first half mean and second half mean, whether it is increasing or decreasing.
So okay, yeah. So in short, when we say global warming, do we have any over how much temperature is increased per annum? Anything like that for a particular specific area or location?
No, as of now, per annum, we consider for per annum, it comes 0.001. Okay. So there is not just year-wise calculations are not done, because up to 2 decimals, it is 0. Because the condition in the global warming, if it takes for the last 100 years, how much is decreasing, that way we are calculating. So in that, almost 1.1, 0.7, sorry, 0.8, 0.9 in some areas. So per annum, we are not, because up to 2 decimals, it becomes 0.
Okay. Well, thank you.
Okay. So nice talk. I also have one question. Is there any study on the correlation between the water currents of the ocean and this atmospheric pressure change? Is there any correlation between anybody studied or?
Yes, thank you very much. What by the atmospheric pressure?
Yeah. So it's like in ocean currents are there, not from cold to hot like that, and how the pattern is changing with the
ocean,
and how that is correlated with the change in atmospheric pressure. Something like that is there or not?
You
are considering only the atmospheric.
Don't have the atmosphere.
This is the only thing you have. Many people have been discussing, not related to the water currents of the ocean and how the ocean temperature is changing, because you told that.
Dr. Gunathur, can you? I think there is an echo.
More hotter than the northern side. Which one? Niraj, Dr. Niraj,
I think there is an echo.
Let me tell you very simply that in my first initial slides, I said the measure atmospheric system or rainfall or monsoon. There's a large influence of land, ocean, and atmosphere. And another statement what I made was this thermal contrast between land and ocean and thermal contrast between the two hemispheres plays a major role in this monsoon system. Yes, we do have a very much large correlation between the ocean currents, mixing ratio, particularly the mixing ratio of the ocean. It plays a major role, and we used to have the oceanic dipoles. We have the oceanic dipoles, and the particular oceans, say maybe in the southern Indian Ocean or Pacific Ocean, and even the oceanic currents, oceanic temperature, what we are talking about, the El Nino and La Nina is based on the temperature of the sea surface temperature over the say American coast. passive ocean.
The ocean temperature also will decide how much is the evaporation rate and things like that, no?
Yeah, correlation, I will see, let me tell you, all the atmospheric parameters is when we talk about the first thing, let me clear you something. See, in the atmosphere, we don't have the pressures. Pressure we count is a geopotential height. In terms of height, we calculate it. Okay, and this geopotential height, see, let me tell you, I didn't show you, I think I skipped it. It was my slide also. The geopotential thickness height and say oceanic temperature, means El Nino. When the monsoon system is very intense, and that time we see the correlation between the El Nino, means the sea surface temperature over the, not the current, I'm not talking about the current now, temperature over the American coast, and over our Tibetan high, about around say 12,500 meters. 12,500 meters is around 600 to 500 HPA level. The pressure level is almost 600 to 500 HPA level, but we are not considering the pressure. We consider it as a geopotential height, and the correlation comes around 0.7. It's very high. But we are not doing
it
the
also
current.
dependent on the water near the source.
Yes.
Pollution of seawater affecting it.
Pollution part, it is not in our study. In fact, we don't discuss it about the, we are not doing the pollution part. We do the temperatures, we do the say oceanic mixing ratio, we do that. And oceanic current, sometimes this oceanic current we do as a wind factors, we consider it, and this oceanic current we calculate it, and we do the correlation. But I will very simple suggestion I will tell to all our researchers, all our scientists, that correlation is not the causative factor. We should not discuss only the correlation. Correlation without physical understanding is of no use. So correlation, we don't use the correlation in our research. Let me clear. Correlation may be if we take a parameter which is not related to something. So we can see the age of all the participants, how old we are. And we take the temperature data or rainfall data, take the correlation. It could be 0.9, and we cannot say that these two are related. So correlations we don't treat as a causative factor, and we need to understand the. We have to go with the physical understanding. This I want to keep in mind with our researchers. We don't go with the correlation. Yes, we can calculate it, but we do calculate the correlation after the physical understanding is met.
Okay.
Okay. Okay, Dr.
Another sir, we don't use the current. In fact, actual current we don't use it, but we use the surface wind, sea surface wind.
Yes, I can imagine. Okay, thank you very much.
I guess the message is clear.
Yeah.
Dr. Puja.
Thank you, Dr. Naren, for interesting presentation. Hello.
Yes, you are audible.
So I'm thankful all the speakers for enrichment in the knowledge. And now I conclude this session. And this session is very diverse, and we learn lots. Like Dr. Barun taught us the modern discovery and various processes involved in the drug discovery. And like Dr. Akshay tell us the mode of action of antibiotic and binding sites, different binding site, how the antibiotic work. And similarly, Dr. Sarjit shows the chitin and chitosan structures, how they can modify and use it pharmaceuticals. And very interesting lecture was from Dr. Shanta on the magic prime numbers. And similarly, Dr. Anurag told us about the nanoparticle for imaging applications. And Dr. Narendra told us the variability in the rainfall and their cause in the due to various causes which shows this rainfall variability. So I thankful to all the speaker. I also thank the organizing committee, especially Dr. Niraj for inviting me. Thank you, Dr. Niraj.
Okay, thank you, Dr. Puja Khare. On behalf of the organizing committee, I would like to thank all the speakers and especially the chairperson, Dr. Puja Khare from CIMAP. Actually, it's a very long session and the field ranges from drugs for humans for the MRI to the world of bacteria and to the world of mathematics and even to the ground reality in the research area of oceans and climate changes. So with this, we come to the end of session 5. Before we proceed on session 6, there were lots of requests that we should give a small break between the sessions. And it is actually a blessing in disguise that yesterday we have 2 oral presentations who are having some technical issues. So in this break, we will present the 2 oral presentations, which will be hardly some 10 minutes. So I will start the oral presentation of the 2 persons. And after that, Professor Rakesh will take over as a chairperson for the technical session number 6. So I am sharing the 2 oral presentation.
I think it's not audible.
Your voice is not audible.
Okay, that concludes the oral presentation. There were some issues with the audio, but in the meantime, the Young Scientist Award Selection Committee member have an access to all the audios. So I would like to request the selection committee members for the Young Scientist Award winner to kindly send the report through the link which is communicated to them before the stipulated time. The award winner will be announced after in the validatory function that will be happen right after this technical session number 6. So moving forward, I would like to request Professor Rakesh M to kindly chair the session technical session number 6. Hello, Professor Rakesh, you can unmute yourself.
Okay, yeah, welcome once again, everyone. And this is the technical sessions and the last technical session of the conference at KHUMANG 2020. And I hope that everyone must be enjoying very interesting and informative presentations from the last technical sessions. And we still have many more here in this, starting from sense, how we have to do the wastewater treatment, and then how we can make these analytical instruments. And then also there are ways how one can, prepare these bioactive components from the natural plants. And then some talks on, the roles of nanoscience and nanotechnology to the society here. So I will not take much time in introducing the speakers, but I will request every speaker to stick with the time because they're running behind the schedule here. So the first speaker which we have for this technical session 6 is Dr. Niraj Luwang from National Chemical Laboratory, Pune. A brief introduction on Dr. Niraj is that he received his PhD in chemistry from Manipur University, and his research focus on development of nanomaterials for applications in developing cost-effective sensors, explosive sensors, instant blood clotting bandages, fabrics, and he has published several research papers in SCID journals, and also has international patents. And Dr. Niraj is presently working as a senior scientist at CSIR-NCL Pune. And he will be presenting his talk on the title Novel Materials for Instant Sensing Applications. I request Dr. Niraj to start his presentation now. Please stick with the time for that. You may have 1 or 2 minutes for question and answer here. Please start.
Okay, okay, thank you, Professor Rakesh. As the saying goes that the show must go on, I would like to thank you for this new role as a speaker. So today I will be speaking on novel materials for instant sensing application. I'm working as a senior scientist in National Chemical Laboratory in this beautiful city of Pune. Here by instant sensing application, what we actually want to mean is that we have lots of appliances surrounding us. Like if we want to know the temperature, we have a temperature sensor. If you want to know the concentration of a solution light, we have a pH meter saying that this is the pH of your liquid sample. Like such kind of sensing application which give instant result, we are working on some novel materials. So here I will be presenting 4 different works. The first one will be for the hazardous material detection, and basically I will be giving emphasis on the arsenic. Second will be for the explosive detection. Third is for the instant hemostasis, that means instant blood clotting. And the last one is a novel material which we coined as a nanogate for the first time. So moving forward, here is the detection of hazardous material in water. So I'm giving emphasis on arsenic detection. And the material that we use is yttrium phosphate doped with europium. Why yttrium phosphate doped with europium? Because actually it's a luminescence material. So even everybody wants things to be simpler. For example, if you want to detect something, and if I say that if there is a change in the color, then the material is present. So to make the things simpler, the material that we have selected, this YPO4 doped with europium, is a luminescence material. By luminescence material, what it means is that it gives out light. So lights are having in different wavelength, but all the lights are not important to us. Or basically what I should say is that the light in the visible range is of a primary concern to us. It means that until and unless we don't see the light, then what is the use of making the compound? So by the visible light, in the technical terms, it is basically in the rainbow range. As we know that this beef grow, violet, indigo, blue, green, orange, yellow, red. So anything, and this is in the range of 400 to 700 nanometer in terms of wavelength scale. So anything in the blue region will give something like 400 to 450 nanometer. Like for example, anything in the green, any material that give green light will give in the approximate amount of 500 nanometer. And similarly for the red, it gives around 600 plus minus nanometer. So the compound, the concerned compound that we are working here, YPO4 doped with europium ion. Europium is a rare earth ion. It's a lanthanide ion, and I think lots of talk has been given yesterday and today also on the rare earth material. So this europium ion gives red luminescence. And here what we are doing is that we are trying to fabricate a new material for a new application. So by the new material, what we mean is that if you make the compound very simple, like if you are looking for a compound C, and if the compound C is formed with few inputs, like if A and B reacts and gives C, then that is the best, easier thing. Now, if you need to maintain the temperature, then it becomes another input. And if you again need to maintain the pressure, and then the variable becomes bigger and bigger. And it is also not good in terms of scale up. And it is also not cost effective when you try to go in terms of a product. So if you want to make it simpler, then one of the simplest method is the co-precipitation method. Here the reactants are put together and we get a compound. So here we get europium doped YPO4 through a simple co-precipitation method, and this compound is used for the sensing of arsenic in the water. How did it sense the arsenic? Because this europium gives luminescence light, and when it comes in contact with arsenic, the luminescence intensity varies. So through the variation of the luminescence intensity, one can detect that the arsenic is present or not, and one can go for the quantification also. So I will explain here. Here, as you can see, YPO4 is an insulator with a very high band gap, and the emission, these are the emission peaks. So here, these are the emission peaks of the compound. They are in the red region. As I am saying that from 600 to 700, they are giving red emission. So when they are in pH 1.5 and 3.5, if you go down here, arsenic is basically observed because they are present in two forms, like arsenic 5 oxidation state and arsenic 3 oxidation state. In 1.5 pH, arsenic is available, they are available in the 5 oxidation state. Now here at 1.5, due to the interaction of the europium ion through the hydrogen bonding, there is a variation and there is an increase of the luminescence intensity. But when they are in the +3 oxidation state, they have this bonding with the europium oxide, and the variation of the luminescence intensity goes down. So this is the pictorial representation. Now, one can also explain through this. This is some technical picture. These are the XPS data. One can see the presence of different elements like yttrium, arsenic is there. This is after the reaction only. Then phosphorus is there. Even oxygen is there. The arsenic is also detected in the final product. And these are the lifetime. By lifetime, basically what we want to explain here is that whenever an energy is given to a compound, the energy will be absorbed by that compound, and there are energy levels like ground state and excited state. All the energy that is absorbed by a compound will be taken up and the electron will go to the higher excited state. After it goes to the higher excited state, it has to give out the energy. Either it falls down, giving out the light, that is called, we have a radiative radiation, or it falls down by giving other forms of energy like heat or something. That we term as a non-radiative energy transition. Now, the time taken for the electron in the excited state to come down to the ground state is basically we term as a lifetime value. So here we are explaining through the lifetime value how long it takes for the electron in the excited state to come down for different PS value for different arsenic in different state like 5 and the 6, 5 and the 3. Now, one need to characterize that as we are saying that we are making YPO4 with europium and it is a nano material, we have to prove that it is in the nano range. So it's a nano rod thing with an average width of 30 nanometer, and the length is somewhat longer. They are formed in the form of a bundle kind of things. And if we take an IITM image, we have a lattice fringes. Lattice fringes are the arrangements of individual atoms in a systematic manner. The distance between the two lines are in the 0.5 nanometer. So these are the technical high-tech instrumentation where you can confirm that your compound is actually what you are making. Okay, now after the work is done, that your compound is able to detect arsenic. Now, what next? Now, the main objective is to develop a simple low-cost arsenic sensor because a general public will not be able to do all this research in the lab. They just want a simple paper or simple, anything. And they want to do in 2 seconds or 1 second that I just want to know that arsenic is present or not. So we as a scientist, we should give them such kind of product to them. So it is our role. So here we already know that there are lots of arsenic contamination, and few of the available kits are from HATS USA, Arsenic Eco Quick 2, and even we also have arsenic kit set up by IITM Kolkata very recently in 2019. And some of the challenges are that if you give an instant sensing, then it is very good. But if you say that you take the water sample, put it in some solvent, and it will give you result in after 30 minutes, or it will give you the result in the next day because it has to go to the lab, because it takes lots of time. So if you give an instant result, then it is very good. So our main emphasis is to give the instant result. So in such situation, the best kit will need to have a quick and instant detection and a quantitative analysis also. How much arsenic is present there? We should be able to give that also. And it has to be cost effective. If it is costing something like 50,000, nobody can afford. So if it is costing something like 100 rupees, then everybody can buy that kit. So, and one of the factor is that most of the available kits, so say few of them, they convert arsenic into arsen gas, which is also very cancerous and very toxic. The good thing of our the sensor that we are making is that it is quick and it is affordable by any person, and it is also based on the and it is also very selective to arsenic ion, and no chemical conversion of arsen gas is required. So so we are doing on 2 parts. The first part is that we are coating it on over a paper strip, and this paper strip will be available to anyone. And by putting 1 drop with a visual change of color, one can confirm that the arsenic is present or not. In the second part, after we confirm that arsenic is present, the next question is that how much arsenic is present. We need to quantify it. So part 2 is the a box type. As you can see in pregnancy strip, we just put 1 drop and we can just say that you are pregnant or not. Something like that will be happening. So in the part 2, we will be making the sensor, and this sensor has also a market in the sense that every gram panchayat can have one is like that. So to do this thing, only CSIR-NCL cannot do it. We need an industry partner. So we develop the product, we develop the material, we give out the proof of concept that this can be done like this. But to take it to the public domain, we need an industry partner. And the industry partner here for this particular work is Scientific Analytical Instrument Private Limited for Navi Bombay. And under the technology development program of DST, a project is running to develop this, and this low cost arsenic sensor, and it is still in the progress. So giving a small highlight of the progress, we are trying to give at the cost of rupees 110 as of now, and we will be trying to reduce it as much as possible. The cheaper it is good for the general public. But for the number of amount of rupees that we incorporated in making the device, we can also compromise on few things. So I think 110 is quite acceptable to the what is other sensors which is available, which is costing above even 1 lakh also. Now going forward, okay, this is another work. There are lots of hazardous material. Arsenic is not the only thing. So this work basically detects selectively iron, chromium, and copper. And here we are working on the similar technology, light luminescence material, sense of luminescence intensity. The only thing different is that the compound is different. Here also europium dope in gadolinium oxide. And this particle gave enhanced luminescence in water due to the host energy that is transferred. And this nano sensor can basically detect iron in excess of chromium and copper also. And we work on some water source in Pune, like Pasan is the place where our NCL is situated. Pasan Water Lake and Khadawasta Dam, these are big 2 dams. There we have tested the content of iron and all these things. And we have also reported that work on this luminescence paper. Okay, the next work is for detection of explosive materials. On the similar way we have detected arsenic, we have also worked on the detection of explosive material. By explosive material, what we mean is like highly explosive aromatic nitro compound. Most of the explosives have nitro compounds in aromatic form. Here we are working on terbium dope lanthanum fluoride. This terbium dope lanthanum fluoride, and we functionalize with para-aminobenzoic acid. The reason why we are functionalizing is that it will give an enhanced luminescence. After functionalization, it gives an 100 times strong emission than the unfunctionalized part. The reason for this functionalization is that, okay, I'm doing dual work of being a force also. Okay, why we are functionalizing here is that if we have a luminescence, for example, let's give some number. If we have a luminescence intensity in terms of number 10 for this lanthanum, terbium dope lanthanum fluoride. Now after functionalization, if it gives 100 times enhancement, like 1000, then we have a window of 990 units where we can work on we can work on the variation of another analyte, which will be the explosive nitro compound here. So first we enhance it, then second in the window we are working. So this work is we also got a US patent over this thing, and it is in force right now. And we have also worked on hello Shanta. Shanta, someone is what is it playing over the screen? Okay, I will continue with the work, with the presentation. Here we are able to detect this TNT, trinitrotoluene, and we have also worked on a series of explosive materials. And luckily, actually in research, luck is also one of the main factors apart from hard work and funding and all these things. So we are able to get a selective detection of TNT up to 50 ppb level. Okay, going forward, these are some technical data. After the functionalization, as I have said that whatever energy we are giving to the compound, it actually absorbs. So this is the absorption data, and this is the excitation data. These are two similar data. See how much energy the compound can be absorbed after the the functionalization of para-aminobenzoic acid. Now, with this high number of excited electron, the electron has to definitely come down. So we can see the emission is also increased here. So these are the energy level diagram, how from a PABA it goes to terbium and how the emission we are observing. Now this is the decrease in the intensity when the compound is interacting with TNT or picric acid. Picric acid is the starting material for all the nitro compounds. So this is the decreasing trend. Okay, this is a simple pictorial presentation. This is the lanthanum fluoride nanoparticle. This is the terbium which give green emission. So this is the compound we have. We functionalize with the para-aminobenzoic acid. This is how we functionalize here. So when some sunlight or any kind of energy falls, it will be absorbed by PABA. It will transfer to lanthanide fluoride, and it will give emission from this. Now, with this system, when the aromatic nitro compound comes into the picture, instead of transferring to the compound, the excited energy would transfer to the analyte. Now the next question is that how it can transfer to the analyte. That's why first there are different ways of research. First is that you plan theoretically and you move forward. The another way is that you do experimentally and you explain later. So in this work, we have worked experimentally and we explain later, and luckily we got that why it is so specific is that the energy level somewhat fits somewhat fits for the TNT and the para-aminobenzoic acid. So there is a specific energy transfer from the para-aminobenzoic acid to TNT. And all the materials we receive from DRDO, ICMRL, this is a high energy material research lab in Pune. And basically when I mention about explosive materials, I'm not accessible to large amount of explosive. Minimum one needs to have a 10 mg of explosive to have a destructible impact. So what we are working is in the millimolar concentration. So there is no harm in working on this explosive detection. Okay, these are the visual color sense, and these are the morphology of the compound that we have prepared. Okay.
Hello, Dr. Niraj.
Yeah.
Yeah, please set the screen. Something is over the screen.
I think
Some
someone
colors
has,
are seen on the screen.
yeah, someone is here. I'm not able to. I will undo.
I think one is you have to remove the erase. I think some while charging the mobile, my daughter did it, but I tried to remove it. I don't know how to do it.
Okay. I have removed it. Okay, moving forward, this third work is for instant hemostasis. This work is done by Dr. Nimrod, one of our research, in our research group. Here, I will cut short. As we all know that whenever we have a cut in our body, the blood flows out. The first thing is that we have to stop the blood. And basically, our human response is such a way that whenever we have a cut in our, in our body, it gives a signal to the brain, and the brain gives a signal back, and where they have to create lots of templates and all this thing, and the blood clotting itself generated by its own way. Whole process took something like 3 to 4 minutes. Above that, one has to physically block them. So here what we are working is that we have synthesized aerosol of chitosan and reduced graphene oxide composite with micropores and high surface area. And this we are doing with our NCL dispensary, because access to blood is also some ethical thing. So we are working with our NCL dispensary, and we could identify a material that has shown the potential to utilize in the emergency hemostasis. So I will move forward. Okay, this is just the pictorial presentation morphology of the compound that we have synthesized here. The basic idea here is that if you consider blood as a liquid kind of thing, from that blood, if you remove the aqueous part, then what will remain is the solid part. So all the solid part keep together, they will club together and they will form a strong bond. So basically that's how we are going to explain here through the blood clotting. The blood clotting has about something like 42 steps from the biological perspective. But what we are explaining here is in the different perspectives. There are lots of products available in the market. Few of them are already banned because they have some heat issues like that. What we are doing here is that when this aerosol interact with blood, water is instantly removed, thereby concentrating the solid material to interact closely and the clotting start. Usually in pre-hospital emergency situation, it usually takes about 2 to 3 minutes, but here it took only 30 seconds. So we have blocked this work for a patent, and we are in the process of developing in the form of bandage and for some clinical trials, and we need to do a lot of things like fast action time, quick responsibility, and portability issue, reusability issues like that. So this work is still going on. Okay, the fourth work is about novel nanogate structure composite. This word nanogate is coined for the first time from our research group. Why nanogate? I will explain here. Many have been reading about the word graphene. Graphene has lots of property. So it comes from graphite. So graphite are layers of sp2 carbon structure, and it was discovered in 1859. Then another by Hammer method it was synthesized in 1957. Then after that, we know when a single layer, say graphene, if this roll, then we know that it is a single wall carbon nanotube, and it is a multi-wall, then it's a multi-wall carbon nanotube. Then we can also see nanohorns and even nanorings. In this work, what we are going to do is that in the single wall nanotube, we are going to incorporate some quantum dots inside through the formation of this nanogates. So this work we have done, we reported for the first time. So we have coined this term as a nanogate. Okay, these are the pictorial presentation. So these are the images that we observe. So if we I will go to the next slide. Okay, here this is the RGO. If we put, for example, in this part we are using cadmium sulfide. So on a plain paper, if you put an object, then the object will push down with some stress and strain, and it will start rolling like this. So it's a natural concept. Now, if there is a bonding here, between the CDS and the reduced graphene oxide, then it will start rolling. So if you go to the previous step, this is the pictorial photograph that we are observing. So that it start rolling from the both the end, and it is not able to roll further. So this is the DFT optimized representation, and it is also actually stable, because when we do the DFT calculation, if the set parameters are not stable, then what we are proposing will not be optimized. So theoretically, it is also reliable. Experimentally, we are also observing. So we just need to prove that thing. So this is the mechanism. So through the modified Hammer method, this multi-layer graphite are reshuffled like this. Then through sonigation, they are single layer like this. And we put the nucleophile, and the bond are formed here, and it start rolling. Now the new thing here is that cadmium sulfide exists in a cubic form. So a single molecule and a 3 or 4 moiety molecule, the 3 or 4 moiety molecule will have a better strength, will be more stronger than a single moiety. So it is able to pull up the sheet like this, and this new complex is formed. And this is the pictorial presentation from TM images. Okay, these are various technical data showing that through various theoretical explanation that we are forming the bond like this, and it is linked through this, and this is the Cuban structure. And these are various other explanations to prove that it's actually happening or not. Okay, this work has been published in scientific report. After the work has been done, we need to show some application. Unless you don't have any application for the work that you are doing, actually it will be useful for the literature, but it should be somewhat commercially valuable. So we have worked on some piezoelectric effect. By piezoelectric effect is that if you give some force over the material, can it generate current? So we have worked on this. This is our compound GNOCC. We have worked with this TPU. It is an elastomer. It is required to make the film. We have worked on up to 20 weight% where it gives a very good piezoelectric effect. And we are also able to explain with a new theory which we propose, active dipole theory. So this slide shows that we have made a flexible piezoelectric material nanosensors, and these are the setup. The compound is still intact after even we roll all these things, and this is the result of the current generation up to 20%. It is giving about 4 times. Now this is the active dipole theory which we propose, that in the nanotube where our compound is there, if we apply the force, it will generate the current, and when we release, this is the applied force and this is the release thing. So on the applied force, it gives higher current, and this is the small current is on the release thing. So this is the active dipole theory that we have proposed. This is very recent work. Due to Corona, we are not able to continue, but still we are working offline in the sense that from our respective home, and we have often given this disclosure for this year in 2020. Okay, in the similar way, the next work is with different quantum dot. Here we are working on zinc sulfide. Previous one was on cadmium sulfide. Cadmium sulfide exists in Cuban structure. Zinc sulfide doesn't exist in the Cuban structure, so a single moiety will be there. So here we are forming multi-layer structure instead of a rolling structure. This mechanism is almost similar. Okay, these are few of the presentation. I think I will be able to explain here. So this is the graphene sheet. So, and from here, they are forming a bond. So this is, okay, I will explain here. In the graphene sheet, there are lots of functional groups like epoxide, then COIs. So whatever functional group that is available at the edge side, these are all accessible to any nucleophile that comes in contact with the compound. But the functional group that is in between the layer, they are not accessible. So it is a waste of all the functional group that has lots of capability. What we are doing is that we are making a, let's go. Okay. So what we are doing here is that we are making a spacer. And when the spacer goes inside, it increases the distance between the two layers. And when we do a sonication, it's a very simple thing. We are able to isolate the single layer. So after we isolate the single layer, the nucleophile can easily attack and break the epoxide ring. So this is oxygen, and this is the zinc, and this is the sulfur for this case. So this is the nanogate formation for one single layer of graphene. This is for the double bilayer, and this is for the larger scale, and this is for the multi-layer. So these are the optimized picture from the DFT calculation. So from the DFT calculation also, such thing can be stable. So experimentally we have proved, and theoretically we have proved. So these are the pictorial presentation. This is not that good. And okay, for the application point of view, we have used this for the hydrogen generation. Here, these are the compilation of the hydrogen generation. This is the compound. Previously, cadmium sulfide in graphene oxide, it is already reported. But in our compound, the cadmium sulfide goes inside the layer of the GO, and through the nanogate structure, they exist there. So the activity is about almost 10 times. So here we are proposing that due to the presence of nanogate, we are giving lots of new activities like the piezoelectric effect and this for the even for the hydrogen generation. And we have to explain this. So we are proposing this mechanism why they are able to generate this much amount of hydrogen or electricity, whatever it may be. Everything is due to the movement of electrons. So as we know, this graphene has high capability of storing lots of electrons because of that pi conjugation resonance structure. And when we have this nanogate structure, the energy that we get from the sunlight, it goes here, and from this nanogate structure, it comes to the graphene sheet, and from here, due to the new band gap, we have a new band gap here. It goes to the co-catalyst, and this co-catalyst will convert the water to the hydrogen. So if we replace this particular part, then we are able to generate this hydrogen. So the previous report is that they doesn't have this nanogate structure. So whatever they get is from the CDS and directly to the platinum. So this is also the new thing, and we have also disclosed this on another patent this year only. And apart from this, as we are also working on different applications, like in the tubular structure, we all know that if you give a potential difference between the two ends, as it is in a tubular structure, the electron will choose the least strength position to travel. So the electron will choose the center of the tube, not on the periphery of the tube. Now, at the center of the tube, we have our quantum dots. So if the quantum dots get the energy, then the normal they will give out luminescence. It is a normal procedure. So we are also planning to work on Kevlar application, that is for bulletproof base. First thing is that it is strong. Second thing is that if you press a button and it start illuminating. By pressing a button, we mean that we will giving a potential difference, and the quantum dot will illuminate. So these are the work which are still running. Okay, all this work were done by my students and postdoctoral fellow. We have this multifunctional nanometers lab at NCL. Previous explosive work and arsenic work was done by Devashish Ghosh. Right now, he is working as scientist in GSI Shillong. And Suman, Devi, Praveen Kumar. Apart from that, Nimrod and Namdev also work as a postdoctoral fellow, and various funding agency collaborator and industry partner. So I would like to conclude here. Thank you.
So the session is open for a few questions. I would request if you would like to start some questions.
Dr. Niraj, are you getting my voice?
Yes, I can hear you.
Okay. Just have one question. As you have mentioned that you have made some arsenic detection for water.
Okay.
Am I correct?
Yes.
So do you also plan to make a detection instrument for any other food or plant, something like this?
Yes, we are actually in NCL. We have lots of division. So there were lots of requests from the biological division saying that even for the arsenic in water, arsenic can also be contaminated in fruits also. There
in soil, in plants, in food.
Report in soil also. So not only for arsenic, for potassium, for any other element. The only thing is that from a chemistry point of view, we just need to fabricate the material. It is just about the transfer of electron from one, for example, as we have presented that there was a bond formation between the arsenic and my compound. So if arsenic want to give an electron, and if I make a compound which can accept an electron, for example, then it will be able to detect selectively. Hello, Dr. Maiti.
Hello, I'm getting a voice.
Yeah, I'm just removing Dr. Maiti. Okay. So right now we are not actually working on particular work, but there are lots of plans, there are lots of requests, and actually we need manpower to actually work on that.
Okay. And my second question is, what is the sensitivity level of your detection, that instrument that you have made for water? Or the low detection
level.
Detection
level, na?
The
Okay,
low level. The low level.
Yeah. For arsenic, we went up to 10 ppm.
Right.
And basically, it depends on the actually in the sensor. Our compound is there. Our compound will interact in the molecular level. Even one molecule of the analyte can be detected. But to detect that thing, our compound will be working. But we need another detector which will be detecting the light coming out. This is for a quantitative analysis. So if we have a good detector, then we can even analyze even one molecule of arsenic.
Okay, thank you. But it's still not in the market?
Yeah, it's still not in the market.
Okay.
Whatever is in the market are basically...
We have them, but they have a load limit of around 1 ppm. They cannot go below 1 ppm.
Okay. I think our compound can work below 1 ppm also, but only thing is that we will be coming up basically...
If that comes out, then we are waiting for it.
This year. So by next year, I think it will be available.
Okay, we have been working for the last 10 years in arsenic, and we still have some projects working on arsenic. So if your instrument comes out, that will be very helpful.
Yeah, we can work together now.
Okay, thank you.
Yeah.
Okay, and then the other questions? And if not, then let's move to the next speaker because we're running behind the schedule. So the next speaker we have is Dr. Geetgovind from Dr. Geetgovind affiliated to, let's see, his affiliation. He is affiliating to the National Botanical Research Institute, Lucknow. And he would be speaking on the post-treatment of treated wastewater, photoremediation, and geofiltrations. Introducing the speaker briefly, Dr. Geetgovind has received his PhD from University of Lucknow, and he has worked in Center Pollution Control Board, Agra, and his research area includes photoremediations, abiotic stress biology, environmental monitoring and assessment, environmental chemistry, and environmental analysis. Dr. Geetgovind has published several research articles in SCI journal, and also have 5 book chapters. And currently, he is working a plant and plant ecology and climate change science division, CSIR-NBRI Lucknow. So I request Dr. Geetgovind to start his presentation, please.
Thank you, Dr. Rakesh, for introducing me. And once again, thank you to Dr. Sapna and Dr. Niraj for arranging this conference. So I would like to share my slide. Is my slide visible?
Yes, it is. It is coming.
Okay, thank you. Okay, the topic of my topic is today is post-treatment of...
Okay.
Hello, are you getting my voice?
Yeah.
Okay, so I'll just maximize. Okay, is it okay?
Yes.
Okay, the topic of my is post-treatment of treated wastewater by phytoremediation, this is rhizofiltration. Actually, why I've taken this topic is in a very sort of span of time, I think Manipur has the first SDP developed in Langol, somewhere in Langol, and the total volume that can handle is around 27 mld, if I'm not wrong. And as at present date, I think only around 9 mld has been processed. So going further, what is wastewater? Wastewater is anything water that has been affected by the domestic, industrial, and commercial uses. And the wastewater, what it contains is that it contains a lot of organic and inorganic substances, but I will be mostly concentrating on the inorganic part. In the inorganic part, it contains a lot of sodium, calcium, potassium, magnesium, and heavy metals and metalloid. So we all know that if the wastewater contains a high concentration of nutrients, then when these waters are released into a water body, then we have this algal bloom and the degrading of the environment. And then in the summer season, we see that a lot of fish populations are dying in the river. So to avoid all this, so there are a lot of technology. Okay, and okay, coming to this, the wastewater that is released usually after being treated in the ETP or the effluent treatment plant, after even treated, and when they are released, it contains a lot of heavy metals, and some of the heavy metals that I've written are arsenic, cadmium, chromium, copper, and so on. And some of the like arsenic and cadmium are known to be carcinogenic. That means it can cause cancer to a human being. And okay, and luckily in Manipur, we do not have a lot of industries, but what are some of the potential sources of this heavy metal that can be found in the ETPs of the wastewater? The sources may be that we have a lot of metal finishing industries in the market area. We have the electroplating some industries. Okay, we don't have minings, and we have a very negligible amount of textile industries. So they are some of the, they have potential sources of heavy metals in this wastewater. What I've shown you is a brief diagram of how ETP looks, a schematic diagram. Actually, the first thing what they do in the ETP is, we have the first part where they do the screening part, where the big particles are removed so that it doesn't clog your pipes and all those. And then we have the grid chamber, we have the primary treatment, we have the secondary treatment, we have the tertiary treatment. Ultimately, actually an ideal ETP has a post-treatment, but some of the post-treatments are reverse osmosis. We have the electrokinesis system, and they are very, very costly to handle. And I think in one of our previous talk by Mr. Sonamani from Mumbai, he said that even the GDP of Manipur is around 22 crores. So for a very poor state like our Manipur, it would not be possible to have post-treatment system in the SDP that has been developed in the Langor. So in the search for a very sustainable and eco-friendly technology, what we do is we can adopt the technology of phytoremediation. So what do I mean by phytoremediation is, it is a technology in which plants are used to remove or destroy or sequester hazardous substances from the environment. And usually phytoremediation can again be further classified into phytoextraction. What it does is it takes up the metal from the roots of the plant, take up the metal from the soil, and then it is transported to the upper part. That is what we call the phytoextraction. The second part is the phytostabilization. What it does that the heavy metal that is present in the soil, it is the root extracts some organic acid. So it stabilizes the heavy metal in that soil, and it is made into an insoluble form, and then it cannot be leached out. Then we have the phytovolatilization, where some of the organics are converted into a methyl form, and they become a volatile, and they can be released into the air. And the final part is the rhizofiltration, which I shall be concentrating on. Actually, rhizofiltration is the use of plant roots to remove metal from the effluent water. And in a rhizofiltration process, what are the things that we need to consider is, if we are planning to have a rhizofiltration system in a project, first what you need to do is that you have to characterize the wastewater. We need to understand what are the nutrient content that is in the wastewater and the heavy metal content in that water, and the pH, because the pH plays a very important role in the solubility of a particular metal. And then comes the next part is a designing of a plant. To design a rhizofiltration process, we need to know how much volume of water that we are going to treat on, and the flow rate that it is flowing per day, and the metal uptake rate of a particular plant. The third process comes the selection of plants. Actually, selection of plants is the most important part of this component. We need to take a hyper metal accumulator. And what we understand by a hyper accumulator is that there are certain plants which can accumulate metals into their biomass above 1000 ppm. Okay, and we also look for a plant which has a very fast growth rate. And the other point is the plant should have a very high biomass, because if the plant has a very low biomass, then the amount of metal that it can accumulate will be very low. So we look for a plant with a very high biomass. And another point that we also look for is it should also have a very root system. In the case of a rhizofiltration technique, the most important part is the root system, because here the remediation part of the metal is going to be done by the root of the plant. And finally, that again we're going to do is that after designing the rhizofiltration, we have to look at the design system, construct the system, evaluate and modify. It is not so simple that you just made it and then you can run it. You need to evaluate, okay, is this particular plant functioning properly or not? And then if it's not properly, then we can again select different plants, or we can also modify a little bit. And then finally, we evaluate the performance. What is the removal efficiency of this particular plant? And this is how we rhizofiltration process is made. Now here I'm sharing with you a project that we had done sometime back in Jasmau, which is Kanpur in UP. The title of the project was Phytoremediation of Ternary waste contaminated site at Jasmau, Kanpur using economical plant. In India, there are some very important ternary processing centers in India, among which Kanpur is one of them. We have Kolkata, we have Chennai, we have Bangalore, but in Kanpur, we have a very big ternary processing plant. And in this ternary processing plant, they use chrome tanning. And this chrome tanning contains a very high amount of chromium. And it is estimated that around 2,000 to around 32,000 tons of chromium are consumed every year in this ternary industry. And what are the environmental hazards that we face from this ternary industry are actually the chromium that is being used are released into the environment, and they are very toxic. It actually contaminates the topsoil and puts chromium into the wastewater in agriculture. Ingestion of the chromium can cause carcinogenesis, pulmonary disorder, deformation of body parts, etc. Okay, I'm showing you a little bit about a picture of the area that we had worked on. Actually, due to the pollution of chromium, you can see the water is turned red, and this is the sludge that has been byproduct of the ETP that is near to this particular Jasmau, and it is deposited to this field nearby in Jasmau. And you can look at this poor old lady due to the contamination of chromium, and now her body has been deformed due to the ingestion of chromium by a body part. In that project, we had a small, out of the many objectives, we had one small objective, which is called the development of small-scale aquatic macrophyte-based root zone treatment plant for improving the ternary effluent quality acceptable for irrigation purpose. This is what I'm showing you is a aquatic macrophyte treatment system for using post treatment of the wastewater. The first tank, after the water has been treated in a wastewater plant, the water is collected in a pond, and then you can have a numerous number of ponds. But here we have taken a 3-pond system, where in the first pond we had used a typha, the second pond I had used a phragmitis, and in the third pond I had used an Iconia. Why I had selected these plants are the typha and Iconia are known to be a very high accumulator of chromium. They can accumulate a lot of chromium into their biomass. And the purpose of choosing this phragmitis was, even though they are high accumulator, their root systems are very, very short. So it can only penetrate up to the high level of the water system. So now I also need a longer root system where it can also remediate metal from the lower bottom of the tanks. So that is why we had to use a phragmitis. And in the system, the chromium level was around 1.5 to around 4.5 ppm in the ternary waste effluent. As per the BIS standard, the chromium level cannot be exceeded above 50. Okay? So after this AMTS system, after the treatment of the water system, this is the parameter of the water. Before the treatment, the pH was around 8.3. After the treatment, the pH had decreased to around 7.6, which is almost normal. EC, which was around 8.11, had dropped to around 6.89. And the chromium level, which was around 3.2, had now been dropped to 0.89, which is around roughly around 60%, 67% efficiency in removal of the chromium. And the water that was treated by the AMTS system was used for the irrigation of the nearby fields. And we found that in all these plants like barley, wheat, urad, ladyfinger, mung, and maize, sorghum, and black mustard, no chromium was accumulated in its edible part. Whereas in some plants like coriander, methi, and spinach, we found that the chromium was in its edible part. And gram, pea, and rice, they could not survive. So it shows that this particular plant is not suitable for chromium affected area. And this particular data has been published in our paper, which was published in 2013. So even though I have only shown a work of removing of chromium by the rhizofiltration system, this particular technique can also be used for removal of various other heavy metals, like as shown by the various workers, like even chromium, lead, and nickel can also be remedied by using pistia, as shown by Abu and Bakar. Whereas again, cadmium, copper, chromium, nickel, zinc can also be used by using brassica and helianthus, as shown by this Disenkov et al. Lead can also be remediated by using carex pendula, as shown by Yadav. And arsenic can also be remediated by using teres vitata and teres elicata by Hong et al. Okay, so what is the benefit of this particular? Why are we promoting rhizofiltration? Is that the conventional methods are very, very costly. These are very costly. So a post state like Manipur will not be feasible for a long term. So if we use such cheap technology and as well as environmental friendly, which will only cost you around $2 to $6 per gallon of water. And this is a cost benefit comparison between remediation between conventional and a phytoremediation. You see in an area which is polluted with lead in 1 acre of land, if we do a phytoremediation process, it will only cost around $150,000, whereas a conventional method, which would cost around $500,000. And the project that we save in this is around 50 to 60% save in the budget area. Similarly, for all other premises, you see that we are almost saving 50 to 80% if we are using the phytoremediation process. What is the advantage of a phytoremediation? All other processes, it may be ex situ, but in the case of this, it can be in situ as well as ex situ. And this particular technology is also suitable for organic as well as inorganic pollutants. It is cost effective compared to the conventional method, easy to implement, maintain, and acceptable by the public. The conventional method, this is where you use a lot of chemicals. If they are not properly removed before being released into the environment, they cause another secondary problem. So that is why a lot of public have hard time in accepting when they have a ETP coming up near their house. But if we use phytoremediation, the public usually accept them because we are environmental friendly. And conclusion and the future prospect of this is that the rhizofiltration technology is relatively inexpensive procedure with low capital and operational costs. The method may be conducted in situ with plants being grown directly in the contaminated water body, or ex situ where plants are grown off-site and later introduced into the contaminated water body. The technology is ecologically sustainable. Okay, and the projected market value of phytoremediation in the coming years is estimated to be around 214 to around 370 millions. So I would request our young researchers that is right now in Manipur that you should take up such project, because now that the STPs has started functioning in Imphal, and they would be releasing this water after being treated. So how efficient the technology may be, it is never 100% accurate. So there are still some amount of nutrients or heavy metals still present in the wastewater, and I'm sure that they will be releasing it in the nearby. And as one of the previous Dr. Narendra has said that due to climate change, the rainfall are decreasing year by year. So if we can reclaim this wastewater and then use it for an agricultural purpose, that will also be sustainable for the future. So with this in mind, I am thankful to Dr. to the KHUMANG team. Yes, I find up. You can ask me if
Dr. Rakesh.
Hello. Yeah, Niraj, I think you have unmuted all the participants. That's why I was not able to speak. So thank you, Dr. Geetgovind, for a nice presentation. So session is open for some question answers. Participants, if they wish to ask a few questions to Dr. Geetgovind, please. So if not, then I have one question I would like to ask
please,
to
please go ahead, Dr. Rakesh. I'm waiting. Because you guys are based in Imphal, I will be looking forward to your question.
Right. So you talk about this reserve filtration process using
right.
So what is the criteria for selection and identification of such plant? And now that is a question. And second thing is that since they are able to, purify or filter or recover this heavy metals which are present in wastewater.
Mm-hmm.
So how is it possible? Is there any possibility to recover all this, heavy metals which are already, taken by the plant? Is there any way you recover it for use again?
The first step of the phytoremediation was we were only trying to accumulate the metal into its plant, but now the next step is to how to recover the metals from the plant. That part what we are now defining as the phytomining. So people in Taiwan has successfully done that. So, but why this particular phytomining is not successful is that because for a cheap metal like copper and zinc and iron, the process of recovering is so high that it is not feasible. But for a costly metal like the gold and platinum, we found that it was feasible. So we were still working with it, and then once we have a viable phytomining system, and I think it will be in the market. We are still working on it.
Okay, great. And another thing is that I was asking, how do we identify or how do we select the plant which are having the maximum reservation?
Okay, there have been a numerous number of work which we have identified a lot of hyper-accumulative plants for different metals. So, for example, you can use sunflower plant, which is a very known hyper-accumulator for your for a lot of metals, like Teres vitata for arsenic. So, if you are wanting to go by a ready-made answer, then you can have a lot of literature in the net where they have identified more than around 150 hyper-accumulators. But if you are wanting to find if your particular native plant is also hyper-accumulator or not, then you need to do a little amount of studies and see that if it is able to accumulate more than 1000 ppm per gram of your plant parts, then we can identify that plant as a hyper-accumulator.
Okay. Thank you very much. Thanks a lot. Okay, then let's move on. Let's thank the speaker once again. Thank you, Dr. Geetgovind. Thank you very much. And then we have a third speaker. This is Dr. Maiti from Advantek Analytical Instruments, Advantek Instruments, Mumbai. And he will be talking on the Make in India in Analytical Instrument to improve research quality. And introducing the speaker, Dr. A.K. Maiti has done his, Niraj, is it Dr. A.K. Maiti is there?
Yeah, he is there.
Okay. So yeah, Dr. A.K. Maiti has done his PhD from IICS Kolkata in the field of spectroscopy, and he has done his postdoctoral research work from University of Mainz, Germany, under that fellow. And also he has carried out his postdoctoral research from University of Windsor, Canada. He has been a visiting professor in University of Belgrade, Spain, and he has almost experienced for 2 decades in the R&D as well as in the development of analytical instrument. Currently, he's having one company, and he's the director of Advantek Instruments based in Mumbai, manufacturing various analytical instruments, which are the rare cases in India. So I welcome Dr. A.K. Maiti and start his presentation, please.
Thank you, Dr. Rakesh, for introducing me. And at the same time, I will thank the organizer of the KHUMANG and particularly Dr. Niraj. You can hear me, no?
Yes.
You can hear me?
Yes,
yes.
Dr. Niraj, to once again associating me with this KHUMANG, and I am very happy to meet all of you once again. One thing I'm missing, the beautiful lunch I got last time in your university.
So we will have.
Okay. Anyway, today I will be very happy to share my experience. Can I operate from my this PPT?
Yes. That will be continued.
You can see all this. Okay.
Can you go to full screen?
Full screen.
Yeah. Slide 1. You can press F5.
F.
F5.
F1.
5. Punch.
F1. F5. No, nothing is. Okay, thank you. Okay. F5, no?
Yeah, it's working.
It's working. So today, practically Make in India is now very common concept throughout India. There are many aspects of Make in India. For example, one is job opportunity, cost reduction, and today another is today I will explain from different point of view. That is the how it can improve the quality of the science if we make something in India, analytical instruments. So practically I was in business since 2011, and that time I was promoting foreign instruments and to different institutes and universities. And that time I felt that when I was in ISCS doing the research, that time I saw the research expense was very less. For example, we were our professor, who was a very all India level great professor, but he also had to struggle to get only 5 lakh rupees to purchase one fluorimeter. And on that fluorimeter, many students, not only my professor, other professors also were depending, their students were depending. And for 15 years, one fluorimeter maybe 15. So we are working on that, still they were not getting. But still, slowly the research financial burden has increased. So that time I felt that research burden has, that is from 2011 when I was marketing foreign instruments, that they are increasing this burden unnecessarily, and in my experience it is too high. In some cases I feel it is unnecessary also. So that time some idea came to my mind that whether we can make something. And I am not from the, I am from the pure science, that is my PhD is spectroscopy, molecular spectroscopy. So to make one instrument, you need the constant idea of the electronics engineer, nowadays software engineer, so many personalities, professionals are required nowadays. So still I took a step on that I have to do something. So that time our motto is basically you think we make, you think we make. That is, scientists will think about the instrument, about the project, and we will make. So by the time we have grown a team who are expert in electronics, who are expert in software, and also mechanical team who can build up the instrument also. So now we have assembled during this, I think last 4 years, from 2016 onwards, I am doing this. So I normally get the idea by interacting with the scientists and see the market potential also. And also being a basic scientist, I also feel some importance of some basic ideas. If somebody wants to try some basic ideas, then also I jump into that idea and try to do something. So that is our idea. The scientist wants to do some experiments, but that instrument is not available in the market, and that we do for them. They increase when we do, that is at the first step, definitely we are not very perfect, but we do something. And then second step, we give it to the scientists. Then scientists give us the idea about, such say you have to increase the sensitivity, ease of handling, aesthetic look, so many things they give the idea. And slowly we improve the instrument and come to more near perfection. We try to come. That is our concept. So far we have developed 3 instruments, 3 products, and that I am going to describe here. So one day I was sitting in a laboratory on NIT, and there one PhD student told me that whether I can give one powder sample holder to study the fluorescence. Then I told, I had someone's simple piece with me, I made it. Then they worked with it and got a good fluorescence graph. And then the scientist told that my that accessory was not easy to handle, also a little bit difficult. It was not going smoothly inside, so I had to struggle for it. So that time he suggested, that scientist suggested me, you do this changes, do that changes, and so many things he suggested, and we are continuously going on developing this. And then finally we had the problem of the coating. So one coating should be there, and that coating must not be washable by the acetone, which is a cleaning for the solvent, cleaning solvent for the sample. So finally we solved that problem also, and now it is regularly being used. And that time he told that he had the fluorimeter. He had the fluorimeter, that costly fluorimeter, around 10 lakhs rupees. But to purchase the powder sample holder, he needs another 1 lakh rupees. So he had not that amount of fund that time. So my students are going to some other institution where they have the fluorimeter as well as powder sample holder. And it is difficult to get the time, date, when they have the time, I don't have time, like this. So many difficulties. And now every day, anytime, they can take many 10, 20 powder sample graph, no problem for them. So this is a how it works. First of all the imported fluorimeter companies who are working, their powder sample holder has a different base from the liquid sample holder. Liquid sample holder is like this. By quartz cube, in the quartz cubed, they put it here. When you have to study the powder sample, first of all, you remove this base, this black base, you remove and put the powder sample base, and they put the powder sample holder, and that cost, I was selling PTI companies, Horiba companies, that is costing 2.5 lakhs. And some Jasco companies, Japanese, they are more than 80,000, almost 1 lakh rupees. And then I did it. I will tell you my price. This is a this I have made a sample like this, and put the sample here. It is just very little sample is required here, and cover it by this quartz plate, and then put a stopper and then screw up. And when this is ready, then there is a mark here on the top, so that if you think that particular position, you have to maintain that position, then you can maintain that position also, particular position. So it is exactly 45 degree is not good to keep it. So little rotation from it, because exactly 45 degree you may get the reflection also. To avoid reflection, little bit rotation is necessary. So ultimately we put where? Here. So this liquid cuvette holder you have not to remove. In the same cuvette, the cuvette holder where you are measuring your liquid sample in the same, you just make it ready and put it here instead of. So it is very easy in handling, and price will be surprised to know PTI company 2.5 lakhs and recurring expense of 1 lakh rupees per annum, and JASCO is more, 1 lakh almost. My company, my instrument, this accessory, powder sample accessory cost only 20,000 rupees and almost no recurring expense. So these are the this is the group size I have given. 6 millimeter is the width, height is 9 mm, and depth is 0.5 mm. And we can also change if you like, even smaller we can give. This we have kept as per the requirement of this scientist. So another thing, important thing of the Make in India, that you can do you can get the customized item. You can get of your choice, of your liking, because if you are since you are a scientist, you will have hundreds of ideas, but market you will get some limited instruments. So your instrument of your idea only Make in India can make for you. So these are the steps. So these are the steps as I described. Now I have another product is portable fluorimeter. It should be basically portable fluorimeter. It is not proper fluorimeter, it is portable fluorimeter. One day I was sitting in an NCL, NCL Pune, one scientist, Dr. Amitabh Das, told me that he wants to keep on fluorimeter in a production department. That is chemical production department, where huge production is going on there. So this costly fluorimeter, which is very sophisticated, which needs AC room and so many things, dust-free room, AC room, cannot be kept. So he knows the excitation wavelength, he knows the emission range also. So in this situation, he wants to excite the sample by one particular wavelength, and he wants to measure the total intensity of the purpose was the initial intensity of initially the molecules were non-fluorescent, reactants were non-fluorescent. As the reaction progresses, the molecules become fluorescent. So as we just measuring the fluorescence and measuring the fluorescence intensity, the reaction chemists in the production department can tell about whether his reaction is progressing or not. So that is the purpose. So the idea is very fantastic, and I thought I will do it. Although that time I had zero sense of making any instrument. I have no electronics knowledge and software. So only theoretically I can say yes. Yes, why not? Because I will give one excitation and I will get the emission and I will theoretically all can say. I took it, but it took time for me to make it, because I need to contact one electronics engineer, and to get an electronics engineer, it's a good electronics engineer, it's a tough job basically. There are electronics engineer mostly as bookies, and they can tell you what is given in the book, they will write that. But if you ask him to new design, that is very difficult to get, very uncommon to get. So it took time, but I did it. How this I did it, and the price definitely low. So I did this. It is the final look. Of course, there are I changed the getup I think 3-4 times before it is before it has come to this stage. Previously it was iron body, now it is a beautiful plastic body. It looks aesthetic, looks also there, and also there are gain selection is also there, so many things. And not only that concept, later another scientist of NCL thought, Dr. Nandini Devi, I can use this instrument, this simple instrument for bacteria sensor. For bacteria sensor, she knows about the gold nanoparticle bonded with the bacteria. And gold nanoparticle will bond if there is bacteria. If there is no bacteria, there is no bonding. So different functional group is attached to the gold nanoparticle, and different functional group will attach to different kind of bacteria. So just so if there is attachment to a particular gold nanoparticle with particular functional group is attached to some bacteria, that means that particular bacteria is attached and it will give fluorescence. And if it doesn't give fluorescence, that means there is not binding, there is no bonding, there is no bacteria is present. So she has established this technology with my instrument, bacteria sensor. This bacteria sensor, because she knows her excitation wavelength, this emission range also. So accordingly, we have made this. So it is mostly mostly customized. And it can be done. And arsenic sensor, as Dr. Luwang was telling, that we have a joint project with Dr. Luwang to make the arsenic sensor, and we are making it. And another reaction kinetics, photoreaction, waste data analysis, environment, community luminescence, and education particularly. This can be, because in education, even in the college level, they can to know some characteristics of luminescence. And one important characteristic of luminescence is that is the linearity. So we have achieved the linearity from 10 to the power -6 to 10 to the power -9 mole per liter. That is one good linearity we have got with this instrument. And there is a gain selection also in this. Historically, I have given this is all are studied, studied in coumarin 6 in acetonitride, because the first scientist who told that he will measure, that was a compound, not coumarin, but emission range is emission excitation emission range is almost similar to coumarin. That's why we are studying coumarin 6. So specification is 405 nanometer, which is Dr. Nandini Devi is using, and the other wavelength we can select. Emission range is like this, and actually as per this excitation it can be changed. And photodiode, LCD display, less than nanomolar range, we have come to coumarin 6, we can detect in acetonitride, and 4 dynamic range linearity, that's a good and we can have the gain selection, very low concentration where values are very less, difficult to get. There we can increase the gain. And sampling mode, here is the system where we can use in a single cuvette holder, liquid, of course, by quartz cuvette you can place my powder sample holder. Even I have keep a place so that in the cuvette holder, there is glass slide also you can place there. Because in other imported instruments, you will get to another cuvette holder to place a glass slide. But I made it all in one cuvette holder. Everything will be put in there. That is the beauty of the Make in India, that you can make of your own. Why not? Why should I why have to change the liquid base to put the glass slide? Why have to change the liquid base to change the powder base? So all in one is made in a. So power is normal power, rechargeable battery we can give, and size is not very big, and weight is almost 1 kilo. And it's not costly, of course, because we are making it. Third instrument we have made is the potentiostat. So for the development of any instrument, I have some small history. When I developed fluorimeter, portable fluorimeter, as proposed by Dr. Amitabh Das of NCL. By the time he has shifted to CMCRI, Bhavnagar, he told me to visit his institute and to find any interesting thing which I want to develop. So what I visited and met many scientists that time, and one scientist told me he has developed, Dr. D.N. Srivastava, he has made a electrode, that is a carbon, that is plastic chip electrode. Plastic, he had made plastic chip electrode, which is very cheap compared to the platinum electrode and this, that, which is unaffordable and disposable also. So after some few uses, you can throw it. Very, very cheap cost, some few rupees only. Then, but it is not getting used because the reason is very interesting, because that time all the scientists, all the potentiostats were imported potentiostats. So imported potentiostats costing from 3 lakhs minimum to it goes to 30 lakhs, depending upon, of course, facilities. So those who have 3 lakhs of money, so they can buy platinum electrode also. So why they will go for this cheap thing? So only we have developed, we have patented, and we are using. So if you can develop a potentiostat, low cost potentiostat, then it will be this electrodes will also be used. And I knew, although I am not from this field, my background is from spectroscopy field, electrochemistry, I didn't have any idea, but I knew while I was marketing these foreign instruments, this potentiostat has a very high demand in India, because it is highly sold in India. And that time I told, okay, no, this is very interesting project, I want to take this project. So we have developed it. It's a very complicated electronics, lot of electronics, lot of software. So really we need some good electronics engineers, software engineer, and ultimately we have developed it. We can measure, so far we can measure only potentiostat, cyclic voltammetry, that is CV, and stripping. Galvanostat we have not still developed. So we are using the plastic chip electrode devised by Dr. Srivastava, D.N. Srivastava of CMCRI. It looks like this. It is the reference electrode, and this is the working electrode. It is same as this, only one plastic packet is given, so that area of exposure is limited, so that a particular quantitative measurement can be done. And not only plastic chip electrode, we can measure we can use the screen printed electrode also. So screen printed electrode so far we have not made, but its technology is not very difficult also. If scientists also encourages us, we can do this. It's not also difficult. So now it is coming from abroad. It's costly. Some I think one piece will be around 10,000 rupees. But plastic chip electrode, very cheap, around 30 rupees. But plastic chip electrode, 10, 20 times you can use after that. So we have made our indigenous electrochemical cell also. This is our electrochemical cell, and this is designed by us. That is this is the working, this is the counter electrode is clamped here. This is the working electrode, and here is the reference, that is the reference electrode, that is saturated KCl, where silver coated aegis, that is silver on the top of which to maintain the standard potential. And three terminals coming out, and now finally our electrochemical cell looks like this. It looks very nice. It is designed by us. But if you go to purchase electrochemical cell in the market, imported one will take 20-30 rupees or more, I don't know. But it is just our own design. Few thousand. Now we have connected finally to the cyclic, this our apparatus is like this, where we can measure stripping as well as cyclic voltammetry and look our graphs, cyclic voltammetry. So this is our cyclic voltammetry. Of ferroferric, it is looking almost as I see in the literature. And stripping portion is very interesting. In this set, mixture of cadmium, lead, and it is mercury. Cadmium, lead, and mercury. This will be cadmium. This is the heavy metals. Deep in these heavy metals, so it can be used in environment purpose also. That heavy metals present in the environment, water, that can also be detected. And it is almost ppm level. I think it is here. So we have to, we can improve our also sensitivity. So now if you think our cost will be very low, very much affordable by the college students, or some around 60,000 including everything. Around including everything means the cost, this, then electrochemical cell, and then some bunches of electrodes. So few months you have not to purchase. And if you buy that Dr. Srivastav's carbon paste electrode, plastic chip electrode, that is little charge. I think 1,000 rupees you can buy many. So this electrochemistry is in 12th standard, 11th standard, 12th standard, even in the college, MSc, and then research. So this instrument can be, at least in the college level, can be introduced. So far it was not introduced because of the cost, but cost is so low. So this can be, so complete package we can give in the 60,000 now. Anybody can, college, university can use, and also can be used for the research purpose also. So finally, I want to tell you the Indian scientists present here. Make in India in analytical instruments will be successful only if scientists use it. Its standard will increase. Today I can give you, you don't expect that I will give you some American standard. May not be, maybe better also. Just like for powder sample holder, it is, I claim to be better, cost wise and performance wise. Its standard will also increase by their guidance. That is, you think, I make. What I told in the beginning, introduction, conclusion also, I will say same thing, that you think and I will make. Its standard will also increase by their guidance. It will increase the boundary of scientific investigation of the scientists. Just like Dr. Nandini Devi is now doing the simple way, studying the that is biochemistry, that is bacteria. At the same time, the skill of making an instrument of us. Basically, we also, unless you give something, we will do something. And if you give a problem, we will ask our engineer to do this, do this. They will study, and they will increase their potential. They will study more, they will do more. Their skill will also increase. Skill of making an instrument make in India will increase. So slowly our skill definitely is increasing. So first instrument what I took, that is portable fluorimeter, took long time. After that, potentiostat much more difficult. Electronics and software wise still not that much time. Slowly our skill is increasing definitely. One day all sophisticated instruments will be made in India. It is not only me, also I request all of you to think in this way, that we will all use Make in India instrument and guide these people like us who are dedicatedly doing this Make in India, to develop these guys. My personal experiences developing analytical instruments is much less difficult than rocket technology. Now I have come to an experience after doing Make in India for some time. It is not that difficult. Not difficult. Everything we are depending upon this foreign countries imported thing. It is not. Previously also it was not like this. If you see the history of development of science, C.V. Raman did a lot of many things. Even XRD also was done by him. But now we are fully dependent on these imported instruments. So if scientists encourage, it will be successful faster. So okay, thank you very much. Fine. Before that, I want to acknowledge Dr. S.K. Ghosh, Chemistry Department, VNIT, who helped me to develop the powder sample holder. Dr. Amitabh Das, who first introduced to make the take the project of portable Raman, also introduced me in the Bhavnagar as a director. Dr. Srivastav, who encouraged me to do the powder sample to the potentiostat. Dr. Nandini Devi, who is using my portable fluorimeter to find the bacteria, and recently he has written one more project with my instrument also. So he was asking the details of it, and Dr. Niraj Luwang, with whom I am long time associated by going through NCL and also associated to KHUMANG also through him. So. And we have taken one new project of detecting arsenic with this portable fluorimeter. Okay, thank you very much for your kind attention.
Okay, thank you, Dr. Maiti. Thanks for sharing your experience in the journey of developing analytical instruments, and very interesting to listen to your talk. I request, like audience, if you have any questions, you can ask one or two questions to Dr. Maiti. I have one question because the audience are responding. Dr. Maiti, I have one question. Talk about the Make in India analytical instruments. I was just concerned about the oil manufacturing, because when you talk about the fluorimeter, that is very nice, very impressive. But the components which you use in making the fluorimeter, for instance, let's take the example of photodiodes. Are you making the photodiodes? Or are you completely purchasing from the company, integrating into a system? That is the question. Like so, in again, like laser, you have filters. So those things I wanted to hear from, because from our institute, since I belong to a technical institute, so develop lots of instruments. We are still face that problem.
No, all the components we are purchasing from some company. Maybe some I have purchased some from China also, but now we are finding alternative source without China. Okay. For example, this potentiostat, there is one TFT screen. So far we were using it from China. But now we have decided, no, we will not use this. We will change it to suppose tab. We will give a tab on which we will see this all graphs and all these things. So we are also changing, and we are also thinking without China how we can survive.
Okay, very nice. Thank you. Thank you very much.
Thank you.
So then thanks once again. Thank you, Dr. Maiti. Thanks for your talk. Let's move on. We have a fourth speaker of this technical session. Let's welcome Dr. Miroslav Novakovic from Institute of Chemistry, Technology, Metallurgy, University of Belgrade. And the title of his presentation is Bis-B-benzyl components from liver world. Let me briefly introduce speaker, Dr. Miroslav Novakovic. Received PhD from Chemistry, University of Belgrade. Has postdoctoral experience from Fukushima Bunri University, Japan, and also University of Bologna, Italy. Research interests includes organic chemistry of natural products, photochemistry, medicinal chemistry, isolation structure, elucidations. He is the recipient of Caustic Foundations, and he has published several papers in highly reputed scientific journals. So let's welcome Dr. Miroslav Miroslav Novakovic and then start his presentations.
Thank you very much. I hope that you can hear me properly. Is it okay?
Yes. Yeah, clear.
Thank you. Dear colleagues, ladies and gentlemen, I am honored to be the participant of this beautiful e-conference, and I am thankful to the organizing committee KHUMANG 2020 and Dr. Swapna for the invitation. My lecture will be about bis-bibenzyls, bioactive compounds from liverworts. Liverworts are small and non-vascular plants which are present on every continent, including Antarctica. They are growing on wet soils or rocks or on the trunks in lakes and rivers, but not in the sea. They belong to bryophytes, which are taxonomically placed between algae and pteridophytes, and they are represented with more than 6,000 species. It is assumed that liverworts are the earliest plants, that they are old for 472 million years, and that they are the ancestors of all land plants that we recognize today. Oil bodies are morphological markers or botanical markers of these liverworts. These are membrane-bound cell organelles, which consist of many different types of compounds in carbohydrate or protein-rich matrix, and they are characteristic only for liverworts, but not for other two phyla, which belongs to bryophytes. Possible function of oil bodies are protection from herbivores, pathogens, cold temperatures, excessive light and UV radiation, and desiccation. Compounds found in oil bodies are mostly lipophilic monoterpenoids, diterpenoids, sesquiterpenoids, aromatic compounds, in particular bis-bibenzyls and bibenzyls, benzoates, cinnamates, naphthalenes, and isocoumarins. Bis-bibenzyls are chemical markers for liverworts, and they are the most characteristic compounds in this phyla. They were firstly isolated from liverworts by Yoshinori Asakawa in 1982. I was very happy and honored to be a member of Professor Asakawa's team when I was on postdoc studies in Tokushima Buni University in 2015. So he entered me this world of bis-bibenzyls and liverworts. Bibenzyls and bis-bibenzyls belong to stilbenoids. They exhibit wide range of biological activities, and to the present date, more than 70 biologically active bis-bibenzyls have been isolated and their structures were established. Chemically, they can be divided in cyclic or the microcyclic bis-bibenzyls and also acyclic or opened bis-bibenzyls. As I said, bis-bibenzyls are chemical markers for liverworts, but there are also few reports that they can be found in vascular plants, in higher plants, like in plants from Primulaceae family, Orchidaceae, and Dihaptelaceae family. While polyunsaturated fatty acids and triterpenes are chemical markers for mosses, and neolignans for hornworts, other two phyla of bryophytes, bis-bibenzyls are chemical markers for liverworts. According to the way of connection of bibenzyl units, bis-bibenzyls are divided into different types like marchanthins, isomarchanthins, ricardins, isoricardins, plagiochins, isoplagiochins, perotetins, etc. Here are examples of each class of bis-bibenzyls. And last example is perotetini, which is example of opened or acyclic bis-bibenzyls. The first liverwort that we investigated in our laboratories was Marchantia debilis from Cameroon, from Africa. The sample was kindly provided by Dr. Kenneth Yongabi Anchang from Cameroon. And in his previous investigation, ethyl ether and methanol extracts were investigated against bacterial infections connected with decubitus and diabetes. And he found that Brio cream, which he prepared with the petrol ether extract, was very effective in resolving open wounds. So you can see this photo where we can see the effect of 2-week use of Brio cream, which consisted, among others, of petrol ether extract of Marchantia debilis. Our further investigation was connected with methanol extract of this liverwort. From the methanol extract, we have isolated 4 novel compounds. It was stigmasterol from 3 terpenoids, but also marchansin C, marchansin Q, and perotetin E from bis-bibenzyls, and also one new derivative, 1'-methyl marchansin Q. As I said, marchansin C, marchansin Q, and perotetin E were novel, and 1'-methyl marchansin Q was isolated for the first time. Here are NMR spectra of this new derivative. On the left upper side, you can see proton NMR with characteristic signals of protons 7 and 8 and 7' and 8' around 3 ppm, 1' methoxy group around 4 ppm, and also characteristic 3' proton, which is shifted to higher fields because of the anisotropic effect of A ring, which is very close to this proton. So it's very nice for us for resolving the structure when we have some isolated signal. Carbon NMR, you can see on the left down part, also with characteristic carbons from methylene bridges, but also methoxy group, which was found at 1' position, but also 2 carbonyls were found around 180 and 190 ppms, which helped us to resolve the structure and see that it is Hinon moiety present in this molecule. Also, HSQC and HMBC spectra helped us a lot to resolve the structure, and high resolution mass spectrometry confirmed the structure we proposed according to NMR analysis. Another story connected with bis-bibenzyls and liverworts is story about Lunularia cruciata, liverwort found in Southern Europe, also in Serbia and in Japan. And in previous investigations, Japanese scientists found bibenzyls lunularin and lunularic acid, but also bis-bibenzyls of perotetin F type. You can see pictures of this beautiful liverwort. On the right side is picture under the microscope where you can see this like moon or luna shapes. So because of this, the name of this species is Lunularia. Our chemical analysis of the methanol extract of Lunularia cruciata revealed 13 compounds. One was bibenzyl lunularin and 12 bis-bibenzyls. On this slide are presented 7 new compounds of bis-bibenzyls, and 5 were novel perotetins E, F, and ricardins C, F, and G. These new compounds are divided in 2 groups. First 3 are perotetin type bis-bibenzyls or opened bis-bibenzyls, and from 4 to 7 are presented ricardin type bis-bibenzyls. All of them are very interesting in structure, especially compounds 2 and 3 and 5, which possess dihydrophenanthren or phenanthren moieties, and because of that they were violet in color, but also compounds 6 and 7, which possess Hinon moiety, and they were because of that orange in color. Analyzing the products of or all isolated compounds, we found the connection between novel perotetin E and compound 2, also perotetin F and new compound 3, and new compound 4 and new compound 5. And we concluded that these new compounds 2, 3, and 5 are products of intermolecular coupling of these two known and this one known compound. So this was an explanation for the structures of these new bis-bibenzyls. I took one example, NMR spectra of compound 5, which I found the most interesting. So on the left side, you can see aromatic part of proton NMR spectrum. On the right side, also aliphatic part with, you can see signal of methoxy group, but also signals of protons, which are from the methylene bridges of positions 7 and 8. Noisy and HSQC spectrum are presented on this slide. Also helped us a lot to resolve the structure. HSQC showed us which carbon possess which hydrogen atom, and noisy spectrum showed us some very important correlations through the space. And the most important maybe are HMBC spectrum, and all correlations helped us to resolve the structure of new compound 5. Especially you can see bolded correlations which confirm the Ricardin type of connection in this macrocyclic bis-bibenzyl. Compound 5, as I told you, I think is maybe the most interesting because it is very similar to Novan cavicularin. Cavicularin is macrocyclic bis-bibenzyl isolated from Cavicularia densa from Japan. And this is very interesting compound because it displays optical activity solely due to the presence of planar hilarity and axial chirality. No chiral center in this molecule is present. Also, this molecule is very strange. This ring A is distorted and for 15 degrees and possess a conformation which can we say it's similar to both. Our compound 5 possessed methoxy group instead of this hydroxyl group. Also in 6' position, our molecule possess hydroxyl group, and in this part it is not present dihydrophenanthren, but it was phenanthren moiety. So these are the difference of our isolated compound in comparison to Novan cavicularin. At the end of this part, new compounds 5 and 3, together with Novan Ricardin G, exhibited significant cytotoxic activity against A549 lung cancer cell lines with IC50 values of 5 micromolar concentration. Novan Ricardin G was even better with 5 micromolar concentration. It is interesting that both new compounds 3 and 5 possessed phenanthren moiety. Sorry, just a moment. 3 and 5, this phenanthren moiety, which is maybe important for this activity. These results are published last year in Journal of Natural Products journal. Another story is about biotransformation of perotetin F. Perotetin F was the most abundant compound isolated from Lonularia cruciata. And we came on an idea to do a biotransformation of this compound by Aspergillus niger, a fungal species. From the literature data and biotransformations of terpenes by this fungal species, we know that this fungi can do biotransformation in allylic positions or in basic positions if we have aromatic compounds, and also that they can, that these species can add hydroxyl groups not only in allylic but also on the double bond. So we firstly try to do biotransformation of limonin, which was the most abundant compound of the essential oil of Calaminta nepeta. And after analysis of this biotransformed oil, and in comparison to original oil, we found 6 new compounds which were not present in original essential oil, and we, analyzing these products, we came to the conclusion that these are products of biotransformation, and all of them, like I said before, was in accordance with literature data because they possessed OH groups or further oxidized keto groups to the keto groups like we expected. Also, thymol was also biotransferred to its 8-hydroxy product or benzyl. It was hydroxylated in benzyl position. These results were published in Nature Product Communications in 2018. Now, when we know what to expect, we try to biotransform perotetin F, and we got 3 new products after biotransformation. All these 3 new products were in accordance with literature data because in first product we obtained a hydroxylated product in benzyl position to B ring, also hydroxylated product with one benzene ring less, B ring, and also we obtained a product with sulfate group. I didn't mention some aspergillus species also can transform hydroxyl group to sulfate group in some compounds. So here is presented proton NMR spectrum of compound 2 with characteristic signal of proton from position 8, which is neighboring to hydroxyl group, and its chemical shift is characteristic in proper position. Then, according to also carbon NMR spectra, we saw that now in this product of perotetin F, we can see this carbon, which is now oxygenated. And also HSQC and HMBC spectra helped us to resolve the structure of this new compound, where also high resolution mass spectrometry confirmed the exact mass. These are 1D NMR spectra of compound 3. We saw immediately that there are less signals in proton and carbon NMR spectra due to missing protons and carbons of B ring. And after analyzing 2D NMR spectra, we confirmed this structure also by high resolution mass spectrometry. Compound 4 was very similar to original perotetin F, but we saw that protons of C ring 3' and 5' are very shifted to lower fields or to higher chemical shifts. And according to literature and also with very big help of mass spectrometry, we confirmed that sulfate group is present instead of hydroxyl group, and we successfully resolved this structure. Also, HMBC spectrum confirmed with all correlations that this structure is proper. Another story is about Marchantia polymorpha, another liverwort, which can be found, like you can see, as you can see in some small rivers, like big carpets. This is a zoomed photo where you can see the talus of this liverwort. And this is one, I will say, dangerous animal which tried to stop me to collect this liverwort. This thalloid liverwort was known for its antimicrobial, anti-tumor, anti-inflammatory, and diuretic activities. This is the monastery from the 13th century, near which was the small river where I found this liverwort species. After analysis of methanol extract of Marchantia polymorpha and in comparison to pure Marchantia A, we saw that aromatic part of this methanol extract is almost the same like this of Marchantia A, This is zoomed proton NMR, and it's easily to see that in this methanol extract is mershantsin A present in very big, in very high amounts, and it can be up to 60 grams per kilogram. So it was easy to isolate big amounts of mershantsin A, and also it is important to say that mershantsin A is also present in other Marchantia species like Marchantia paleacea, plicata, tosana also. And also beside mershantsin A, in Marchantia polymorpha were also present other mershantsins like mershantsins B, C, D, E, F, and G. Mershantsin A is maybe the most known bis-bibenzyl. It possesses a very wide range of biological activities, and the best known are cytotoxic activities against breast cancer cell lines. You can see that mershantsin A efficiently induced inhibition of growth of MCF7 breast cancer cells. Also, this compound isolated from another Marchantia species, Marchantia marginata, subspecies tosana, induced apoptosis in MCF7, the same breast cancer cells. Also, mershantsin A isolated from Marchantia polymorpha from Iceland induced reduction of cell viability of breast cancer cells A256, MCF7, and T47D. We know also that lipophilicity is an important physicochemical property in drug discovery. It influences several stages of drug action, including transport through cell membranes, binding to the target, as well as absorption, distribution, metabolism, excretion, and toxicity properties. In accordance with this, it has been reported that increasing lipophilicity of various target molecules resulted in better activities. So in our investigation, we did chemical and biocatalytic derivatization of mershantsin A. We obtained 7 ester derivatives in 2 ways. We did chemical derivatization by and obtained 3 totally esterified molecules or peresterified products, while enzymatic method using enzyme lipase produced regioselective monoderivatives, IME1 to IME4, and antiproliferative activities of all prepared derivatives, including original mershantsin A, were tested on MRC5 healthy human cells, but also A549 human lung cancer cells and MDA, MB231 human breast cancer cell lines. You can see the schemes on the left side, the schemes of these reactions. By chemical esterification, we obtained peresters, but lipase enzyme produced only monoester derivative in position 11, and hydroxyl groups from catechol moiety stood unchanged. So here are some data about yields, and after investigation of bioactivity and comparing to mershantsin A, we saw that monoester, monohexanoyl, as you can see, derivative displayed the same activity against breast cancer cell lines, but selectivity was significantly better. Also, other monoester derivatives in ME1, ME2, and ME4 were very similar in activity, a little bit weaker, but also they were much better against healthy cells. So in each case, selectivity was better. These results are published this year in Phytotherapia journal. At the end, I will present the story about bis-bibenzyls in higher vascular plants, because as I said in the beginning, bis-bibenzyls are characteristic for liverworts, but from 2007 they were found in some higher vascular plants. For example, in Primula species, where Russian scientists found in roots, in extract of roots and rhizomes Ricardin C in Primula veris subspecies macrocalyx. And also they reported that Ricardin C was present in significant quantities. In our laboratories, we confirmed Ricardin C. We have isolated this compound from the same sample from Russia or Siberia, this part of Russia, but also from iron. And we also wanted to confirm the presence of these biblins in Serbian species, Serbian Primula species, like it was Primula veris subspecies veris, but also Primula acaulis or Primula vulgaris. And we did it. We found in the highest amounts Ricardin C, but also two new compounds, 8' ketoricardin C and 8' hydroisomercansin C. This is the photo of Primula veris subspecies veris from mountain Zlatibor from Serbia. And here are examples of NMR spectra of these new molecules. First, this is 8' keto Ricardin C. Its proton NMR spectrum is on your left side, and carbon NMR on one part of it on the right side. Also, HMBC, like in previous cases, helped us to resolve this complex structure and very important correlations with carbonyl of keto group, of course. Also, these bolded correlations are proved the Ricardin type compound. As you can see, correlations from H14 prime to H11 prime to C14 and H13 prime to H13 to C12 prime. Also for 8' hydroxyisomercansin C, on the left side is proton NMR spectrum, and on the right side also its aliphatic part with characteristic 8' proton, which is near hydroxyl group. So because of that, it's shifted towards higher chemical shifts. And carbon NMR spectrum is presented on this slide with also characteristic 8' carbon now shifted because of the presence of hydroxyl or oxygen in this position. COSY and noisy spectra helped us to resolve this complex structure as in previous cases. And maybe the most important were HSQC and HMBC spectra, which helped us to resolve these structures. At the end, according to all these data about 8' hydroxyisomercansin C, another structure was also can also fit well. We didn't know if this bridge between C13 and C10 prime or C13 and C11 prime, because this hydroxyl group can also be present in 10 prime position, and every correlation will be almost the same. And because of that, we did methylation of our isolated compound, and we want to make these methoxy groups in these positions in these hydroxyl groups on benzene rings, and to find noisy correlations with protons in C12 prime position and C6 prime position. And after derivatization in alkaline solution by methanol and after reflux for 2 hours, not so complicated reaction, we obtained dimethoxy derivative, and noisy correlations confirmed our proposition that OH groups were present in position C1 prime and C11 prime, and so confirmed our proposed structures. So at the end, I want to thank all my colleagues which took part in these investigations, Dr. Jasmina Nikodinovic-Ironic from the Institute for Molecular Genetics and Genetic Engineering, Professor Dr. Igor Opsenica from the Faculty of Chemistry, University of Belgrade. Their teams were very effective, and together we did a very good job, and I'm very thankful to them. Also, I want to express my gratitude to Dr. Danka Bukvički, Associate Research Professor, Faculty of Biology, University of Belgrade, Serbia. And also, I want to say big thank you to Professor Dr. Karin Valant-Večera, Institute of Botany, University of Vienna, who provided us the samples of Primula species from Russia and Iran. Here is one photo from plant collection, my professors and me, and also to Professor Vlatka Weiss and Professor Slobodan Milosavljević, who are retired now, and my mentor Vele Tešević. Also, I am very, very thankful to Yoshinori Asakawa, professor from Japan, who opened me this beautiful world of liverworts and Bisbee bacills. And for the end, thank you very much for your attention.
Okay, thank you, Dr. Miroslav, for the very nice presentation. I would like to pick up 2 questions from the participants.
Sorry?
Participants, participants, if you have questions you would like to ask.
Yes, please. Yes, please.
Okay, I will ask one question. Like I have one question I wanted.
Yes, please.
Dr. Miroslav, you talk about Bisbee benzoyl from this lower natural plant, and also show lots of its interesting property on the cytotoxicity, antioxidant, antibacterial activities of this particular compound. So, to what extent, I mean, how much large did you want to be able to extract such type of compound from the this liverwort plant? Because it's going to have a chemical importance in this medical applications or any kind of cancer treatment. So how much we can extract it? Is it like cost effective? I mean, I have lots of money to extract this compound from the liverwort plant, or like is this like you can?
It depends from species to species. Marchantia polymorpha, for example, is very wide distributed, and preparing extracts from these species is not so expensive, and I think it's very cheap. So it depends of bioavailability of these species, but these species, what I talked to you about, are very distributed, and it is not a problem to make not so expensive, not expensive at all, extracts from these liverworts.
Thank you. Thank you very much.
You're welcome.
If then, let's move on to the next speaker and last of this technical session. So we have Professor Kumar Sharma from Shivaji University from Maharashtra, and he would be talking on topic laboratory to society, role of nanoscience in nanotechnology. I'm briefly introducing Professor Sharma. Professor Sharma received his PhD from Pune University from the Department of History in year 2003. He did his postdoctoral research work from University of Paris, Switzerland, France, and also he visited University of Rochester, USA as an NIH research associate, and he was also visiting research at the Radiation and Mixed Stability Unit, a medical research council UK at Oxford. His research includes photochemistry, engineering nanohybrids for photocatalysis, biosensing, environmental pollution, remediation, multifunction hybrids, energy storage and production. There are many applications in working on it. He has published several research journals in the very reputed SCI journals, and he also acquired more than 3 patents. Currently, Professor Sharma is presently a Deputy Director as well as Associate Professor, Affiliated School of Nanoscience and Technology, Shivaji University. welcome Professor Kiran Kumar Sharma to share his presentations. Thank you.
Thank you, Professor Rakesh, for the nice introduction. And at the very outset, I would like to thank the organizer from Manipur Technical University and the KKT who had organized this conference during this crisis time. And I'm really lucky to present some of the works that we've been doing. And I'll try to finish on time, although this thing, and I'll run over as, when we do something, when we want to produce some product to the market, we need to go step by step, and it is, we cannot just jump. We cannot do a long jump or a high jump. So that's the story that I'm going to be telling you, that basically I'm from a chemistry background, and I was not knowing what is nanoscience and nanotechnology, but somehow with my career, as I progress, I could get into this nanoscience and nanotechnology, and that is what I'm going to be telling you as a story. Okay. So many of us know, and already all the expertise have already talked about what is nanoscience and nanotechnology. And the only thing that I would like to add up here is I like this figure where, nano is at the center and it's the star. So that's nanoscience and nanotechnology is going to be the future of science, because it is both inter as well as multidisciplinary. And of course, we are dealing with something which is very extremely small, and which is in the range of 1 to 100 nanometer in the scale. So that is what amazes us, and the philanthropists and the physicists, the men the scientists who said that there is a large room at the bottom. That is what we are now trying to build now. So basically the properties, everyone knows that as the size decreases, like it's just like when you take an atom, the atom may be stable in terms of chemistry or physics or biology. When you take an atom, the atom may be stable, but when you try to break down the atom into particular electron, neutron, proton, and nowadays the physicist knows better that all these proton and neutron and electrons are no more fundamental particles. So when you break it down, then their property changes drastically, and that is what exactly what we want to see with the material, and that is why this is so much of an demand. Now there are many applications, and basically in nanoscience and nanotechnology, there is little room for discoveries. It is just like we innovate the basic fundamentals, and we use them and then apply them to these small materials, and we try to find out what is the fundamental behind all this behavior. Then only we can kind of make things which are very functioning very promptly or very efficiently. And these are some of the innovative application of nanotechnology and nanoscience. I always say nanoscience and nanotechnology because without nanoscience there cannot be nanotechnology. So it is both this thing. Sorry, there was a phone call. So today, I will be mainly focusing on nanofabrics, where during this COVID lockdown time, I was able to do some kind of research, and then I was able to bring up a product, and that how I came about it. I will be talking on that in the later part of my talk. So nanofabric is one of them, and I'm also working on environmental use of this nanotechnology, where we are fabricating some kind of silver-based material where you can use as a filter for filtration, and that's what I'm going to be talking about today. Also, that's already in the stage of proof of concept, and we are now going ahead with certain things. And today I'll be disclosing a little bit about that also. So where are we in terms of nanoscience and nanotechnology? So this is the latest table that is being shown, and India is third. Now, when we say that we are in third position, it is basically in terms of articles. So these articles are no more, no more accepted in like in the current or in the coming up future education policies also, as we have seen that the new education policy has been now implemented, and the universities and the institutes are more focused on trying to do translational research than rather than publishing paper and so on. So basically, although we are in third in number in terms of publication, but when we come into when we question into how much this research is being translated into for the use of society or for the development of the nation, there are not much. So that is where the vacuum is, and that vacuum has to be fulfilled by all the youngsters and the scientists who are working day and night, thinking that we should develop, India should develop into one of the hub of nanoscience and nanotechnology. So what are the challenges that we face now? Well, we like R&D, research and development, and also indigenous companies who are really willing to put money down there. For example, if I take a example of a country which is very small, like South Korea. South Korea, actually, all the university and all the research institutes are partially or fully funded by what LG or either Samsung. And these are all dealing with nanoscience and nanotechnology. Now, in our case, the Indian companies or industries, what they think about is before investing, they think about, okay, in 1 year, how much I should gain. And this, I guess, I know yesterday Sonamani was there talking about the business, and I'm sure that is what it is. And I guess the Indian industry needs to change and then invest some money onto this field also, because this is really going to be the future. Then there are still the knowledge of this potential of nanoscience and nanotechnology in domestic market is, it's quite lacking. So it's towards a very minimal. And then, of course, there is a scarcity of translational research. And we know as a faculty, and we has been involved with the university and the research institute for a long time, of around 17 to 20 years now. We understand that when you use the word nano before the project, you get the project, right? But now it is very difficult to get the project unless you try to prove that, okay, it will be useful for the society. So things are changing, but the pace is quite slow, so is to say. And therefore, the translational research, like whatever you do in the laboratory, you're not able to make it contribution towards the society or the government. So that is exactly what we all should think about, that whatever we do, we should try to translate into some kind of useful thing which will be useful to the government or society at large. So here I would like to show this slide where I would explain how we can go about the translational research. to say that, okay, how we say that we always, most 99% of us will say that, yes, we want to do the translational research, but we do not have the facility, we do not have the things and all these things. So all the excuses and all these things we have. And of course, that is true also. But however, the government also has a limitation of providing the fundings. And the fundings cannot be, if you want a FESM everywhere, it cannot be funded. So that is how it is. After all our the tax that we are paying, the government is giving a part of it to the research and development. So we should also think about it. And when we see these three wheels, I'm sure that I would be able to somehow convince that critical thinking is we experts, so as called the we researchers and the experts, we need to know how to do critical thinking. That is one of the major thing, because without this critical thinking, there cannot be connectivity between the the institution or the government and the grassroots level student. So that is what the yellow wheel is basically the individuals, and these individuals consist of UG, PG research students and young faculty. And of course, it can be a common people who wants to be an entrepreneur. So individual means any individual. And then the second part is the organization. The organization includes such as university and the experts and all these faculty, staffs, and technical engineers, and everyone who have at least contributed something in their field very seriously. And institution means the like in our case it is UGC, or the NCL, it will be CSIR, and then so on. So there are the institution which actually controls them. So when the individual moves, and if the organization doesn't move, then the institution is not going to move. So now the thing is, both the individual and the organization, when it moves, then only it becomes easier for the institution to move ahead. And when the institution moves, it means the country is progressing. So when it is stagnant, then so now how to make this things work? So this is where the critical thinking plays an important role. And this critical thinking includes reasoning and problem solving and evaluating. So this is what the experts and the scientists or the research people who are actually putting their thoughts as to do, right? Now, in this critical thinking, what we should be careful or what we should have is, we should have societal issues which are society, so we can see, and then local need based. This is what we should be focusing on, and that these two are nothing but we can solve this by what we call as remembering and understanding. That means there includes our what we call as fundamental education, the education that the fundamental principle of science or either humanity or any kind of things that you need to know. So when you want to identify societal issue or local need-based problem, then you have to use what we call as remembering and understanding thought process. Then these stakeholders are nothing but these UG, PG, research students and young faculty. Why? Because they have been taught the fundamentals during their colleges and during their graduate times, and then during their research time they pick up something, and because of that knowledge they should be able to reason, okay, the society is having such problem, is there any way that I can contribute? And that is where what we call is finding the problem for a research. So that is exactly, the research can be science, the research can be social issues, the research can be of economy or anything. So that is what it does, and that is at the lowest order of skill. So the skill, and why we say today that the school has to be very prompt, and they have to be very, there are bad schools, there are good schools, if that some teacher doesn't teaches properly the fundamentals and all these things. So we are concerned about the school system, because if you do not train the students, the youngsters, which is the lowest level of skills, if you do not equip them with the enough skills, then what they will do is they will not be able to identify the problems that is they face, or the issues that are coming in the society. So that's going to be a difficult thing. So we need to be very realistic. Then the second higher rank skill level is problem solving. Once you find out the problem and the issues, then it should be solved through the problem. The society problem is enlarged. So basically, you have to design syllabus or curriculum, and also you have to kind of schedule some training session, so that these students or the young faculty are exposed to, with the initial idea, they can further go ahead and solve the problem. So that is how it comes into the second higher skill. Now, once this stakeholders includes like me or any other scientist, or any people who are faculty or who are scientists in any field of science or arts or anything. So they also include industrialists, because if an industrialist doesn't know what is the problem that the industry has, then it is never going to be solved. So that is exactly. Now, the third and last part is what we call as evaluation, and this evaluation is done on the basis of performance and compatibility, employability, adaptability, progression. So who is going to be monitoring this? There are stakeholders like industry, linkage institution, and organization. Because if you do not have the capability to do anything, or the industry is not going to like you. So the thing is that in our system, we should have this harmony and the wheel that is rotating, and this critical thinking has to be put major emphasis, so that we give the best, and we give the best to ourselves as well as to the society, by bringing up youngsters in the right way of thinking. So once those who are successful in that, then they create. And when we create, this creation doesn't get lost. It helps the institution. So that is exactly what capacity building is. So in the capacity building of a nation, then critical thinking is an important parameter, and we are the middle wheels. So we should be able to teach whoever is a teacher. We should be able to disseminate the knowledge and the principles very thoroughly, so that the youngsters develop themselves to higher skill levels, and someday they can create. And when you create, you are helping the government. So this is what it is, and my journey started like that. So I was, after my PhD, my MSc, I joined the UGC BAE Kolkata Center as a JRF, and then fortunately I joined for my PhD in the Pune University in chemistry department, and somehow I was lucky enough to be abroad for 1 year during my PhD time, and this is how it kick-started. So I worked basically worked with azodized degradation, and we were the first people who actually identified that this azodized react, they are different tautomers. They have different tautomers, and these tautomers react differently with different, very reactive species such as hydroxyl radical and so on. So this is the beginning of my journey in the research and the academic field. Next thing was that after my PhD, I joined this Department of Biotechnology, where I learned the skills of biology. So I was also interested in biology, so I took the offer and I joined as a research associate in Department of Biotechnology. And here I could get all the knowledge of microbiology as well as molecular biology. Although I'm not very expert, but I do have a hand in this. And luckily, with my analytical skills that I have developed during my PhD time, I could get a US patent from here. So that is my patent. And then the patent had nothing to do with anything, any new idea. But as I said, my during my boss was actually, he was looking for some very commonly used dye, whether it can actually stain protein or not. So in biochemistry or molecular biology, when you do a protein staining, you have to use Kumasi blue. That's a very expensive dye. Now, my boss, during that time, he thought this is what we call as innovation, that the women in other than Manipur, in all over India, during various or kind of function time, they actually paint their feet and the hands with a red color, and that red color is called as alta, and it doesn't move away. So the basic thing that he discovered, he thought, well, it doesn't get removed easily from the hands and the feet. That means what? Hands and feet are nothing but their cells, and these cells contains protein. So his idea was that why not I use this alta instead of Kumasi blue, and we got success, and we published a paper as well as we filed a patent, and the patent is still out there. So for alta, and we compared everything in that. The second part was on the cell culture, where you know, like I work on protein synthesis, and this which is related to heme-regulated eukaryotic initiation to alpha kinase. That is where so it was being fed with some kind of iron, and you can see the change in the color of the cells. So these cells was colorless, but then it develops color. So that is where the heme binding is actually initiated by the iron that we feed in. So that is how we estimated how much iron is optimum for such kind of initiation of heme binding. So that is the second part. And the third part where I spent my long time was in the University of Rochester, that's New York, where I studied this very thoroughly, the direct effect of radiation on DNA. And this DNA was, the model was like synthetic oligonucleotides and the plasmid DNA. And from where, like we could establish that the direct effect can induce base as well as sugar damage. You know, like the biochemistry and molecular biologists, they may be well aware of what we call as strand breaks. So what we did was using analytical techniques such as GC and SPLC, we were able to elaborate a huge number of mechanisms on how this direct effect of radiation can harm the DNA. And we also compared with the real DNA, like plasmid DNA, which is PUC or PBR222 DNA. And so we had this couple of papers coming out of that. And then after I came back to India, I started my career. Means I was having an intention to work on nano, and somehow like nano catalysis became one of my favorite topic, and that is how I went to nano catalysis, because there were so many. I had my training in kinetics, and this actually was one of my opening field. So the reports were that size and shape actually can influence the kinetics of any catalytic reaction. That's what it is, and that is from where it is. Now the problem was that how do I get the nanoparticles in a stable form? We know that nanomaterials are not very, they are very high surface energy, so they try to agglomerate unless you kind of stabilize the surface. So basically, in order to get the very uniform nano materials, you need to choose the right synthetic technique. Otherwise, the problem is you will never get the reproducibility. So that is why I thought about it, and there are templated methods. One is what we call as hard template method, and another one is soft template method. Well, hard template methods basically has got disadvantage, because while you remove the nano material from the hard template, you need etching by very strong acid like HF and so on. So basically, I landed up with the soft template method, and here comes the role of my missus, my wife, who was also working on this, and I could learn all these things, and we have been collaborating with her to do all these things also. So this soft template method, during our, I remember during my physical experiment time in practical time in college and university, we used to do some kind of titration where there was a surfactant, and we were to find out the critical CMC, what we call as critical micelle concentration. So basically, that becomes now the hand to understand this soft template. And this soft template are basically made out of this surfactant, because they, with concentration and with the oil and the water mixture, co-surfactant ratio, you can tune this the micelles, that the size of the micelles that is being formed. And that is what you can see, that you can tune into hexagonal, cubic, or lamellar. That means what? You can get, with the soft template method, you can get these three forms of nanomaterial. So that was the most interesting part where I dive into this. So the technique, well, I actually kept my radiolysis technique, because it is the most clean. The nucleation is clean, and the homogeneous distribution of nanomaterial is out there. Since I am basically a radiation chemist, so I knew this technique, and it came handy to me to progress in this. And these are some of the basic fundamentals of radiation chemistry. And this, if you take any solute in the solution, aqueous medium, now water is around 50 molar there. So all the energy is being absorbed by the water. And this water actually breaks down to reactive species. And these reactive species can be interconverted or scavenged out. And so you have a very clean system where you can react, you can study the reaction of your desired radical, whether it is oxidizing or whether it is reducing with your solute. And that is one of the finest way of studying this. Now, the first thing that we tried was we synthesized some porous platinum nanostructures, and this is how we did it using the soft template methods. And you can see the TEM image that, this is a bowl. It is around 60 nanometer in size. And then this is some net, it's a broken bowl. So with the dose of irradiation, how much time you irradiate on the gamma source, this transforms. So basically, that means what? Even with the structure that you get, you can also still manipulate using the radiation dose. So basically, that is one of the basic fundamental basic playground for us, like for scientists like us. So that is how we got it into it, and we studied these nanomaterials for electron transfer reaction between hexacyanoferrite and sodium thiosulfate. And the model, the very generally used model, paranitrophenol, conversion of paranitrophenol to para-aminophenol. And also we did some Suzuki reaction also. And other than that, we also did the conversion of chromium 6 to chromium 3. This chromium 6, they are released from tannin, like leather industries and many other industry, and they contaminate the water, and chromium 6 as such is carcinogenic. So whereas chromium 3 is not non-carcinogenic. So therefore, whenever there is a need to basically convert this chromium 6 into chromium 3 before you do. Then we also did with some of the dyes, which this is a general research that I do carry out. And most interestingly, we also study the interaction of intermediates, reaction intermediates with nanoparticles. And that is where we took this amino acid called tyrosine, and we reacted with OH radicals using radiation chemistry. And then we introduced gold, and then what we saw was that there was a what we call as gold nanoparticle tyrosine complex that is formed, and because of that, you get one of the reaction faster, and that because of that reaction being fastened, what happened is it reversed the product formation. So that was very interesting, in fact, and it got just published in this year. So now coming to the real life. So this was my, normally this keeps going on.
Hello, Professor Kiran. Hello. Hello, Professor Kiran, are you still there? I think there is a loss of electricity or something.
Yes, I guess Professor Kiran has some problem with the connection. Yeah. So, should we wait for him to join it back, or what should we do? Yeah, yes. I
think if there is no electricity, then there will be no chance.
Yes, yes. So can you please, call up him, and then if he can rejoin or he cannot, then we can decide what to do.
Yeah. In the meantime, I will share some information here. Actually, it is raining very heavily in Pune right now.
Okay.
Professor Kiran is also staying in Pune, just about 1 or 2 kilometers from here.
Okay.
So, let's continue.
Okay.
Professor Rakesh.
Let's go ahead. And
yes.
Okay, then since there is some technical issue, so we couldn't finish Professor Kiran's talk. But indeed, it's a very nice and a very interesting talk. And even, I guess everyone must be enjoying. So this talk concludes the technical session of the last session of this conference, KHUMANG 2020. And indeed, it was a very nice, very interesting presentations covering very wide ranges of science and technology that has been, engaged in different parts of the world. I will just summarize about the talk. So Dr. Niraj, we had started with the doctor's talk where he various aspects of cost-effective colorimetric sensor for arsenic detections using luminescent nanometers, and he also discussed about development of these nano gates and instant blood bandages and etc. It was very nice. And Dr. Geetgovind discussed about wastewater treatment using this filtration process.
In the meantime, Dr. Kiran is back.
Yeah. Okay. Okay, then he can continue. Yeah, before I conclude this, please.
I think because of the lightning, can you continue?
Yeah, because the light went and I, yes, I'll continue now. Okay.
Yes, yeah, please.
Yeah. The light went and my university, this thing, Wi-Fi also went. So basically, all right. So let me continue. Try to. So am I audible?
Yes. We were stuck on this slide.
Okay. I'll just do the slides. Yeah. So yeah, somehow like it was here. So we come to some of the useful research, and Niraj, my internet is sometimes unstable, so I don't know how far it will go. But yeah, let me try to finish it up. So one of the thing that I want to go for application was how to make, how to incorporate silver nanoparticles on materials so that I can use it for a filtration purpose. And that is what basically I demonstrated. Why? The reason why I choose for water purification is because we know that water and almost due to the lack of sanitation as well as proper hygiene. And according to this World Institute, safe water is an indicator of development of the country. And if you look into India, then there are about 100 million of people who lives under area of poor water quality. So they need to have at least some light. That is the reason why I choose this. There are challenges for these traditional disinfectants, and we need a robust and new method for water purification at lower cost, minimizing the chemical and the environmental impact. So that is one of the reasons. Now, in normal process, we have a water source, and then this population takes place by mixing with alum and certain other sedimentation occurs, and after sedimentation we filter it, and then it is being disinfected. Here, during the disinfection stage, we either use chlorine, ozone, or some UV light to actually kill the bacteria. Now, after this process, it goes in there. Now, the challenges is that when you use these chemicals, the traditional way of killing chlorine, then there are disinfectants byproducts, what we call as TPBs. So these are very dangerous to their health. What our aim was to develop something which we avoid this. That means there's the bacteria should be killed by some nanomaterials. Now there are many water filtration units that are available, from advanced to very low cost water purification system that is available in the market. Right. So one of the most interesting, exciting water purification system is that is being used in NASA in the space. So they have actually used this in their space program, and these are areas of single piezoelectric effect. It's the slight supply of effect actually induces water molecule, only water molecule. So therefore, this is the purest form of water that you can get. And there is a technical difficulties in arranging those carbon nanotubes fibers into array. And of course, there is another thing that in space you don't waste any waste survive. So the other thing is, in India, we have come across those people who are associated with NCL. It was being, it's a byproduct of, and he based this on the rice husk. He incorporated the silver nano.
Yes, Niraj, can you?
I think he is gone for the final time.
So what should we do? Because
yeah, let's continue because his light is gone and he's using the Wi-Fi. So I think he's again
because we're running behind the schedule. So I think he comes back again.
He comes back.
So I request Professor Kiran, if he could conclude his presentation, the final words, that would be very great.
Okay, I think that that is what I have to do, because I think this is
Professor Kiran, you can stop your video. If the bandwidth is low, then it sometimes works.
Okay, what I'll do is I'll stop
your voice and
video.
slide only.
Stop video. Okay.
Yeah.
Now, yeah. So this is what it is, and there are in market there are this available.
You are not sharing the screen.
Okay, I've stopped sharing the screen. 1 second.
No, you have to share.
Where it has gone? Yeah, somehow. Okay. All right. So I'll run over it.
Yeah, it's coming. It's coming.
Okay, it's coming now.
Yeah, I think it was very interesting, but yeah, we are running behind the schedule, so I would suggest, everyone.
So basically, so silver is one of the material which is universal, which has got antimicrobial property. So what we did was we incorporated in what we call a sabudana. Sabudana is a staple food that is all over India. Of course, in Manipur we don't eat it, but then in all over India, sabudana is one of the staple food. So we did all these processes like carbonization, and then trying to estimate how much this thing silver is there, and this is the process tested this. Now this is the pictures that we have taken, and we know that it is being incorporated, and this is the carbonization process where we saw that, okay, within certain times, if you don't do the carbonization, then there is a problem. So this is one of the, without nanoparticle, and with the nanoparticle, there is a change in the color. And this is the HRD, where we could get the AGMPs are embedded inside the sabudana powders. And this is how we did our filtration. We use a very small syringe to go ahead with our this. Okay, yeah, so light has come, I guess. I will be able to. Now, here I have given all the specification here. 7 gram of carbonized sago silver nanocomposite we used, and then it was packed and it was done in a biosafety unit. Now here are the results. So we take some few 30 to 50 microliter of whatever it is, the filtrate, or where we have spiked the bacteria, and we spread it on what we call as TSA, that is the nutrient agar plate. And what we saw is A is basically normally the control water, whereas B is the spike water, and C is after filtering. So we see that there is in the filtrate as well as in the A. So that means whatever the bacteria that is being spiked in is being removed by this. And not only with the distilled water we did, we also went for a real dosing, like how much bacteria it can tolerate. So we went up to 500 microliter of bacteria, which in terms it is, like 1 ml of bacteria contains about 10 to power 6 colony forming unit. So you can imagine that 500 microliter contains almost 10 to the power 3 colony forming unit. And we saw that it can actually get rid of the bacteria, how much you load. Now we go into the real water samples, and we take 3 samples from nearby ponds, and these are the names that we have. And what you see is when you do the filtration, you do not see the bacteria out there. So that means that the silver particle that is inside is killing the bacteria. And also one of the requirement for filtration or potable water is MPN test. And this MPN test is basically, it should be, as per the WHO, it should be below 100. And now when we do the testing, what we see is that the upper part, there is a coloration, different coloration. That means there is a lot of bacteria. And whereas in the lower part, what we see is that this is done in a triplicate, and you can see that there is the colors are almost the same. That means after the filtration, you do not see any bacteria out there in the lower part, whereas before the filtration, the different coloration gives you the, the number of bacteria. And this is the table which we present. And as I said, according to WHO, the MPN should be, for the potable water, the MPN should be less than 100. And here we get, after the filtration, we get 4, 1.8 and 7 counts, etc. So now, again, another problem is silver leaching is a problem, because it is sometimes known to be toxic. And this toxicity, basically, we have to see that we are under WHO guidelines, and this is how we did. And we show that the silver leaching out, from the sago silver nanocomposite was very less. So this is where I'm at the stage of somewhere around what we call as TRL 5 to 6, and this I'm waiting for some company to pick up this and then to go ahead and then fabricate the things. So this actually will help people who are facing natural calamities such as flood, where there is scarcity of potable water and you have to deliver water. Here I plan to make some kind of tank where all these essential parts are there, and you just need to pour in and the potable water will come out. So that is the future ahead of this. The second part is dependent on the what we call is development of fabrics, nanofabrics, where we basically deal with antimicrobial fabrics, and this center is being granted by Rashtriya Uchchatar Shiksha Abhiyan, RUSA, and we got around more than 1.25 crore from this center after a hard work. And we propose this because this nanofabrics is one of the upcoming technology where you can use nanotechnology for all this field, and one of them is antibacterial, and that's where we focused on. And the rationale behind is that in Ichalkaranji, which is near to the Kolhapur, is known as the Manchester of India, and it has got around annual turnover of 8,000 to 10,000 crore rupees. So basically, there was a chance that if we develop a technology, then it can be incorporated. That is how the reason. And when you look worldwidely, you see that by 2020, you have almost 800 US million dollars as this thing. But with this pandemic, what is happening is the medical part, transportation part, protection and military, all these things are now coming into this increasing because of the demand of antimicrobial and antiviral protection kits. So this is how we established that, okay, we can embed silver nanoparticle into fabrics and show their antimicrobial activity. So as we did also selenium, and this selenium can be also done, and it shows a very good antimicrobial property. And over the time, this eco-science approach us for product development. And this is where our breakthrough came with this expertise. Now, the problem is that, with this COVID-19, the causal virus, which is called as SARS-CoV-2, this actually is spreads out from droplets, from coughing and so on. So these are called as aerosol, and these aerosols can stick to the fabric and the virus can remain there. So basically it becomes a mode of transmission, both in the hospital, for the corona warriors and those who are going out daily for certain things, So basically this is how we can address, that was the challenge that we had. Now, that on the surfaces, this SARS-CoV-2, how much time they can survive, and these are all hard surfaces, including plastic, which we are developing something for this also, but it is at the early stage. However, with in terms of paper, tissue paper, wood or cloth, glass, etc., you see that the virus can almost remain for around 4 to 7 days onto clothes. So that means if you are wearing a mask and going out to a contaminated area, then what is happening is the virus may be sticking onto the surface of your mask, although it may not enter your body, but then you have a chance of contamination. And if you do not disinfect or wash, then what is the possibility of virus contracting the virus is very high. So basically, how to do that? That's what it is. So there are many chemical or nanomaterial-based things available, and these are the mode of action and the limitation, which I will not be going into detail because of the lack of time. And this is how we plan. The plan was that we want some eco-safe, which are environmentally benign, EPA approved material for this antiviral property to be exhibited. So that is what we plan. And these are the ingredients that we use. This is citric acid and benzyl conium chloride. So these two ingredients, along with some other binders, we formulated a spray, and that liquid spray we tested with the agar well diffusion, and we see that in this, we see that there is a zone of inhibition. And the liquid is actually inhibiting both the types of bacteria, gram-negative as well as gram-positive. And then we sprayed it spray onto fabric. And what we did was we used what we call as JISL 1902, which is a Japanese protocol for antimicrobial testing for fabric. So we got all the issues sorted out, what are the bottlenecks with this protocol, and we sorted out that thing, and we were able to do demonstrate successfully that whatever the liquid that is being developed by EcoScience and Shivaji University was showing antimicrobial activity greater than 99.9%. So that is what it is, and these are the table that we there are many fabrics that we have tested, but these were the successful ones, and we can see that even at 5 minutes it can deactivate the bacteria. So this is one of the interesting part of imaging, and you can see that upper panel shows the same image of the uncoated but loaded with bacteria, and you can see S. aureus, which has got grapes like berry colony that hanging out there. So that is one of the most beautiful picture that I could get. Whereas in the treated one, you do not see this, but you see all these small particulates, and there is a small nanolayer, we call it nanolayer, formed by this material onto the yarns of the cotton fabrics, and then that actually helps in deactivating the bacteria. So this is carried out in one of the biosafety lab level 4 categorized this thing in US, and you can see here that the fabric were tested with newly developed protocol, where people may ask me that why you didn't do in India, but unfortunately in India we do not have this technology to do on fabrics or hard surface, so we had to go to US to do this. And what we got the result was that it is inhibiting in one set, it is inhibiting around like almost 99.99+%, because what you see here is the blue, yellow, and the gray lines are the control ones, where you do not have the spray, whereas on your right of the screen, what you see is 020, those are the treated fabrics. So what you see is this SARS-CoV-2 virus, which is responsible for COVID-19, is actually deactivated to 99.99%. And this is how we think that it is. It works on when it comes into contact with the coated fabric. You know, the virus membrane gets dissolved into the layer of nano coating, and that nano coating can remain at least till the next wash. That is what we claim. But as per the, you know, when we claim, there is a reality here. When we claim, it doesn't get accepted. So there are FDA regulation in India, and we need to follow their guidelines. And even though you show many of your results, what they will say is you do this. So now in the product that is being developed, we only mention for 48 hours because of the FDA insistence. So there are many hurdles where bottlenecks, although you may be developing the product, but then till you get the license and the FDA approval, you cannot market, and that's why we delayed for almost 21 to 22 days in releasing this product. So finally, we could come up with this, what we call as antiviral, virus covers antiviral fabric disinfectant. Now it is being classified as a disinfectant, and it was being released in our university in the hand of our Vice Chancellor. And then I also had the chance to meet the Higher and Technical Education Minister of Maharashtra, where we presented all this gift, and now this is in market and you can buy this product in, with this link. So finally, I would like to acknowledge all these students who have helped me and my faculty colleagues. And of course, Dr. Niraj has been, very helpful, and we have been doing a lot of collaboration. And of course, the fundings are very, essential. And Creton Polymers, which is based in Texas. So this was the first one which we are trying to develop, hard surface antiviral coating by their polymers. And then, of course, EcoScience Innovation Private Limited, we have already gone to the product, and other, funding agencies of India. And lastly, with this, nice building, I would like to thank everyone for listening to me, although there was a stopover because of the light. Thank you for bearing with me.
Okay, thank you, Professor, for such nice presentations, and also discussing several aspects of roles of individuals, organizing institutions, and capacity buildings, discussing about his research on nanostructure fabrications for development of wet fields, and then also for national nanotextile for antimicrobial and antifungal activity here. So I would like to take one question since we're running behind the schedule. Just one question I would like to get it from the audience if they wish to ask to the Professor Kumar.
Yeah, Professor Rakesh, can I ask?
Yeah, please go ahead.
Professor Kiran, it was a very nice talk. Talking about your virus covers, the spray that you are using to destroy COVID-19, can you give a comment on how it was acting upon different bacterial strain? Because okay, it is effective on COVID-19 virus, but does it also destroy some other microbes or bacteria? Or it may be, for example, if you are having a spray and you are spraying it on a fabric or whatever, a surface, hard surface, maybe you have an intention to touch the surface sometimes. So it may have your spray may come in contact with the biological tissue that your tissues. So it may have, so if you give a comment on how it is acting on different microbial or bacterial strains, other than COVID-19, it will have toxicological, in that toxicological framework, we could have some kind of idea.
So that is, Nimrod, it's a good question, and yeah, it's a challenging task, you see. So therefore, when you choose when you want to bring out a product, yes, you should have the basic fundamental of what kind of material you should use, so that there should not be much of challenges when you try to produce as a product. So basically, as I said, that citric acid and BKC, we call it benzyl cholium chloride, they are actually US FDA approved, and they are considered as safe. Okay. So basically, that is one of the reasons. But again, when we went along with that, there is a bottleneck. So the FDA India has said that no, this cannot be done like that. So you have to prove that it is not harming the skin. So we did also the skin test also. So there are skin tests, and as I said, well, the mechanism, it is since it is a confidentiality, I cannot disclose how it acts. Okay, but
yeah, I understand.
Yeah, because it's a industrial work, and it's a market, and therefore that I cannot disclose how it. But then it basically it's a simple chemistry that it will dissolve the membrane of whether it is a bacteria, whether it is a this thing, or cell wall and cell membrane. If it is a virus, it will dissolve the membrane. Once the membrane is dissolved, then the RNA components cannot do anything, or the DNA or RNA component. So that is how it works. Yeah.
Thank you, Professor.
Okay, I understand that the participant made lots of questions, lots of queries. But, I suggest that you can take Professor Kiran because he can explain much better in details. We would like to, conclude this session because, as I said, we're almost running out of time, and we have a valedictory after this. So let's thank the speaker, Professor Kiran, for his valuable presentations and sharing lots of informative, which are beneficial for all of us. So, Niraj, since I'm running out of time, I will not summarize it because I have did it. So I will pass on to you, and thank you everyone for your patience, and also for for attending all the valuable talks. Thank you very much once again.
Thank you, Professor Rakesh M. You have excellently chaired the session. So I will just quickly summarize this session. We have got talks basically from sensing application to wastewater management treatment, and even to Make in India and how to improve the Make in India products, even for the biological world in the world of liverworts. And finally, I think the talk of Professor Kiran completely winds up the whole meaning of this conference, where you have an idea coming out in the lab and it passes on through various processes and finally coming to the market, and it is actually beneficial to the society. So let's close this technical session number 6. So, and lastly, I would like to thank Professor Rakesh again. With this, let's proceed to our valedictory function. I would like Sangeeta to take over from here. Sangeeta, you can unmute yourself.
Now, we come to the last part and the most awaited moment of the event, announcement of Young Scientist Award. We are going to select 5 young scientists. The KKT Young Scientist Award carries cash award of ₹5,000 and a certificate. For this announcement, I would like to invite Professor Kiran Sharma, Deputy Director, School of Neuroscience, Shivaji University, Kolhapur.
Okay, so thank you, Sangeeta, and thank you, Dr. Niraj, for giving me this opportunity. And I think if it was not online, then this would have been a tense time for all the presenters. And I know everyone has put in their best effort to their work and their presentation, but somehow there are some or other way. So everyone who presented was a winner. That is what I would say. But then, as a formality, from that winner, we have to choose out 5. And that was one of the most difficult tasks that we had, although it is online, but we had to really listen to them very carefully, and then we had to choose out that. So not numbering them, the first one, the first winner is goes to OP28, that is Dr. Nimrod Anand. So he's one of the 5 winner. The second one of the 5 goes to OP29, Sirom Mairaba Maiti. So he's the second one, and the third one goes to OP16, Dr. Sonia Verma. And the fourth one goes to OP24, Dr. Sanogzam Dhiren. And the last one goes to OP number 8, Yankhom Dhanapriya Devi. So congratulations to all the winners and those who were presented. And it was a really excellent presentation and a very nice data. Thank you, Dr. Niraj. I hand it over to you.
Thank you, sir. Congratulations to all the awardees. The cash award will be paid through online. So now we move to the concluding part of the day, the 3-day e-conference KHUMANG 2020. For this, may I invite Dr. M. Niraj Luwang, Senior Scientist, NCL Pune, Acting Chief Consultant, KKT Imphal, to kindly deliver the word of thanks.
Thank you, Sangeeta, and thank you, the organizing committee, for giving me a chance to propose this vote of thanks. Finally, we have come to the end of day 3 for KHUMANG 2020. KHUMANG 2020 is basically done to create a platform where one can come up with an idea, then come up with a proof of concept, then convert in the form of a technology and goes to the market and finally to the society. I think the last 3 days we were able to successfully fulfill all the aspects or all the aim and objective of KHUMANG 2020. We have done it with a variety of speakers from diverse research area and from different parts of the world. And on behalf of the organizing committee, I would like to thank all the speakers for their time and for sharing their research data, especially for the international speaker. Because of the difference in time zone, some speakers have to wake up at around 5:30 a.m. just to be a part of KHUMANG 2020. So, and I also like to thank all the persons who have shared their session for their excellent management skill and keeping the speaker and the audience in the loop. And finally, I would like to thank all the participants who have registered for KHUMANG 2020, and not last but the least, for the oral presenters who have taken the exhaustive step of making their presentation in the video format, which is basically a new way of communication. And I think it is the new format which is here to stay, and in the days to come, I guess this is the only format where we can have conference. Apart from this, there are lots of persons behind the screen who have taken a big role in making KHUMANG 2020 a successful one. Firstly, I would like to thank Professor L. Tombi Singh, Vice Chancellor of Manipur Technical University, for the approval and for the motivation in conducting this e-conference. I would like to thank Registrar of Manipur Technical University, which is also a TEQIP-III Phase 3 coordinator, Ms. Bidarani Ayekpam, for approval and also the funding the whole e-conference. And I would also like to thank the director of KKT for his visionary inputs. And I would also like to thank Dr. Swapna for staying during these critical times. And finally, I would also like to thank our Honorable CM of Manipur and Biren Singh for his support towards the conference. And lastly, if I have missed out anyone, I would like to thank all the persons who I am not able to mention here. And with these few words, I would like to share is okay, I would like to share a small screen here. Actually, KKT is coming up with a new campus at KIB. Right now, we are having our head office at the Airport Road in Imphal. The new campus is around 140,000 square feet. It will have the main office of KKT, R&D lab, incubation center, and all these things. So we need support in terms of moral support and any kind of support. So this campus will be launched by the end of this year. So I would like to share that this will be coming up preferably in the month of end of September or October. So let's hope for the best, and I will end up here.
Thank you, sir. The YouTube link of proceeding video of the KHUMANG 2020 will be shared to the mail ID of its delegate. Now, KHUMANG 2020, the second International E-Conference on Science and Technology for Society, jointly organized by KKT Imphal and Manipur Technical University, is officially closed. Thank you all the delegates for making KHUMANG 2020 a grand success, and see you all in the upcoming KHUMANG 2021.